Welcome back, everyone, to day 2 of R Cubed. So we're really excited for a brand new day of learning about, how regeneration happens. Can you guys hear me? Are we good? Okay, I see some nodding heads. So yesterday, the day was broken up into understanding different systems and what they require for regeneration. We learned about tissues that readily regenerate, tissues that maybe take a lot more. We discussed issues of scale and stemness and hurdles to achieving regeneration in different contexts. And then we also learned about the wonderful research organisms that regenerate and what lessons we can learn from them, how adult regeneration may be really distinct than developmental growth, and maybe how we can leverage these different programs to repair and regenerate tissues. So today we're really looking at how the whole is greater than the sum of its parts, and we've broken down regeneration into distinct components. And we're going to kick off our sessions with Dr. Amy Wagers and Dr. Kara McKinley leading the session on stem cells. So I'm going to turn it over to Dr. Amy Wagers from Harvard Medical School to take over. And actually, before I do, just a sort of logistical issue, I really want to encourage the audience to participate. Yesterday, the questions were fantastic. The discussion was fantastic. And so we want to keep that momentum going. And the other thing I really want to encourage you folks to do is look at the bottom of your Zoom screen and ask your questions in the Q&A if you're in the audience, because that really allows us to track your questions and address them directly rather than putting them in the chat. So go for that Q&A box and not for the chat box. So thank you so much. And I'm going to turn things over to Dr. Amy Wagers from Harvard Medical School, who's going to lead the stem cell session.
Great. Thank you so much, Shruti, and welcome to everyone. I'm Amy Wagers, and together with Kara McKinley, I'm really delighted to co-chair this morning's session on stem cells. So we have another fantastic lineup of speakers today who are going to address applications of both pluripotent stem cells as well as tissue-specific adult stem cells, drawing particularly on the hematopoietic or blood-forming system, which is arguably one of the longest studied stem cell systems and one that produced many of the conceptual frameworks like self-renewal and differentiation potential, which are applied now across many different systems. And so without further ado, I'm very happy to introduce our first speaker of the morning, Dr. Lorenz from Memorial Sloan Kettering Cancer Center. Lorenz, please go ahead and share your slides.
Perfect.
Great. So you can see that?
Yes, looks good.
Beautiful.
Okay, great, wonderful. So first of all, just also thanks for this introduction and for the invitation. I really very much enjoyed yesterday and look also very much forward to today's discussion. Hope it will be a lot of fun. Now, just to start off, I have to put the one disclosure slide because I'm scientific co-founder of a company called Pluripotent Therapeutics, who's very interested in developing cell-based therapies based on pluripotent stem cells. And I'm obviously going to talk about pluripotent stem cells. And in this audience, I don't really need to give a big introduction. We all know there are both human embryonic stem cells and human iPS cells that have been widely used, trying to recreate the diversity of human cell lineages in a dish and to apply that to studies of development, disease, and the topic of today, do we really make the building blocks of cells for actual cell therapy and regenerative medicine? And in fact, in my presentation, I'll focus particularly again on this idea of bringing new cells into the regenerative process. Now, this is no longer just a pipe dream. And in this case, actually, we are at the stage where we have entered the clinical realm. For example, in Parkinson's disease, where my lab worked on for many, many years, we are able now to replace dopamine neurons, which are the cell type lost in Parkinson's disease and responsible for the characteristic motor symptoms. It took us about 10 years to really develop the developmental biology to convert pluripotent stem cells into dopamine neurons via floor plate intermediate. And in proof of concept studies in mouse, rat, and monkey models show that these cells can function in vivo. It took us nearly another 10 years to then take this approach and make now billions of those cells under clinical grade conditions, make them off the shelf, so we can actually test the exact same lot of cells for hundreds of animals, so for the regulatory agencies, and have the same lot ready to go for actual patients. And just by last year, we actually got the green light from the FDA to move forward. And indeed, that has now entered, again, the clinical sphere. We have now grafted several patients with those off-the-shelf dopamine neurons. And we are very excited now after this journey of 2 decades to see how we really can make progress there. Now, we try to replicate that in other contexts. For example, in the peripheral nervous system, we hope to replace the enteric nervous system. And in Hirschsprung disease, for example, a specific loss of the enteric nervous system in the gut. We can make the precursors from the neurocrest, have the right Hox code to label them as vagal neurocrest that gives rise to the enteric nervous system. And we can rescue mouse models of Hirschsprung disease. And we now go again through the same journey of making the cells on the clinical grade at the right scale and so forth to really hopefully in the future get into the clinical realm as well. There's still a lot of interesting biology on the road that again points to understanding mechanisms of integration as key. For example, we can make not only the vagal portion of the enteric nervous system, but also the sacral one most recently, and show that they have surprisingly different migratory and integration behavior. And in the past, we didn't really know what the sacral neurocrest does in the enteric nervous system of animals. And now we have a really powerful system to study to figure out also, do we need these cells for actual translation? But the point of my presentation is really, can we make that more general and what are really the challenges? And I kind of divide them in three challenges. One challenge being the challenge of the cells itself. What we've learned is it's really key to make the right cells. Many groups have tried with a similar cell and it hasn't really worked very well in vivo. So we need to make authentic cells. And we need to do that more systematically. So in the lab alone, we can make about 50 different cell types. But I think as a community, we need to be able, similar to the Cell Atlas project, where people now describe individual cell types. We need to have an atlas project where we make all the various cell types and have commonly available protocols for people to use and to access those cell types as you can isolate the protein, you can isolate any cell type you want in any numbers. Now, the bigger challenge we think right now is actually to bring in time. So what we really learned again is also it's very critical to graft cells at the right stage. If you do it too late, they're not going to integrate. But on the other hand, to have the cells, the full functional properties, that can take months, if not years, to achieve, for example, in the nervous system. So how can we toggle force and back and make that an efficient process? And it turns out while fate is largely conserved across species, timing is not conserved at all. And we have no idea why it takes so much longer in certain organisms than in others. There's some interesting work starting to tackle that. For example, work by the Briscoe lab and the Ebisuya lab looking at species-specific difference in biochemical rates that might explain at the early stage, in the somatite clock and spinal cord development, the differences. We have looked at epigenetic barriers that we think, for example, are critical in controlling the timing of neuronal maturation. And interesting, some of those same barriers that seem to slow down maturation, we find conversely, if you upregulate them, can actually be critical also for controlling basically the competency of a cell to undergo transformation. So again, maybe a general topic we can discuss a bit more, but there's an evolutionary reason for those barriers. But we need to be able to toggle those force and back. And finally, in many instances, it might be not enough to just put the dopamine neuron back. No, you want to have more complex tissues. And we know more and more from the pioneering work of Yoshiki Sasai with the optic cup, brain organoids that many labs do like ourselves, or even gastroloid neuroids. We know a lot that we can make some of those structures, but everything has been done largely empirically, finding the right conditions. We understand very little about the actual mechanism of that self-organization. That's going to be critical if you want to co-opt those mechanisms to not only make better organoids, but actually to co-opt those mechanisms to in vivo get these processes to work to make more complex structures. So that's challenge one. And challenge two, I think I would define as the challenge understanding the graft-host interface. So there are points such as simply delivery, which is obviously critical in cell therapy, where you're going to inject them like we do for dopamine neurons neurosurgically, the right place. Or in some cases where this really might not work, where you need to have systemic delivery to many places, or systemic deliveries to then home to a safely way to the right organ. And again, we understand very little about the mechanisms to achieve that, and to really engineer such homing properties, that's going to be, again, key. Now, another kind of dirty secret in the field is that when you graft neurons, when you graft skeletal muscle, cardiac cells, typically more than 90% of the cells die within the first week. Nobody knows, again, why that is, and what's really the mechanism, and it has been not systematically addressed. People add certain growth factors and so forth, but it doesn't really address more the mechanistic side. We've started now performing CRISPR screens to really get at that question, and we found some, again, barriers. Some of them are kind of, again, actually p53 related, for example, where we can then coax the large majority of post-mitotic neurons, so it's not a proliferation effect, but post-mitotic neurons to survive at much larger numbers than if you don't have if you just have the standard procedure. But again, we need to know what triggers those barriers when we graft it and eventually come away with a strategy that really allows grafting much more systematically with much less cell loss across tissues. Now, we already talked quite a bit about the host response, and I'm sure it's going to be much more today about, for example, issues like fibrosis, glial scars, inflammatory responses, and so forth. And so that's something clearly very important in grafting. But something maybe less well thought about is something which might have to do with cell competition or selective growth. So for some cell types, for example, microglial cells, or for oligodendrocyte precursors, not only can these cells be engrafted, human cells into a mouse, they look quite nicely, go to the right place, do the right thing, but they actually outcompete the endogenous cells. To understand how that mechanism works would be very powerful. You could, for example, then replace disease cells with normal cells using such a competitive advantage. But you don't really know, is this just a species-specific phenomenon here? Or is it maybe more developmentally mature cells that outpace the more differentiated cell types? So there's a lot of work that needs to be done. It would be very powerful if we can harness that in such an approach. Then finally, I divided into an area where it's basically the disease itself and the host more generally. So we need to actually know what needs to be replaced. And the reason why we chose those diseases, we know what needs to be replaced. We know the numbers, we know the cell types. In other cases, we don't really know what we need. If you have a stroke in one place or another place, you have different neurons affected, different glial cells. And so how do we address that? We need to understand the disease. We need to come up with much more sophisticated way to measure when the region is obstructed, what actually needs to be replaced, and how can we match that with our tool set that we have from pluripotent stem cells, and what are then the limits for those admittedly much more pie in the sky approach of diseases. The second part there is the ongoing disease process. It turns out that, for example, in our Parkinson's graft, when we do fetal grafting, we know from patients that after about 10 years, takes a long time, but after about 10 years, the Parkinson's disease can actually enter the graft itself. So we need to understand how can we protect those cells long-term. In some cases, it's much more dramatic, such as from multiple sclerosis, where oligodendrocytes die in an autoimmune process. Obviously, need to address the disease to really make that transplantation approach work. And then finally, obviously, a big topic, again, we can discuss much more broadly, is the immune response. With any extrinsic cell type, we need to tackle now how this immune system is going to respond to those cells. And right now, it's a very interesting time because all those various approaches described here have started to enter, again, the clinical realm from an autologous approach, original Yamanaka ID. We can match each patient, which has obviously its own challenges. They might carry the disease, difficult to QC and so forth, to having purely allogenic approaches, to having HLA matching, or to engineer the cells to actually have so-called universal cells, which is a technology that has a lot of promise, but also still a lot of challenges with regard to its own safety issues that are brought up. So then finally, really as a conclusion, I think we are now at the stage here where, again, it's not just a discussion, how can we get to the clinic? We are actually in the clinic. So we have made a lot of progress, and we understood that the cell is really critical and the disease indication to make that work. But we have many new insights and tools needed with regard to cell lineage, stage and assembly. We need gold standard data for cells from atlases to really understand whether we make the right cell. We need to have reporters, lineage fate mapping reporters to make those human trees, make the human cells a true genetic system. We need to have reports of biochemical rates, chromatin state, self-organization to tackle those big mechanistic questions. With the graft-host interface, it's really the imaging that's very critical to really actually look what happens when we graft the cells in real time, how the cells respond, and again, using genetic screens to get at the real barriers of what's needed to get survival, homing, and how to use those for engineering. And finally, then, for the disease processes is really this idea of long-term tracking the cells, again, by imaging, and understand mechanism of competition, and basically make disease-resistant cells, among others. And for the brain, it's actually quite remarkable how little we still know about the immunology and to what extent the brain is really immune privileged with regard to choosing the right strategy to really get an allogenic versus isogenic graft. And that leaves me then just with acknowledging, obviously, many people in my lab that make a lot of fun to work on those problems, and the many collaborators and funding institutions. I think I'm going to stop here and obviously look forward to a discussion and happy to answer any questions.
Thank you so much, Lorenz. We have time for a few questions, and we have one already in the Q&A. How different are DA neurons in 2D cultures versus 3D organoids? Sure.
They're actually, for most instances, quite similar, I must say, which again, maybe a little bit heretic. Many people think it's very important to have the right organization, the right 3D structure, vasculature, and so on. But it looks like these programs to make sulfates are very hardwired into the genetic sequence of events. And so that's actually why these protocols work. So you need a relatively limited number of signals to get a cell that behaves remarkably similar. Now, if you compare them side by side, you feel some differences. But if you graft them in vivo, they seem to be indistinguishable actually from endogenous cells. They make the same size, they make huge processes, they can make millions of synapses. So it's actually, again, not that complicated to make the right cell.
That's great, thank you. Actually, maybe I could ask a question and follow up to that. I was really compelled by your sort of plea for a systematic atlas of the cell types that can be generated. And one thing that plays around in the back of my mind with that is that in many cases, what you're cataloging are the stable cell states that these cells can be in. And maybe that's a different thing than their fate. And how, when you're thinking about this, how do you think about these transitional states and how they impact cell function, how we might be able to capture that dynamic aspect of stem cell systems?
Sure. No, I think that's an important point. But the way I think about the map is not necessarily just have the stable states, but actually track each stage in time. So you'd have a day 15 cell that might be a transient state, but might be the perfect state of using these cells for cell therapy. And obviously, once you have each cell of the tree, you can exactly study the transition from one to the next state and see how robust those are and what are the factors that really influence those state transitions. So I think, again, it would be a powerful tool. Obviously, the counter argument is you just make an organoid that has all the cell types in one shot. And I think that's also an interesting point. And you try to re-isolate the cells at the end. Right. But again, from having gone through the process of trying to get those cells clinically approved, it's actually something you need to really do under very high stringency.
Yep. Yeah. Oh, and congratulations on that. That's a huge accomplishment. Any other questions coming in? Maybe I could ask one more.
Sure.
In your screen for mediators that facilitate engraftment, have you seen any bias towards intrinsic limiters versus interactions with the environment being a major component of the barrier?
Yeah, I mean, the way we did the screen is obviously we did the library in the grafted cells themselves. So it has to be something that affects ultimately the cells and something that can be changed to then result in survival. But we think that some of those genes that really are critical for survival are clearly triggered by extrinsic factors. And so the inflammatory response to the graft seems to be critical. There is clearly an issue of hypoxia in certain graft paradigms. And so we try to figure out, obviously, how do we not trigger those cell death pathways and how can you optimize such paradigms, again, to make it a much more broadly available technique for many cell types.
Great, thank you. I know we're at time. Can we get Dennis's question in, or should we bring that to the discussion?
Yeah, let's bring that to the discussion. I think we need to move on.
Okay, great. Thank you so much, Lorenz, for a wonderful opening talk. Dennis, please hold your question for the discussion. And the other questions in the Q&A, maybe, Lorenz, you could answer those as we go. So our next speaker in this session will be Agnieszka Czechowicz from Stanford University. Agnieszka, if you can go ahead and share your slides, we're looking forward to your talk. Yeah, that looks great.
Okay, perfect. Thank you so much for the opportunity to speak today. Again, my name is Agnieszka Czechowicz. I'm an assistant professor at Stanford. And today I'll tell you a little bit about our work on progress towards safe curative treatments of blood and immune diseases, and specifically our learnings from the development of novel conditioning regimens for hematopoietic cell transplantation. So as almost everyone in this virtual room knows, hematopoietic cell transplantation is this pretty remarkable procedure that allows us to cure many different blood and immune diseases. And it's really based on the concepts of taking healthy hematopoietic cells, often from a donor that can be obtained from bone marrow, mobilized peripheral blood or cord blood, and transplanting them into a recipient that has a disease. And these remarkable cells have this ability to generate an entire new blood and immune system that can persist for the life of the patient. And this isn't something theoretical, but this is something that we do frequently. And as a stem cell transplant physician, I take care of patients that undergo these procedures. And as a field, we've been doing this for now many, many, many years. And I would say that when we started as a field, we really knew very little about this system. And over now the many decades, we've learned a lot more, but there's still a lot more that we can learn that I think will ultimately improve this process. And that this is a great example of cell therapy that can be used in many different systems. And so far, over 1 million patients have been treated with these hematopoietic cell transplant procedures. So with time, we've learned that these procedures are primarily functional due to the hematopoietic stem cells that reside within them. And this is just a high-level schematic of the hematopoietic stem cell tree. Now this has been redrawn by many people in lots of different ways with additional cell types and additional markers, but the general concept remains the same, that there are these very unique cells that have the remarkable ability to self-renew for life, as well as give rise to many different types of blood and immune cells. And because of this property, they can be used for the treatment of many different blood and immune diseases. And I think sometimes we forget how many different blood and immune diseases there are. As categories, there are genetic diseases, there are malignant diseases, there are autoimmune diseases. But these cells really infiltrate so many different systems and interact with so many cell types that there's many other diseases that are intermixed that could be considered blood and immune diseases, and that can be cured by hematopoietic stem cell transplants, either using normal healthy hematopoietic stem cells or even gene engineered cells, which can allow us to treat even additional diseases. So on the right is just a list of some of these different diseases, some that I'll additionally point out in addition to the more common ones, like the hemoglobinopathies or malignancies that people talk about. There's also diseases like multiple sclerosis, HIV, inflammatory bowel diseases, amongst others. And so really trying to understand how this hematolymphoid system works, how it can be recreated, and how it can be used for the treatment of many different blood diseases is critically important. This is just a diagram showing the number of transplants that we do today. As you can see, it's been nicely growing and increasing over the last several decades, but we really still think that there is a huge opportunity to expand the use of these types of transplants, given how many patients there are with those diseases on the prior slide. And when we've done a back calculation, we've predicted that less than 10% of patients who could benefit from hematopoietic cell transplants get transplanted today. And primarily, we limit these procedures to patients who have absolutely no other treatment options and are states of extreme desperation. And so why is it that we don't use hematopoietic cell transplants in more settings? And why is it that they're not more common? Well, I think that there's two reasons to that. One is due to the complexity of the procedure. And on the left side here is just a schematic showing the many different pieces that go into a hematopoietic cell transplant today. And then the other big limitation is the toxicities of the procedures. And those can be really grouped into three main categories: graft versus host disease, serious infections, and gross tissue damage. And sometimes these complications can be worse than the initial diseases that we transplant for. And so they are taken very seriously. And this is part of the reason that we are very cautious for who we recommend transplants to. And then not only is this a complicated process, it also takes many months and has extreme costs. And so again, if we could work on optimizing various different pieces of this and make these procedures safer, easier, and more effective, then they could be used for many different patients in many different indication settings. So how do we do this? Oh, I think there's a lot of different opportunities for how to improve these types of transplants. And our ultimate goal for these types of HSC therapies is to have 100% survival and 100% cure rates. Right now, as a field, we tend to talk about survival rates and survival curves. And on the right is just a schematic showing what we're talking about. For many indications, we have 5 to 10% mortality rates, if not higher. And that's not even talking about the toxicities of these procedures. And so again, the goal is to have 100% survival, 100% cure with no short-term or long-term toxicities. And on the bottom here, I just listed some of the potential opportunities for things that people are studying to try to improve these transplant procedures, which could then have tremendous implications for our field and for many patients. And specifically, my research group has been focused on the first point here, the elimination of genotoxic conditioning, as I truly believe this is a big component to causing a lot of the toxicities and a lot of the complications associated with these procedures, including graft versus host disease due to its triggering of inflammation, including the serious infections due to destroying the immune system and creating barriers or creating destruction of barriers that then allow different types of pathogens to enter the body and then directly causing tissue damage. But there are other beautiful opportunities for how to improve these types of transplants, including expansion of stem cells, better collection of stem cells, derivation of stem cells from iPS cells, genetic manipulation of HSCs, skewing of them towards specific cell types, stabilization of them to prevent additional problems like clonal hematopoiesis, and improving immune suppression to allow allogeneic transplantation much better. This is just some figures showing how we've started to think about improving conditioning for transplantation and what conditioning is. Conditioning is this process that we give to patients prior to a stem cell transplant that currently involves chemotherapy and radiation. And through our research efforts that were started when I was a graduate student in Irv Weissman's lab, we showed that immune suppression was not enough for stem cell engraftment. And we showed that actually hematopoietic stem cells compete with other hematopoietic stem cells in a unique fashion, and that this was an additional barrier to hematopoietic stem cell transplantation. So you couldn't just give more hematopoietic stem cells. If you did that, what you saw is that on a per cell basis, they're actually less likely to engraft. And so again, this led us to think that HSCs compete with other HSCs. And then this led to the broader concept of depleting your own host hematopoietic stem cells prior to a stem cell transplant to then enable more effective transplants, and to do this in a targeted manner. So again, current methods involve total body radiation, chemotherapy, things that destroy hematopoietic stem cells of the host, but also destroy the rest of the bone marrow, destroy and damage the niche, and destroy all sorts of other organs along the way. And our goal has been to specifically target the host hematopoietic stem cells, deplete them to leave room in the niche for the new incoming transplanted stem cells. And we've now been able to do this in a number of ways. And the histology here is actually just a picture of a mouse that was treated with an antibody that is specific for hematopoietic stem cells. And what you can see is the marrow is very normal, despite having 99.9% of their hematopoietic stem cells depleted, because we've left everything else intact. The way we've primarily done this to date is by targeting an antigen on hematopoietic stem cells known as CD117 or c-KIT. This is an important receptor for the ligand stem cell factor and has minimal expression on other cell types, although we've also done this with other antigens, including the ones listed below. And this is just some data from our first generation approach, which was an antagonistic antibody to mouse CD117, where we showed a one-time treatment of immunodeficient mice with this antibody could beautifully deplete the hematopoietic stem cells in the animal, and now enable much enhanced engraftment of donor stem cells. And then similarly, as a postdoc, I showed that this could be done with antibody drug conjugates in an even more profound manner, together with Derek Rossi and David Scadden's group. And now a much smaller dose could have even more depletive effects and enhance engraftment in even more profound ways. Since these initial findings, other groups have built upon them and added additional agents to our initial antagonistic c-KIT antibodies and have showed increased efficacy in this manner as well, either by combining with low-dose irradiation, CD47 antagonism, or low-dose chemotherapy. Again, these approaches are extremely specific, safe, and well tolerated. This is just some histology showing that the rest of the organs are completely intact with no damage. And we have observed none of the classic issues with traditional conditioning. And on the left is just a picture of a mouse that's been treated with our CD117 antibody drug conjugate that looks like a totally normal, happy mouse as composed to an irradiated animal, which is from a high level shows you the differences in the ability to do this. We've also really beautifully shown that these antibodies and antibody drug conjugates in particular can be used to condition animals without causing any cytopenias, which is pretty remarkable. So these animals have not required any transfusions and have had a relatively normal hemoglobin, platelets, myeloid cells, and lymphoid cells. So it's really sparing both the myeloid system and the lymphoid system. And so it's a really targeted way to deplete the hematopoietic stem cells while leaving the rest of the mature system intact. And because of these exciting preclinical findings, now several companies have been developed that are working on parallel clinical agents, the first of which is already in clinic, and the second and third of which are reported to be in clinic in the near term. And this is just a little bit of data from the first clinical trial that derived from our efforts on the antagonistic CD117 antibody that was used in patients with severe combined immune deficiency. Dr. Judy Shazura was the sponsor of this trial, and this was run by Dr. Rajni Agarwal, who is a clinician and faculty member in our program. And what we found was that in our patients with SCID that were treated with this antibody-based conditioning only, we saw very similar results to what we're seeing in our animals, where we saw beautiful engraftment without any toxicity whatsoever. In fact, we had nothing to talk about on rounds on these patients because they were not neutropenic, they had no mucositis and no other issues. And so we're really excited about this approach and combining it with other approaches in our field to really enable expanded use of hematopoietic cell therapies, in particular combining this with gene-corrected autologous cells and even augmenting HSC function through genetic manipulation in addition to treating patients with genetic diseases, and then combining this with immune suppression for allogeneic transplantation in immunocompetent patients, which potentially could be even used to combine these types of transplants with solid organ transplants due to the induction of immune tolerance. And we have some beautiful preclinical data in both of these settings that has recently been published. So many questions still remain. I think we're at the tip of the iceberg here. We're still trying to understand how this antibody-based conditioning really works. We're trying to apply it to various different disease settings and understand how it can be used differently or optimally in different settings. And then we're trying to use these agents as actual tools to try to understand hematopoietic stem cells in the microenvironment in much more profound ways through various different collaborative efforts. And so with that, I'll just pause and take any questions. Thank you so much.
Thanks so much, Agnieszka. We do have a question from Rahul Gupta. Can these be used to specifically target pancreas or GI tract as they're used for treating diabetes, as they're used for treating diabetes and IBD? I think, yeah.
Yeah, so people have shown that hematopoietic stem cell transplants can be used for different types of autoimmune diseases. So by depleting the immune system, the hematopoietic system, and retransplanting it with cells that are not primed, you can use it in those settings. And so patients have been treated with type 1 diabetes in early stages or IBD in early stages, and then had those diseases completely eliminated. So that's the idea for how it can be used in those areas. Although I think that the broader concept of depleting certain stem cells and other organ systems using antibodies to then enable engraftment of stem cells to help with that is something that should be explored in other settings. And I've been surprised by how many other treatment settings we've also used low-dose chemotherapy or radiation as ways of helping those cells engraft rather than thinking about what we could be doing in a more targeted manner.
Great, thank you. I'm not seeing another question in the Q&A box, but audience, please put your questions in there so that I can see them. Agnieszka, I did have a question about the stem cell competition, which is something that came up in Lorenz's talk as well. In most of the images, of immunolocalizing stem cells in the marrow space, they're quite distant from one another. So I'm wondering what your model is for how those cells are competing with one another in the transplant situation.
I don't know. And I think it's really interesting is the bottom line. One of the things that we found in these studies, though, is that you could co-transplant many progenitors, what you see in that smaller right panel, with the long-term stem cells, and they didn't compete. So that you didn't get a decreased engraftment with those, even if we gave 100 times more of those. And so there is something special that causes hematopoietic stem cells to compete with each other. There's some sort of regulation that the body has to limit the number. If that's a physical niche, if that's more of a cytokine milieu, some combination of both, some other sensing mechanisms, that I think remains to be understood. And I'm excited to try to work on that with other folks and think there's huge opportunities in our field to try to understand that.
Great, thank you. All right, well, thank you so much for this fantastic talk. I'm going to hand it over now to Kara McKinley, who's going to introduce our next speaker and then lead us into the discussion.
Perfect, thanks, Amy. And I am delighted to have this opportunity to introduce Anastasia Tikhonova from the University of Toronto.
Yes, all good.
Looks great.
All right. So good morning, everyone, and thank you so much to the organizers for having me. I'm really excited to be here. So we know that the field of regeneration medicine is heavily driven by advances in technology. But it's challenging to stay on top of all the current trends, and let alone have a forward view on every aspect of regeneration. So today I will discuss some of the challenges in the field of hematopoietic regeneration and propose new approaches aimed to enrich our understanding of these fascinating processes. So I'm an early career investigator at Princess Margaret Cancer Center in Toronto, and my team focuses on delineating molecular crosstalk between hematopoietic stem cells and the microenvironment. So my goal today is to draw your interest to the role of the hematopoietic stem cell niche in hematopoietic transplantation. In our 10 minutes together, I will first share my perspective as to what I think are the biggest challenges in the field are. And secondly, I will discuss some of the new promising approaches that I think would really lead to transformative advances. So blood is highly regenerative tissue. Hematopoiesis gives rise to billions of cells on a daily basis. It gives rise to all blood cells and vast majority of the immune cells in the body. And this is necessary because, for example, blood cells have a relatively short half-life and they need to be replenished. So hematopoietic stem cells reside on top of hematopoietic hierarchy and give rise to erythroid, myeloid, and lymphoid lineages. And one of the striking qualities of HSCs, as Dr. Czechowicz just told us, is the ability to completely repopulate the entire hematopoietic system when transplanted into the recipient. Hematopoietic is dynamic. Hematopoiesis is dynamic following stress such as infection, for example. It switches into production of predominantly myeloid cells and then has the ability actually to result back to the normal output of hematopoietic lineages. Within the bone marrow, hematopoietic stem cells reside within really highly specialized bone marrow microenvironment. And term hematopoietic stem cell niche, identification of the cellular population that constitutes the niche have been an active effort in the field for the last 10 years, and identification of the SLAM markers allowed to localize them within these perivascular spaces. So hematopoietic stem cell niche consists of two main cell types, vascular endothelial cells and leptin receptor positive mesenchymal progenitor cells. And for example, stem cell factor, which is a ligand for the c-KIT, and as well as stromal-derived factor 1 are produced by these populations. And functional studies demonstrate that when you delete 6L12 KIT ligand from the niche compartments, it impacts HSC homeostasis or localization. So I want to note that these mesenchymal progenitors can give rise to osteoblasts or adipocytes and are themselves a great interest to regeneration, as Chuck Chan told us yesterday. And I just like to note that in addition to these two main subtypes, there are other cell types that were implicated in maintaining hematopoietic stem cells, such as megakaryocytes, macrophages, Schwann cells, and T regulatory cells. So bone marrow niche cells are really rare. So vascular and mesenchymal progenitors are really rare. They constitute about 0.1 to 0.3% of total bone marrow, depending on the subset. And we and others use single cell sequencing to really understand the transcriptional heterogeneity of this cellular population in depth. So there are results of our analysis are these really precise molecular maps, which revealed a previously unappreciated level of heterogeneity within perivascular cells, for example, the mesenchymal progenitor cells, and results cellular sources of number of prohematopoietic factors, chemokines, as well as membrane-bound factors. So we know hematopoiesis is dynamic. We found that bone marrow niche is dynamic as well. We uncovered that there's a considerable molecular reprogramming of the bone marrow microenvironment following treatment of the animals with chemotherapy with just acute dose. You can appreciate this shift from red to green of mesenchymal progenitor cells, and this just shift due upregulation of this fast proteopogenic signature, which later results in increase of adipocytes in the bone marrow. And interestingly, over time, as the hematopoiesis is restored, these adipocytes disappear and the homeostasis is restored. There are multiple course, that both inflammation and neoplasia can remodel the bone marrow niche. This is a famous figure from Darcy Sipkin's lab, demonstrating that following onset of B-cell acute lymphoblastic leukemia, you're completely losing expression of CXL12 in the niche. And the idea, and because it was found that multiple different neoplasm actually change the microenvironment, the idea was put forward that it's possible that malignant clones capable of rewiring niche to support their own propagation. So for more than 50 years, bone marrow transplantation has been used to save the lives of patients suffering from blood diseases. And transplantation is unarguably the most successful therapy in stem cell in regenerative medicine. However, there's really high risks of toxicity, at least as the way it's done right now. And because of that, this really kind of meant as a last resource. In addition, there is also a lot of transplant fail due to immune rejection, and as Agnieszka just told, malignant relapse and poor graft function. And there's also many of the patients that require transplant cannot actually find a match. So I think some of the challenges, really critical challenges in the field, is having this limited source of functional HCCs and improving hematopoietic stem cell transplantation efficiency. And I will focus on the second one today. I think that niche is likely a facilitator of things, and we know that it's remodeled during pathologies. It's remodeled during malignancy, it's remodeled by chemotherapy regimens, as we've recently found in my lab, and aging. And what I think we really don't fully understand how this cellular molecular rewiring takes place. Is it transient or permanent? Can we reverse it? Can we precondition niche and preserve its function and support of normal hemopoietic stem cells? And I think that understanding the bone marrow microenvironment, we really will allow us to significantly improve clinical potential of hemopoietic stem cell transplantation. So, but how can we do it? So I think we have to understand niche better. And I think that intersectional genetic is a great tool for that. So many of us who work with mice use create recombinase animals to target specific populations. And we all know that there although there are often many different limitations, such as precision of the lineage tree. So CRE is not the only recombinase we can use. We can also use DRE and combine CRE and DRE systems together to have high level of self-specificity, but also target things in a sequential manner. For example, deleting genes sequentially or inducing mutations sequentially. I think, so recently I saw a report in cell stem cells from Gonzalez labs that generated 70 new DRE lines. I think that's a really amazing effort. And it's surprising that it comes from just one group. And another major frontier I think that will help, as we all alluded to, really understanding the cellular communities. And it can be done by proximity labeling. So this is a cherry niche system from Ilaria Malachi's group in Creek, and they utilize cancer cells that are expressing mCherry that's conjugated to basically modified lipid permeable transactivator that allows for mCherry to enter into the surrounding neighboring cells and label it. And I think this is a really cool system that has a lot of potential in hemopoietic research and regenerative research in general. So with that, I think I hope I convince you that niche research promises to improve prevention and treatment of hemopoietic disease. And I hope to pique journalists' interest to really generate these new functional tools and make them available to the community, because I honestly think it's a hard undertaking for just one group. And with that, I just want to give a shout out to amazing people in my lab with whom together in less than a year, we built a functioning lab in this very interesting environment. And thank you so much. And I'm open for questions.
Terrific. Thank you so much, Anastasia. That was a really gorgeous talk that I think is giving us an important perspective going into the discussion to take a really aggressively look at the niche and not focus exclusively on the stem cells. Please, audience, go ahead and put your questions into the Q&A, but I think maybe I can start off with, you talked about contexts in which this niche rewiring would be detrimental. Do you mostly envision that your goal would be to block rewiring in those contexts, or are there contexts in which you think that like supercharging the niche and in fact inducing that rewiring might be the path forward you'd be wanting to go for?
Yeah, exactly. I think the key is really understanding the molecular mechanisms of the rewiring, like, the genetic landscapes, what's involved. And I actually cannot tell right now. I think when disease is fully developed, it's too late. But I think early stages of the diseases, and we know that, for example, for AML, it takes years to develop often, right? And we can, if we start looking at it early, I think we will be able to really manipulate it.
Very cool. We have a question from Shruti, which I'm excited to hear your answer to. What are your thoughts about examining the niche in humans?
So yes, great question. Yes, my thoughts are that it must be done. We know there's major differences between mouse and human. For one major difference, for example, adipocytes. So in human, if you look at the bone marrow or even of healthy people, you see a lot of adipocytes. In mice, it's very hard to have adipocytes in the bone marrow unless you have a full myeloablation. And so the rules might be slightly different. I think the key things are preserved through evolution between mouse and human, but I do think it's very important to try to deconvolute it in people. Of course, the challenge of that is impossible. It's very hard to do, right? But there's some effort done by other people now to really look at the human niche. I think it's very important.
Great. Let's take a question from Dennis.
Yeah. I'm glad it was a great talk. I'm glad you brought up that it's a fatty marrow issue, right? Which is profoundly different, as you say, between rodent, mouse, I should say, right, and human. But there are other species that also have fatty marrow, right? And so are those understudied? Dog, cow, I think. I'm vaguely recalling. Is it rabbit? How often studied are those in these contexts?
So there's a really beautiful work recent from John Dick's lab, actually compared the bone marrow between dog and pig and mice and human, and showing that mice are probably but you also have to remember, right? Even dogs, we don't breed dogs in SPF conditions, right? Not really. Like in mice, breeding is really controlled. So we don't really know if it's due to necessarily genetic mechanism or some of the environmental mechanism. I think nobody even looked at the wild mice yet in terms of what does the bone marrow looks like. I'm really interested. So to me, it's unclear. I think that I personally couldn't study anything in dogs, but but I think it's very important to really, find these optimal systems and decide how much, where the overlap is, and really pursue this mechanism of overlap.
And so do you think the fatty marrow matters a lot? And the mouse studies have been marked in that sense or not?
Yes, I think fatty marrow matters. We actively pursuing it now in the lab. So I will tell you the exact answer in a couple months. Fingers crossed. Yes.
Okay, thank you.
Perfect. Thank you, Anastasia. I think in the interest of time, we should transition to the broader discussion, although there are still some questions in the chat that maybe you can get to after the discussion. So for this discussion, we're really excited to welcome back our three speakers, as well as Ahmad Nabhan, who will be joining us in the Emerging Leaders session later. And maybe to kick us off, I can start by asking a question that I've been sort of mulling over for the last 2 days, which is, and I actually, before I say this, I want to strongly encourage all of our panelists to get involved in this conversation, because of course, we know we have a sort of hematopoietic focus to this session, but all of our solid tissue friends, please feel welcome and encouraged to be here. And I think that sort of relates to my question, which is to come back to what Lorenz was talking about in terms of what cells should we be making? And to Amy's question, that a lot of what we might want to, the cells that we might want to be targeting might be relatively transient. I guess I sort of want to pose that question of what cells should we be making, and especially in the context of the conversation yesterday from Jason and Laura, in terms of, in some tissues, is the stem cell the right goal? And actually, in many contexts, the stem cell that we consider as the driver of renewal is actually a pretty different entity than the driver of injury or repair. And so what really should what should be our target? And I do definitely want to also, yes, hear from our panelists and encourage broad participation from all who have stem cell interests.
I mean, I can start otherwise if nobody else starts off. I mean, so we thought about that a lot, and I think it's going to clearly depend on what cell you, again, need to replace. It's also dependent on which cell you want to use to actually achieve that, because in some tissues, some cells like nerve cells, they last your whole life. So it's not like an hematopoietic system. If you just make a differentiated cell, after a short time, it's going to get removed, and it doesn't really have a big impact. You really need to target the stem cell component. But if you have a tissue where, again, you have terminally differentiated cells that persist a long time, or in pancreas as well, islet cells probably self-renew at a very low level for a very long time. So there you can go more for a differentiated cell and just empirically figure out how you get best to that cell. But in other cases, yeah, you might need progenitors, you might need stem cells. And I think that's really the question now. That's why I think we need to have access to all the cells, to all the transition stages. And I think it's not necessary, actually, again, from our experience, the stage that we use for dopamine grafting is highly transitory. It's like a day 16 cell. It's neither a dopamine neuron completely, it's neither a progenitor completely. We just freeze it down and we make billions of those and inject them. So it's not required, like the stem cell concept, to have a long-term renewing cell, and it has to be stable. And engineering-wise, you can definitely make cells at any stage if you can cryopreserve to make them at scale. And so I don't think there's any limitation in this regard. It's more the biology figuring out what actually works and what's needed in each organ.
Yeah, I think it really depends on the disease and the situation that you're trying to address, and thinking about what is missing or what is having issues, and how do we augment that. And in many situations, actually, it might be multiple cell types that you may want, either cell types within one organ, or even cell types of an organ and then immune cells that interact with those cell types. So I think it's complicated, and there's a lot of work to be done, but working on each of these individual pieces and thinking about how they work together is an important piece of this.
Maybe I'll just, I had one question to like the blood folks, I guess, which is why it seems like a huge challenge is actually expanding the niche. So why are we not trying? Is anybody trying to expand the niche? And is that possible? Wouldn't that be I guess that will be, I'm sure we'll be at work. It's clear that we can make the cells, and now we're having trouble in grafting them. So I assume in a few years, we'll be back talking about how we can expand the niche. How do you think, is the approach the same, or how would one go about doing that?
The people are trying to do that in vitro. It's because it's vasculature, you actually need real blood vessels with blood flowing through. So it's not a trivial undertaking. And also, I think perivascular cells are really unique in the bone marrow compared to other organs. So there are efforts to do that. I think it's challenging. Interestingly, in mice, you can do it without niche. You can just change, you really just change the culturing conditions and make 100-fold of functioning HSCs. It's really amazing work from Stanford, actually. But it's so far impossible to do in people. So I haven't seen it. So I think people trying to do it actively. It's really the holy grail. You like hits in their own head. Yeah.
Yeah, I agree with that. Although at the same time, I'm not sure we need 100-fold more HSCs in many disease settings. The homeostasis that the body has set up to maintain the hematolymphoid system with a certain number of HSCs is pretty profound. But in certain bone marrow failure settings, like some of the patients that I take care of there, it would be particularly advantageous. And then people are definitely working on this in various different ways, and specifically also working on it for immune cells in the immune niche for different types of immune deficiencies, immune dysregulation.
Yeah, add in there, it's kind of interesting in the sense that sometimes we talk about the concept of the niche as though it is a stable state, but in fact, it is actually quite varied as well and dynamic. And there are multiple niches actually that HSCs or any stem cell might occupy, and how that changes in the niche over time. For instance, Agnieszka, you were just saying, I don't know that we need 100-fold more stem cells. In aging, you get 10 times more stem cells, but they don't function appropriately. And is that, how can we address that by maybe targeting those cells? I wanted to ask the panel a slightly different question as well, since many of the approaches that were discussed in this session targeted cell transplantation. And one feature of transplantation that's come out in part from the blood system is that it creates a bit of a bottleneck for engrafting by a small number of sort of founder cells that win. And Agnieszka, you kind of made this point in your talk as well. And I was just wondering, what your thoughts are about that and the potential for oligoclonality or for highly competitive cells to win in a transplant setting, and how we might be thinking about that in the longer term of regenerative medicine with these applications.
So I think oligoclonality could be problematic, especially if some of those clones have specific mutations in them, or a skewed ability to differentiate or lack of differentiation capacity in other ways. But I do think a lot of it has to do in the hematopoietic system with the conditioning that we use. And we had a paper together with Rong Lu's lab that showed that with different conditioning, you got very different engraftment of lentiviral labeled hematopoietic stem cells. And with kind of irradiation or chemotherapy, you're more likely to see oligoclonality than with our antibody-based conditioning that created more space for more clones to engraft.
So do you think clonality analysis should be a sort of a quality control measure as you're trying to improve transplantation efficiency?
And I think people are starting to do that. So for all these gene therapy studies, people are looking at vector integration, and clonality is one piece of that. People are tending to look at it more in terms of being more prepared and aware when patients are moving towards a potential pre-malignant or malignant state. Do we have a clone that is problematic because it's expanding more than others? But I think through more of the work on clonal hematopoiesis in the elderly and in younger people, we're learning more about clonality and other issues that patients can have, not just malignant mutations.
I think there's a couple of hands up, but I just want to ask a quick question about this, which is there a selection pressure as you transplant the cells for their ability to engraft? And is that different from your ultimate goal of the cells becoming, physiologically functional? So can you, through these, massive data sets of patients that go through your clinic, sort of look at clonality in that way and say these aspects of the cell, help engraft, but actually the cell ends up being less useful later? Are we able to do that way, or is that going to be tackled experimentally to get at this question of clonality?
I think where people are seeing this right now is in the gene therapy settings. We're able to look at clones because we're putting in vectors that integrate in different places, so you can look there, or in patients or donors who have had clonal hematopoiesis and so have had known mutations that were identified, and then following those clones in that capacity. But we're only really starting to do this, and people haven't looked at enhancement of engraftment. In general, though, we have seen worse outcomes when patients have had donors who have had a chip within them. And that's what some recent studies have shown, but that's something that people are only starting to do on a research basis and isn't done for the hundreds of thousands of patients that have been transplanted to date.
I think another thing to think about is that with clonal hematopoiesis, with certain types of mutations, you're going to have this increased inflammation. And it's not going to be acute inflammation that resolves. It's going to be chronic inflammation within a person for years, right? Because we don't know when clonal hematopoiesis can be started early, or it is associated with aging, but it can also start really early. And you can imagine that chronic inflammation is going to remodel your niche in a very substantial way. And then it's going to affect the competition, even if you're putting in healthy hepatic stem cells. If the niche is actually remodeled to support more inflammatory clones, that might be a problem. So this is something also to think about.
I think Jennifer has a question. Maybe we can bring her in, and maybe as we're doing that, I wonder, Lorenz, if you could comment on whether clonality is an issue in non-blood transplantations.
Oh, sure. I mean, there's a number of different angles there. I mean, just simply think about IPS cells. Obviously, we see a lot of variability between different IPS lines, which are clonal, and some might engraft better, and some might engraft worse, or give better rise to different cell types. And obviously, you can ask the ones that really engraft better. If P53 is a critical factor for engraftment, you could obviously ask that's maybe then not necessarily always the best cell you want to use for a safety purpose. So I think there's definitely also this issue there. And with regard to engraftment in the brain, for example, there is obviously we mostly try to have mostly post-mitotic cells, but there are a few progenitors like glial cell types, and it's really not completely clear now how level of stringency you need in those iPS-derived cells, where there is similar issues there that they could long-term engraft. We hope this patient's going to live for 30 years as those cells. What do these glial cells do? They're embryonic fibroblasts long-term. They are embryonic astrocytes in this case, long-term. And so far, we haven't seen any negative effect. People have actually grafted astrocytes directly, but it's definitely something we need to consider very well too. And we have this additional complication that obviously in vitro things can happen that are carried on long-term and might impact engraftment quite a bit.
Thank you. Jennifer, did you have a comment or question?
Yeah. So actually this relates to, Amy, what you were talking about in terms of competition among the stem cells. So there's some recent work from Elaine Fuchs' lab where she's showing that at least in the hair follicle stem niche that apoptotic cells can actually release Wnt and other molecules that can impact just the whole niche in terms of what stem cells are taking off versus not. So my question is whether in each of your systems, are you thinking about how these caspases, which are regulating the death of some of these stem cells when they're hitting different situations as they're trying to replenish, is that an area where it's worth thinking about how you may want to control the system? I mean, she could show that by manipulating the caspase 9, she could actually lead to proliferation of hair follicle cells. So, and that's something that's not what we're thinking about. We're thinking about proliferation. This is cell death. Trying to engineer a type of selective cell death in certain types of populations, to allow for proliferation of other types of cells. Anyway, I'm just throwing that out as whether anybody's thinking about this.
I think one major challenge to this is that you can't measure cell death in the same way you measure cell proliferation. So I've tried to convince Vishva that what we need is like a lineage tracing for cell death. The problem is right now you look at everything in a snapshot. And so if there's an effect of having necroptosis, for example, versus apoptosis of a period of 5 months, you have no hope of seeing that. So, I mean, I think that's one area where the tools would be extremely helpful. But it's clear from genetics that, the different, there are mutations in, patients that have various autoinflammatory diseases that affect stem cell functions over, the course of the patient's life. But that I think has not been studied, it's been overlooked because we just can't look at it. We don't have the integrative tools.
And in the hematopoietic system, I think it's definitely something that should be considered more. So part of what we do in these hematopoietic stem cell transplants is primarily genotoxic conditioning right now. And we do think that in part works because it depletes the host hematopoietic stem cells and so creates the space. But there's a lot of cell death that's going on. There's a lot of other signals and how are those then allowing for or inhibiting regeneration of either the native system or the system that you transplant. And we really have not looked at that in detail, even though we do this regularly.
Yeah, I certainly think it's a very exciting direction to consider from the perspective of the endometrium and sort of the fact that you get the most proliferation and recovery during the period of active shedding and during menstruation. And I think that the coupling of that is really poorly understood, and whether that's immune mediated is, I think, also an important question to be asking in a tissue like that. I think, yeah, the crosstalk between death and growth is a big space for discovery.
Thanks.
I think this is a really important topic. I want to make sure I get to Dennis again, as I see the clock winding down. Dennis. Oh, I think you're muted.
Thanks. Sorry. Yeah, very quickly. So sort of 4D physiology, maybe follow up to what you were just asking about death creating space for infiltration, migration. I was wondering whether there's lessons from migrating through adult tissues by metastatic cells, if not other cell types. How do we understand the 4D physiology of getting from A to B? Hematopoietic system, marrow filled with fat, or the brain, where you have to navigate the cell, whether it's if not a stem, a progenitor cell, a day 16 or something like that. You really want to get from A to B. How do we visualize, track, and ensure that that you get from A to B? Maybe that's the first question, Lorenz. Yeah.
Sure. I mean, I'm happy because obviously I work also at the Cancer Institute, and so obviously it's a lot of work done both in zebrafish models and in mouse models. And Joan Mastigay published a lot of work, for example, how cells, breast cancer cells, get into the brain. What are the pathways that get upregulated? How do you get basically out of the vasculature to the blood-brain barrier? And so there's definitely some effort considering now can we co-opt the same mechanisms,
yes,
get
exactly.
cells into the brain, so we don't need to inject them directly, and
exactly.
That's something we are studying for them. We're also studying it in simple systems like kind of 3D organoid systems where we have different cells enter it and see how they can actually enter such a system that gets more and more complex, and what are the key signaling pathways, and actually imaging that all in real time.
Yeah,
It's something we're definitely pursuing, but there's obviously the different components to it that are still not well understood and might be not the same for all the cell types and all the tissues.
Well, that's good. That's good, right?
A lot of interesting work to see now how much is basically organ specific, for example, for homing in this context, or again, what migratory modalities are used. That you all like, I know very well for melanoma that we are looking at, they have a quite different way to migrate than other neural populations. I think that all will be very interesting to see, can we understand it better and see can the same mechanisms be co-opted to get the stem cells or the cells we want to the right place.
Right. And it occurs to me that, as you
Oops, I think we lost him.
He's lost.
Choice of Zoom.
He might come back. I know Valerie has a question. Maybe we can bring her in. I think I don't know. I don't know if I can want to guess what he was about to say, but I was thinking that it actually kind of links the components of metastatic cells' ability to engraft are both their migratory ability and their resistance to apoptotic signals in that context. Valerie.
Yeah, I just wanted to raise an interesting thing that we've been thinking about related to Jennifer's point, in that when skin regenerates, there's an overproduction of all the cells. So there's an overproduction of fibroblasts and vascular cells, and there needs to be a pruning during resolution that occurs over a very extended period of time. And we know very little about that process. And I think that's a really important idea, because that's essential for making sure the scar is a functional scar rather than a dysfunctional scar that we might see during a keloid or another type of disease. And so I think it's a really great point.
I think Jason had a point in the chat, but I'm having trouble going back to find the original point. Jason, do you want to make your point here?
I don't know if can people hear me? Yeah. So no, I was just echoing what Shruti had made a point about my point. That's why it's a little confusing for you, Amy. But the point that we were talking about is that you can see where, since you need injury a lot of the time to induce plasticity in neighboring cells, or to be able to make space for engraftment, you can see where caspase-9 and inducing apoptosis in a cell that's on its way out would be important for signaling to other cells, either to engraft or to change fate and become stem cells. That was our little sidebar there. But fascinating discussion on this.
Yeah, thank you all. Thanks to all the speakers and all the panelists and all the audience for fantastic discussion, great questions. I think we are coming to the end of time. Kara, did you have any last comments or thoughts you wanted to share?
No, this was terrific. Thank you all so much. And I think we pass it off to Janine to turn us over to the break.
Yep, perfect. Thank you guys. That was a great session. Great start to the day. Let's take about a 7-minute break and we will return at 11:50 a.m. Eastern. All right, let's go ahead and get started with our next session. And to do that, I'm gonna introduce the chairs and discussion leaders, which are Mike Longaker from Stanford and Valerie Horsley from Yale.
Thanks, Janine. Valerie and I are excited for this next session entitled Fibrosis. And this kind of gets right to the heart of one of the reasons that we don't regenerate in humans, which is our global kind of problem to be solving, or at least beginning to address. So we have 3 speakers that will talk about fibrosis from different perspectives. And I think we encourage audience participation and a lively discussion. So Valerie.
Great, I'll introduce our first speaker, Boris Hinz from the University of Toronto. Boris, the floor is yours. Looking forward to your talk.
Great. Well, let me just pin this timer, which is an essential tool for me at least. And share my slides. Here we go. Should see my slides now. Well, it's an absolute honor to talk in this workshop, which so far has been great. I had a lot of discussions, even fights with my ego about how much data I should show, and I think I won. I only have one slide with a bit of data, but then decided to rather take one for the team and introduce fibrosis and leave it to the other expert speakers in the upcoming sessions to give more detail. But wouldn't be me if I wouldn't take a fibroblast-centric view on fibrosis. So fibroblast rule. And let me just get the clicking starting here. So just to bring everyone on board and interested is, no matter what organ you're working on, fibrosis is likely going to happen in those organs. So I don't know of any organ which is completely excluded from fibrotic processes. You pick your favorite one, and some of them we heard yesterday already. And I would argue that at the beginning of every fibrotic disease, there's some kind of injury which cannot be handled by the regeneration or repair mechanisms of our body. And most often than not, this injury becomes repetitive or chronic. And that's why rather than repairing the organ, we end up with fibrosis. Fibrosis, by definition, is the accumulation of fibrous tissue, collagen tissue, which in all those conditions replaces functional tissue. And this is why if the scar tissue, fibrous tissue takes over, depending on the importance of the organ, it is a lethal condition in the end. And believe it or not, at the effector side of fibrosis, no matter what happened before, again, I would claim that fibroblasts are the most important cells to create fibrosis, because these are the cells that produce the collagen and organize the collagen into stiff scar tissue, which then impedes organ function. And we collectively often call these cells myofibroblasts. So what I'm going to do is I'm going to raise a number of questions, 5 actually, during my talk, which then we throw into the open space. And one of the questions is, you may think that's trivial, is what is a myofibroblast? And if I throw that question to this audience here, I guess half of you are going to say, I don't know, never heard of it. And the other half is going to give me 15 different answers. So most people in this space would probably use the definition of a mesenchymal type cell, which produces collagen or is associated with collagen, which also expresses a smooth muscle actin isoform called alpha smooth muscle actin, coincidentally, as being the defining feature of a myofibroblast. And this smooth muscle actin is organized in stress fibers, meaning actinize and bundle. So a functional aspect of these cells is contraction. I would argue, and there's good reason to do that, part of it is in the history of how these cells have been named, is that myofibroblasts do not have to express smooth muscle actin to be myofibroblasts. So the defining feature would be that they are contractile, that's the myo part of it, and they have to be associated with collagen, which is the fibroblast part of the name. And again, as long as they're stress fibers or actinize and bundle, that would qualify for myofibroblasts in these repair situations. And this is where things become complicated, because these are activation states of different cells, not necessarily cell types. And there are a number of other activation states of fibroblastic cells, which obviously are different from a quiescent fibroblast that we probably find in most of our normal connective tissues. For instance, proliferating cell, migrating cells or fibroblasts, collagen production. Those are all activation states. They're all associated with the development of fibrosis. But again, I would claim those are not myofibroblasts because they're not contractile. They're just missing the myo part of all this. And they're obviously specialized fibroblastic cells, which specialize for secretion, inflammation, you name it. And those cells are found in the synovium of arthritis or in the tumor space. Now, the question of what is a myofibroblast is obviously related to where do these cells come from? And I'm not going to go into any detail of the left side of this panel here. There are a number of cells or multiple cells which can develop this myofibroblast phenotype, and they come from different origins, different spaces. The only take home I want to transmit here is there's probably a reason why so many cells can attain this phenotype and this function of producing collagen and correcting collagen, because their aim is to repair tissues where regeneration fails, and that has to be done independent of where the injury occurs all throughout the body. And it makes sense that many cells would be able to at least temporarily attain that function. And there's also the right side question around the myofibroblasts. Are these reversible states or are these terminal states? There's good indication. You see a lot of those arrows going back and forth, that some of those states are reversible and can not plastic within each other, but some others like apoptotic myofibroblasts or senescent myofibroblasts would not be reversible. And there are some specialized senescent associated secretory phenotypes as well. Now, again, without going into the detail, you may imagine that there's a sequence of events leading to a final highly contractile myofibroblast state, which may or may not express muscle-actin. And all these cells produce extracellular matrix of different quality. I'm just going to comment here on the biophysical aspect of the extracellular matrix, which is very different when it's produced by a highly contractile cell or organized by highly contractile cells rather than an early state in this activation process. So you end up with stiff tissue and the stiff that's a one word. A stiff tissue impedes organ function because it just blocks certain aspects of the normal functioning organ, but it also creates a positive feedback loop where the cells producing extracellular matrix and making it stiff are activated by the very stiffness of this matrix to become the cells they are. So that's part of the fibrotic problem here. And this is just one of many, many examples you find in the literature. This is from our lab showing that if you grow fibroblastic cells in the same culture conditions on planar substrates with different stiffnesses or in 3D collagen gels, the level of activation shown by smooth muscle actin expression, in this case in red, is higher on stiff than on soft substrates. So mechanics are a main driver of the activation state. And there's going to be a session, and session 9, and looking at the speakers, I'm pretty sure there's going to be one of the subjects. Now, where do the growth factors fit in? Well, we all know that there are profibrotic growth factors, and this is just pretty much a random table out of one of many reviews that I could have picked, which show growth factors which are targets for antifibrotic strategies in clinical trials. And given the energy and the amount of time and money which went into this, I'd say the outcome is rather abysmal. So Tom will probably disagree, but or not, and you hear about him. And I'll just give you a flavor why that might be. And I'm going to pick one of all those growth factors which have been considered as antifibrotic therapies. I'll pick TGF-beta. For the reason, it's a very interesting growth factor, as you will see, in the way that the biology works, but it's also the most potent profibrotic factors that we know of. So you add active TGF-beta to your fibroblasts in a culture dish, they always invariably turn into myofibroblasts by expressing muscle-actin. Now, TGF-beta is a dimer, which is never secreted that way. It's always secreted at least in a latent form, with this propeptide, which is called LAP. So that's already unusual. In this form, it cannot bind to the receptors. And Dean Shepard's group had shown that integrins bind to the proform of TGF-beta and somehow activate TGF-beta. And then Rifkin's group has shown that in order for this activation to happen by integrins, TGF-beta has to bind on the other side of its latent proform to the extracellular matrix through a protein called LTBP. And our contribution to this space was showing that mesenchymal cells, myofibroblasts, express integrins which bind to latent TGF-beta in the matrix, and by simply contracting and pulling on this complex, they release TGF-beta, pretty much analogous to how you would activate or release a candy out of its wrapper, imagining that candy is TGF-beta and the wrapper is the latent form, and the pulling then leads to liberation. And they're obvious, if you just look at that scheme here, there's so many targets in this activation procedure, and integrins have been identified as being a more or less specific target. And again, it's Dean's group showing that targeting alpha V beta 1 integrin or alpha V integrins in general in a mouse model in fibrosis creates or protects from fibrosis and creates almost like a TGF-beta null phenotype without side effects, which we can talk about later. Okay, I'll speed up a bit. So what about immunity? I would claim, well, you don't need immune cells to get fibrosis, which is obviously not true, or inflammatory cells. But I like to stay in this universe of TGF-beta just to say that there's beautiful work from Sophie Lucas' group and followers showing that T regulatory cells also present latent TGF-beta, but not in a matrix, but rather in a membrane protein, which they then present to other T cells to regulate their activity. And Springer's group, Tim Springer's group, has shown that astrocytes present latent TGF-beta using a very similar membrane anchor to glia cells. So that's how we think some scarring in the brain may happen. And it really pains me for time reasons. I can't show you any data, but data in our lab shows that this might be a concept here which is reality, where macrophages carry latent TGF-beta on the surface and present it to myofibroblasts, which have the activation machinery to activate this TGF-beta. And that, I know my time is running out, brings me to the very last question or last slide, and just showing you a bit of data how proximity or location matters. So this is kind of a message to all the single cell RNA sequencers, which digest tissue and create beautiful profiles, and we lose the information of space. In this simple experiment, we just throw macrophages directly onto myofibroblasts, and we get beautiful activation of these fibroblasts into myofibroblasts, indicated by the amount of redness here. But that's not happening using the very same macrophages and fibroblasts by physically separating them and not letting them touch, but they share a medium. And you can drive this to the extreme where you just grow the cells on top of each other, give them different spaces, and only in the contact situation, you get a TGF-beta signal in the fibroblast portion of it. So proximity matters, and you may understand if you look at this slide here, why that is the case. And that's not only in vitro. We're not the first showing that macrophages and myofibroblasts, so the green are the macrophages, the blue are the myofibroblasts, in this case an IPF region of a human patient, are very close to each other. And while we force them in these experiments to come together, they have ways of sticking to each other in these fibrotic conditions, in this case by expressing adherence junction proteins, cadherin 11. And that's pretty much it. So if you forgot everything that I told you, just keep in mind myofibroblasts rule, and we're looking for people. Thanks very much.
Thank you so much, Boris. So we'll now take questions. We have a couple of questions from the audience. Why do you think that evolutionarily fibrosis would evolve instead of a regenerative process?
Oh yeah, that's a big one. Give me 2 hours.
I know.
I think fibrosis at the beginning is a dysregulated wound repair response. And I attended the panel discussions yesterday So the body can't regenerate. Either the injury was too severe or it's chronic. So we have to find a quick fix, which is scar tissue. So it saves us from dying. And then Mike can bring the predator thing again. So the saber-toothed tiger, which attacks the caveman. The caveman doesn't care about the scarring; he just wants to survive. So it's a matter of time, basically. It's a non-intended, misregulated wound repair response.
Right. And related to that, Claire Ng asked, what degree is fibrosis diametrically opposed to regeneration? So are there aspects of fibrosis, like stages, that might be maintained that allow us to regenerate?
Wow. Honestly, I don't have an answer for that. So it comes back to the question of reversibilities. I mean, even if we stop fibrosis, if we would find therapies that stop fibrosis, which we're kind of far away still, we're still faced with a pretty destroyed architecture of the organ. So we have to somehow find ways of reconstructing the organ. And as much as we know, at least that's my opinion, is it's kind of opposite. So either you regenerate or you fibrose. But in a normal wound healing, a normal tissue repair response before fibrosis happens, I think there's a way back and there's an equilibrium here.
Right. And then Andre Inova asked, is there a therapeutic potential of inhibiting actomyosin cytoskeleton in clinical fibrosis?
Yeah, well, I'll just shoot that for the panel discussion to Mike, and he and others. Groups have shown that if you block mechano signaling processes, but also if you use Rho kinase inhibitors, Fasudil, they are used in clinical trials at least. Problem with those is they're non-specific. So specificity is a big issue in fibrosis. We can throw anything at these fibroblasts and make them go away or deactivate, but those agents will have side effects. So if you block contraction in heart fibrosis, well, I let you think that through, what that's going to mean for the heart muscle contraction. So you'd have to find something which goes specifically to the fibroblast population.
Right. And so we have a couple of questions from the audience about TGF-beta. Do you think that there is a way forward therapeutically targeting TGF-beta? Is there too much crosstalk?
So again, I'll give this to Tom, but history has shown us that blocking TGF-beta globally is probably not a good idea in most situations. That's where this idea comes in to block either the presentation mechanisms of TGF-beta or blocking the integrins or other activation mechanisms, which seem to be specific to certain environments and cell types. So this is a much more directed way of blocking TGF-beta effects without blocking TGF-beta itself.
Great. So there are a few more questions, but just in the sake of time, we need to move on. Dennis, I hope you'll contribute to the broader questions later. And I'll hand it over to Michael Longaker for the next speaker.
Thanks, Valerie. That was very nice, Boris. Next speaker is Ruslan Medzhitov from Yale. And Ruslan's going to talk about some fundamental properties, I believe, that have direct impacts on fibrosis. Ruslan, take it away.
Thank you, Mike. Can you see my screen?
Yes.
Okay, so thank you for having me in this great meeting. I will talk a little bit on broader questions, mostly focusing on what we don't understand about tissue organization, homeostasis, and vulnerabilities to various diseases. So one motivation for our interest is that if we compare our knowledge or understanding at different levels of organization, we know what the common themes are in animal organization. So we look at these different mammals, and we know that these are all variations on the theme of a mammal, and we know what the basic design is and how different universal parts are connected to each other. Same is true at the cellular level. These cells look all dramatically different, but we are not fooled by that because we know these are all variations on the theme of a eukaryotic cell. But then if we look at tissues, then I would argue that we don't know what whether what we're looking at is also a variation on some common theme, and we don't know what that theme is. And that's what I think is a major question, major gap in our understanding of tissue biology. And one sort of conceptual roadblock, I think, here is that tissues are products of emergent properties. So the tissue structure, patterns, spatial characteristic, functionality, they're all emergent properties that arise through self-organization. And these are two familiar examples of self-organization, a flock of birds, a school of fish. Here, these structures emerge in a self-organized manner. And in order to understand them, it would not help to know precise details about positioning of each of these animals and their, you can sequence the hell out of them. None of that would help to understand how that emerges. But what is needed to understand how it emerges is that there's some general principles that dictate that emergent properties are products of interactions of individual agents of the system. And the agents interact according to a small number of simple rules. That's the kind of surprising aspect of these complex systems. And what is needed, therefore, is to understand what these simple rules are. And that is an achievable goal, and that would be the only way to understand how you get these types of structures. Related point is that the common confusion between the notion of blueprint versus self-organization. Buildings like this one on the right is a product of a blueprint. Every detail of the building has to be pre-specified, and that information has to be available before building is built. Biological systems like this termite mold, for example, are not products of a blueprint. There is no pre-specified information about what they will look like. They are a product of self-organization. And again, that is executed as an interaction between individual termites based on some simple rules. And many of these rules are, by the way, well understood now. So we can understand how termites can build this. And the principle here is very different from the blueprint principle. So in other words, what we need to understand is some of these simple rules of interactions between the individual agents of the system, which in our case would be cells within tissues. And that's what we don't know. So that's basically my message for today. And I'll just discuss some of the examples. So one thing that we want to appreciate is that there are different types of causality. This one is the one we most familiar with, and we try to feed the world into this type of linear causality model, which almost never exists, but it's something we can understand. And then there is nonlinear causalities, which are harder to understand, but still we can grasp them. Here, arrows indicate that A causes B and B causes A, for example. But what is most common in complex systems, like biological system, is emergent causality, where C, which is what we observe, is not caused by A or B, but it's caused by interaction between A and B. These are not causal links here. These indicate interaction between A and B. A and B could be different cells, genes, people, anything. And if we understand the rules of these interactions, we can understand how C is generated. If we don't understand the rules, no matter how much detail we know about A and B, we will not understand how they generate C. And that is basically what is a universal fact about complex systems and something that biologists need to really embrace. So I will talk a little bit now, a couple of examples of what I mean by some of the simple rules. It's just going to be like a small snapshot. So to understand tissue organization, we need to, as usual, it's useful to look at what are the more evolutionary primitive systems look like. And we know that epithelial mesenchymal modules are probably the most primordial units of tissue organization that are present in all metazoans. And not only that, but epithelial mesenchymal cells have a special type of relationship with each other, where mesenchymal cells commonly control the fate of epithelial cells. And this exchange of morphogens then dictates different types of developmental programs. And these classical experiments from Spearman and his students demonstrated that mesenchymal cells dictate the fate of epithelial cells, here dermis and epidermis. And what's critical here is that mesenchymal cells possess some information, in this case, positional information, that dictates the fate of epithelial cells. In other words, mesenchymal cells know something, and epithelial cells can do more than one thing, several options, and mesenchymal cells dictate which option to take. And if we formalize this in a more general way, we can say that cells can be divided into M-type and E-type. They don't have to be morphologically mesenchymal or epithelial. It's just that they would be defined by M cells having some information relevant for fate choices by E cells. And E cells decide what to do based on that instruction from M cells. And these are several examples that fit into this type of paradigm. And so another point I want to make, a last point, is that tissues are organized, made up of multiple cell types, but the basic foundational cell types for, at least for vertebrate tissues, I think are these 4 types of cells that include a tissue-specific functional type of the cell responsible for core function, like commonly epithelial cell, for example. And then there are 3 types of cells that are nearly universally present that perform supportive functions, this microvascular endothelium, fibroblast, and macrophage. And one last point I want to make here is that to understand tissue organization, we really need to understand how, since every cell type requires a specific growth factor that dictates whether a cell survives, dies, or proliferates. And so local production of appropriate growth factors dictates tissue composition. And what I want to say is that we don't know the rules for growth factor production. Surprisingly, we know very, very little about that. We know a lot about signaling, biochemistry, genetics, but we don't know the rules of growth factor. How do these cells know which growth factor to make, when, and how much? And the answer to those questions would dictate what kind of tissue composition you get in the end. And so we studied that, I'll just show one example. We studied that with macrophages and fibroblasts, where we could have a system to analyze how cells interact. And we found this type of a circuit affords a stable relationship between macrophages and fibroblasts. And what we then realized is that fibroblasts follow and macrophages follow these 2 distinct strategies to control cell numbers. For fibroblasts, it was dictated by space availability. So cells divide until they fill up the space. This is true for epithelial cells and many other cell types, but of course, this is not true for hematopoietic cells, where they're not restricted by space, but instead the cell numbers are restricted by growth factor availability. And we thought that fibroblasts and macrophages actually represent sort of this paradigmatic example where one cell type limited by space, the other is by growth factor produced by first cell type. And what we then asked whether this production of growth factor is a function of space availability sensed by fibroblasts. And we found that that's indeed the case, that production of CSF1 and many other growth factors is controlled by space availability sensed by fibroblasts, in this case through HIPPO pathway. And we found that there is this space or density sensitive enhancer that is fibroblast specific, activated, regulated by HIPPO, and controls production of CSF1 for macrophages. And that can be expanded then to many other cell types. We found there are 3 classes of density-dependent genes in fibroblasts: HIPPO-dependent, TGF-beta-dependent, and Wynn-dependent. And then we know also other examples that fit into the same kind of paradigm. Here, the space availability sensed by fibroblasts to control CSF1 production for macrophages, thereby automatically adjusting the numbers of macrophages and fibroblasts as a function of available space. In this example, a more familiar example, oxygen availability through HIF pathway controls VEGF production and endothelial cell numbers through angiogenesis. and then some additional factors like nutrient sensing, ECM sensing, and so forth, then work to control production of additional types of growth factors. And we know that there are a whole number of growth factors are regulated in this manner. And so this is my last slide. These are some of the questions that I was typing in the last moment. What are the rules of cell interactions resulting in emergent properties of tissues? I think that's the key question. What are the rules for growth factor production? There's surprisingly little understanding of that. And finally, what are the vulnerabilities in tissue design that result in fibrosis, degeneration, and neoplasia, which is 3 types of diseases at the tissue level. And finally, Ruth Franklin and Xu Zhao were former postdocs who now have their own faculty positions who did the experimental work, and Miri Adler and Maria Loan were collaborators. And I'll stop here.
Thank you, Ruslan. That was provocative. There's at least some questions coming in. Let me just think. How do you propose studying emergent properties if your experimental system's largely focused on measuring predetermined variables?
Yeah, so to study emergent properties, we need to know the rules of interactions, right? So what I showed is one example that relates to rules related to growth factor production. So here we focus on this one question and find that, well, there is this couple of rules that dictate that first of all, that there is a symmetry between cells involved in cell circuits. One is restricted by space, another by growth factor produced by the first one. But this would be one example. So if we combine it with other examples for different classes of growth factor and determine what controls their production, this will contribute to understanding what are the rules of the exchange between them. Then one can study dynamic properties like stability, robustness, and so forth. And this would be kind of building blocks. But expanding that to other types of information and rules like cell-cell contact versus ECM contact and so forth, this would be all building blocks that eventually kind of will create a library of the rules. And that's what would be needed to understand emergent properties.
And then maybe one last question. Do you think tissue history, injury, infection, et cetera, could change the rules of tissue organization, not only temporarily, but permanently?
Yeah, I think so. And actually, Shruti has a very beautiful example of that. So tissue history, to the extent that for different rules can be tuned by prior experience, and there is a memory component that definitely can affect how tissues behave in the future. The question is whether it's true for most tissues, for some tissues, and so forth.
Okay, thank you, Ruslan. I think we'll continue this in the panel discussion. And now I'll turn it back to Valerie to introduce Tom Wynn.
All right, our final speaker for this session is Tom Wynn from Pfizer. Maybe we can start with Dennis Disher's question.
Yeah, can you hear me?
Yep.
Yes.
Yeah, you can still hear me. Okay. Hello?
Yep, we can hear you.
Sorry. Yeah, thanks. Yeah. It's actually, it maybe sounds like a technical point, but the world is increasingly filled with single cell RNA-seq, and Boris brought this up, or at least I interpreted it as such, right? Where that doesn't provides information on cells, but not so much this sort of microenvironment, right? The fibrosis, the matrix, right? Is a fibroblast nearby and making matrix nearby a cell that it influences. So am I wrong in thinking like that? Is there a solution? Is there tools to be built to make those connections with a very cell-centric kind of measurement that increasingly is accumulating there? Have you thought about that? Does that make sense, Boris?
Boris, do you want to handle that?
Can I pretend I didn't hear Dennis? No. Yes, there are tools. I think probably as single cell RNA sequencing is obviously a great tool. I don't want to diminish that. we'll need spatial information at some point. And I think tools are developing down that way. So I don't know, cell painting, DNA painting, spatial omics, where you do single cell RNA-seq, or antibody detections with multiple targets on a slice of tissue, for instance. I think this is where it's going to go. But like any technology, as great as it is, single cell RNA-seq just has its limitation in the way we can interpret the data. Just have to be aware of it. There's nothing wrong with it.
But most of the atlases being built lack that information. Fair to say. I mean, you're not. So you have to. New tools are required. So you can't take one of the standard single cell RNA-seq where there's dissociation of tissue and sort of infer something about the fibrosis matrix-based interactions between cells. Is that fair to say? It's really just limited.
I would carefully say that's true. What do you say?
Okay. Yeah.
I think there are some hypotheses that you can build from those about, how cells interact, and then you can go experimentally test that. But let's move back to Tom now that he's got his presentation up. Thank you for that question, Dennis.
Thank you.
Okay, sorry again for that technical glitch, but I did figure out how to get it to project from my computer, so we're good to go. Great talks by Boris and Ruslan. We had a really good talk about what is fibrosis and what's important. And Ruslan has some really cool basic science going on in his lab always. So it's great to see that. And it's great to see those fresh perspectives in the fibrosis field. So I thought, since I moved to Pfizer, so I'm having more of a drug development approach to my research now, I thought I would give an overview of translational research priorities, at least my view. And so I have to disclose that I am an employee of Pfizer, and my views are not necessarily Pfizer's forward-looking statements. So fibrosis, shoot, I was going to try and see the timer. So maybe, oh well, my pin is lost on my timer, so someone's going to have to keep me straight on time. Fibrosis, and we've touched on this, is both good, bad, and ugly. And I'm going to leave you hopefully with 8 key takeaways from the talk. Just a bit of a background about what is fibrosis. Of course, fibrosis is an injury to a tissue. The goal is to heal that tissue. There's an inflammatory response for sure associated with at least the early stages of fibrosis. And the goal of that inflammatory response is to induce various growth factors that educate endothelial cells and reform blood vessels and allow reepithelialization of the tissue. And that's great. When you have an acute injury, our bodies are pretty good at repairing those injuries. The problem comes from when you have repeated injury, invasion of microbes, dysbiosis, can set up a chronic inflammatory response that leads to a dysregulation in that repair process, accumulation of excess extracellular matrix components, and fibrosis. So that's the bad, but it's not necessarily terrible. We need to heal tissues. And in some situations, if you don't throw down collagen and plug that hole, you'll get invasion of microbes and you'll die from sepsis. So that's the bad, but it's also good. We need to have that kind of repair. When this repair goes on, the fibrosis goes on for too long in organs, it can ultimately lead to organ failure, organ dysfunction, and you need to get a transplant. Or you're going to die from loss of organ function. We have learned from the example of hepatitis C that we can reverse fibrosis, or fibrosis can be reversed naturally. You eliminate the pathogen. We can see some significant reversal in fibrosis. And I think there's a lot of interest now in understanding what are those innate mechanisms that drive the reversal of fibrosis. And there's a lot of interest in macrophages because they're produce a lot of enzymes in proteolysis that can lead to reversal of fibrosis. So one of my first key takeaways is, and lesson that we've learned over the years, decades, is broadly inhibiting inflammation is probably not the ideal solution to ameliorate fibrosis. And in fact, the second takeaway is that features of inflammation, they need to be exploited to reverse fibrosis and promote tissue regeneration. As Boris pointed out, the fibroblasts are sort of the center of the universe in this field. They are the key collagen producing cells. I think when I came in to this field, it's amazing to say 30 years ago, fibrosis was almost all focused on the myofibroblasts, the collagen producing cell. And there was the the following decades, there was a lot of focus on where those fibroblasts come from. Epithelial cells that undergo EMT or endothelial cells have now been described to undergo endo-MT. There are pericytes that line the blood vessels. There's resident fibroblast populations like the stellate cell in the liver. There's fibroblast-like cells that come out of the bone marrow. The idea in all this fate mapping work has been if we could identify the key fibroblast population that drives fibrosis, we may be able to target that population specifically and leave the other fibroblasts that are important in structure maintenance in organs alone. So really go after only that pathogenic population. So that's been the hypothesis. Can we target that pathogenic subset? And a really cool explosion that's happened in the past, 5 to 10 years is the introduction of these multimodal single cell approaches, single cell transcriptomics, proteomics, metabolomics. We can now understand the transcriptome, the proteome, the metabolome in single cells. And we can begin to integrate this information and get a very deep understanding of different populations of cells in tissues. And this has been a really exciting evolution or revolution that's been going on in the past few years. So takeaway number 4 is single cell omics approaches are deepening our understanding of the pathogenesis of fibrosis, but much more needs to be done in disease tissues. So there's a lot of work going on in cell atlases in different organs. We're trying to understand what cells, and Ruslan's talk touched on this. What are the cells that play normal homeostatic maintenance functions in tissues? The cell atlases are really revealing a lot of those important interactions in healthy tissues. I think the next stage, and this is already starting too, is understanding disease atlases. Now go into patients, human tissues, where diseases are happening and understand how those cell populations have changed. And then ultimately in drug development, we'd like to know how our drugs are changing that environment. So getting treatment atlases would be the next future wave, and that's also already starting. So a lot of really exciting stuff going on in this field based on these new technologies. And Ruslan also touched on this. I think spatial transcriptomics, which is also starting now, is really trying to understand, once we understand these populations in these different environments, in healthy, disease, or following a treatment, where are these cells located? Which cells are interacting? Can we identify those key cells that may be remodeling the matrix in specific regions? And that may be revealed through spatial transcriptomics and spatial proteomics, which are coming on board. I will add there are a lot of interesting stuff that's already come out of this work, a lot of exciting papers coming out in top tier journals. There's a really cool paper in Nature a few years ago about different populations of fibroblasts in the synovium of RA patients, unique populations of fibroblasts that were in the lining and sublining layer that drive either inflammation, cartilage and bone destruction. We've been fascinated by the differences in different muscle tissues, like the cardiac muscle, which is not very good at repair. What's different about the fibroblasts there versus fibroblasts that hang out in skeletal muscle? Why do those tissues repair much better? There are fundamental differences in the populations of fibroblasts in those different types of tissues. We've learned that fibroblasts are very closely linked to adipocytes, and they can go under phenotypic conversion through various processes. When the disease irritating agent is eliminated, these myofibroblasts can undergo a reversion process where they become more like a quiescent resident fibroblast population. We've identified transcription factors that can drive the repolarization of myofibroblasts into a more quiescent phenotype. And there's even exciting work going on. Of course, this has been done in stem cells, that you can take out fibroblasts and differentiate them into different populations. Can we learn how to do that in situ to turn a tissue damaging or pathogenic fibroblast into a cell that may support that organ or tissue? So like reprogramming to hepatocytes. So takeaway here is that fibroblasts are much more complex than previously appreciated, and they remain really interesting therapeutic targets. Again, Ruslan touched on this a little bit. Of course, there's a lot of cell-cell interactions going on. I agree with Boris, the myofibroblast is important, but in a lot of the work that I did when I was at the NIH, we showed that simply manipulating the phenotype of T cells could redirect the function of myofibroblasts and completely block fibrosis that develops in response to a parasitic infection. So there's different cell types that communicate and feed in to myofibroblasts activation, homeostasis, reversion, et cetera. We've been very interested in macrophages, in the communication between macrophages and fibroblasts. We've identified various populations that appear to be pro-fibrotic through the production of pro-inflammatory factors or growth factors. We also identified populations of macrophages knocking out specific genes that convert them into macrophages that suppress fibrosis. So they actually play an important protective role by slowing the progression of fibrosis. So understanding this dialogue, as Ruslan pointed out, is really important. And of course, we're interested in translation. So the mouse models are wonderful. In vivo mouse model validation is always really important. We're also trying to develop systems to understand the role of human cells. And I think these, so my next takeaway is that I think the development of these complex models, 2D, 3D organoids, spheroids, systems under flow, et cetera, are really important for being able to take human cells and cassettes that we think are really important. The cell populations are really important in fibrosis, and understanding how those human cells behave and interact to drive a fibrotic phenotype. So that's a really big area that a lot of work, a lot of companies have sprung up, and I think we'll see a lot of exciting stuff come out of that area. Again, as I mentioned at the beginning—
You're at 20 seconds. I just want to give you an update on time.
With 5 minutes for questions?
Correct.
Perfect. Okay, I have 2 slides. This second to last slide, I think. Macrophages, again, are really important. Different subpopulations. They're clearly populations of macrophages that get activated, produce growth factors that communicate to a lot of cells, like endothelial cells, like VEGF. They produce Wynns that can activate stem cells that participate in repair. There's populations, different factors that drive resolving macrophages that turn down inflammation, that allow repair to happen. So really understanding this complex interplay between these different populations of macrophages may allow us to slow the progression of fibrosis, but even more importantly, reverse fibrosis. So takeaway here is that macrophages are chameleons that exhibit very dynamic phenotypes, and controlling this may be key to controlling fibrosis and actually reversing. Fibrosis, I really don't have to put this up. I think this audience is well aware fibrosis is a big problem. A lot of people are infected. Most organs in our body where we have chronic inflammatory diseases lead to remodeling and development of fibrosis, and that impairs organ function. So it's a huge unmet medical need. I will say that, inflammation has been a distinct discipline for well over 100 years. I think fibrosis has only been recognized really as a distinct discipline and distinct mechanism and a distinct pathology for the past 3 to 4 decades. And I think there was a real awakening with the Panther trial, which was an IPF trial where 3 different anti-inflammatories were tried. Let's just really shut down inflammation, see what impact it has on fibrosis. Perhaps not surprisingly now, based on what we know, it made patients worse. So just targeting whole-scale targeting of inflammation is probably not the answer. We only have 2 drugs that have been approved for fibrosis, pirfenidone and nintedanib. Certainly not perfect, but a great first drug for patients with IPF. And then my last and final slide is takeaway number 8. What is my prediction for success with fibrosis in the coming years? Of course, number 1 priority for drug development is agents that slow progression, like pirfenidone and nintedanib, would be really valuable for patients. Of course, the next phase would be trying to reverse established fibrosis. That's going to be more challenging. And then even more challenging are finding agents that will induce tissue regeneration in situ. So just end there and thank a bunch of people. And thanks for organizing this terrific meeting. Happy to take questions.
Thank you so much, Tom. Thank you. So we have a question. Does injury-independent fibrosis exist from one of our panelists?
Well, I think NASH is maybe a good example. There probably is some kind of underlying injury to the liver. I think the working hypothesis with NASH now is there is an interesting dialogue going on between the gut and the liver. Of course, accumulation of fat is not alone sufficient to trigger the inflammation that can lead to fibrosis and severe disease. Birds develop fatty livers all the time. They do that for migratory purposes. They feed in the spring and summer to fatten up. They get really fat livers, and that allows them to migrate large distances. So that itself is not necessarily a bad thing. The bad thing is the inflammation that develops along with fatty liver. Why that happens, we don't still really fully understand, but that's not really an injury. It's feeding, it's excess fat. Somehow the gut gets leaky. We think LPS and bacterial components activate cells in the liver that lead to inflammation, that lead to fibrosis. So there may be examples. I think there's always some type of underlying injury that initiates it, though, some kind of event.
Right. Kevin Healy, did you have a question for Tom? I see your hand is up.
Actually, it was for Boris in the last session.
Okay, just hold on to that for our discussion.
Yeah, great, thank you.
We do have a question from Samir Baez. Would you consider cancer-associated fibroblasts as fibrosis? And how similar, sorry, I lost it. How similar and distinct are cancer-associated fibroblasts and fibrotic fibroblasts in a given tissue context?
I think that's a good question. I think that's sort of the second wave of transcriptomics, right? And is understanding these different populations in different settings. How similar or different are they? I certainly don't think we fully understand that. TGF-beta is expressed in a lot of tumors. There's a lot of interest in clinical studies going on inhibiting TGF-beta in some solid tumors. Some ALK5 inhibitors are in clinical development. So far, they seem to be, the dosing regimens, et cetera, seem to be okay in those patients. There's not a lot of tox, and they're being used in combination with checkpoint inhibitors. So I think there's a lot of similarities between the CAFs and tumors and fibrosis more generally. But I think until we really do those comparisons, we're not going to know how similar they are. It's a good question.
Thank you, Tom. So I think we're at time for your discussion. We'll now move to our broader discussion with all of the speakers. Okay, great. Thank you guys so much. This was a really very interesting, and I liked how your talks really connected with one another to think about the tissue level. One question that I've always had is, how can we understand the ECM more? So this is clearly changing in fibrosis and in tissue repair, but we don't really understand how it's functioning. And so what do you think are the tools, or how can we further understand the ECM and these processes?
I can probably start with this. That's a killer question, obviously. So I kicked out a few slides of my presentation, which was one of a Ferrari and then of a Ferrari compressed by a garbage press or a metal press. They have the same, if you do mass spec on those two Ferraris, they give you the same result, but the organization is very different. So even with similar or same composition, the organization state makes a difference. And then there are a number of proteins, take fibronectin as an example, where again, mechanical factors or proteolytic cleavage, you name it, create different signaling events, and we don't grasp them. So we have no idea in which mechanical state and which of these signaling portions of the molecules are open or accessible or not. There are now antibodies which recognize active or cryptic epitopes, which may help, but that's a very complicated question and answer.
Yeah, I'm staying away from it. Matrix is so complicated. I mean,
everyone's going to go,
if I started all over, that would be an entire career. It's kind of like where chemokines were in the very beginning, where it was so confusing with so many different chemokines communicating to so many different things and so many receptors and commonalities between the chemokines and the receptors they use. It's very complex. So I think we're paying attention to the outcome rather than the nitty-gritty details of the matrix.
Yeah.
Well, I have a good one, sorry. Because I'm always dissing single-cell RNA-seq, but that's not entirely true. So if you profile those cells and you find profiles of receptors for a certain matrix, or that would tell you something, or might tell you something about the environment that these cells are in. So if you find somebody in a tuxedo on the street, you may draw some conclusions where that person is coming from and what kind of environment that person was exposed to. So it's not, well, obviously it's deductive, but
it's a great project for systems immunology, right? I mean, you could create a whole career based on publicly available data at this point.
Yeah.
Valerie, I just wanted to put out there that we uploaded a couple papers to bioarchives that look at single cell transcriptional activity, single cell attack, and then merge those with Archer, and then do Visium, and then impute spatial epigenomics, both in the context of cancer, mouse and man, and wounds in 2 different regions. And I'll simply say that there's less than 7 types of cancer-associated fibroblasts in mouse and man when you do that analysis, which is quite a bit different than the field keeps reporting on. So I think we're getting the clarity in 3 dimensions. And maybe Hal Dvorak is right or not right. 30 years ago, are tumors wounds that don't heal? Looking at that through the modern thing, I do want to ask the panel, though, wounds are stiff, fibroses are stiff. Is that a chicken or an egg? And how do we reverse that? In other words, remodeling is not well understood at all. The stop signals in repair, keloids being a spectacular example where they're not there. I'm just trying to throw out concepts that I don't think we understand well.
I think it's the timing. I think, when you look early, most fibrotic models are very inflammatory. I think that was debated a long time, like particularly in the IPF field, where they were looking at biopsies that were very late in disease. And that's probably the problem, is that when you have fibrosis, of course, it's not vascularized, it's fibrotic. There's no inflammatory cells there. So there was this hypothesis that it was not inflammatory. I think that's wrong. But I think what you need to do is actually get the inflammation back in there and remodel the tissue with the right cells. It's a matter of timing.
Tom, how early is a fibrotic response different than a regeneration response, or any of the panelists? And how a scar is born, how early does that happen?
I'm not sure I really understand the question. I think that regeneration is happening all the time. I think the goal of that whole circuit, like I showed, I think in one of my first slides, is that the tissue is trying to heal itself. The problem is that you get microbes that invade, you have this chronic stimulus. It can never go completely into that regenerative phase, and it drifts off into more and more matrix deposition because it's trying to fix a bigger and bigger problem. And schisto, which I studied for years, a parasitic infection in the gut, is a great example of why I think we have fibrosis evolutionarily. This parasite lives in the vasculature of the gut, produces hundreds of eggs a day that migrate across the lumen into the lumen of the intestine. So it's literally punching holes into the wall of the intestine. If we didn't throw down collagen quickly and plug that hole, we'd get bacterial invasion and you'd die from sepsis. That's not a very smart parasite. So I think our ability to fibrose and heal it, keeps people alive for 20 plus years. That's all that Mother Nature cares about. It doesn't care that we live to 80. She only cares that we live to 20.
I was thinking in terms of more of the acute response in a wound. How quickly do you go down those processes, and can you switch back and forth?
Oh, I think you can. I think we've even learned through fairly advanced liver fibrosis, at least, that fibrosis can be reversed and the tissue can regenerate. It's, I think understanding why, and that's the topic of this symposium, is why some tissues are good at that and others are not, is still really fascinating and not well understood. I think the liver, I think these organs like the gut and the liver that are, liver is a filtering organ, the gut is dealing with microbes all the time. Our lungs are okay at regenerating a little bit following pretty massive infections. Those tissues that are under insult all the time have to be good at regenerating or we won't survive. The other tissues, like the heart, other tissues, maybe it's just not as important because they're not bombarded as much.
I do want to give Kevin Healy an opportunity to ask his question. Kevin?
I just wrote it to Boris. I think we can handle that, but I do want to maybe just highlight lecture. Can you hear me?
Yes.
Session 9, which we will address the matrix a bit and some other issues I think that are coming up. So I want to make sure people hang on. And it's much longer discussion than just we have here. But clearly the matrix can be manipulated synthetically, can control both what we think fibrosis by just actually having a physical presence in the wound space to prevent prolapse, right? And so rather than chemically, maybe think a little bit differently, chemically targeting the prevention of fibrosis, you possibly could do this with a combination of a physical entity. And this is a case, and of course, for examples of trauma, not other cases.
So, well, if you don't mind, the question is there a marker for myofibroblast which is definitive? That's an easy answer. If you ask me, no, there's none, and that's a big part of the problem.
Okay, Boris, thanks. I was that was my question, yeah. and we have struggled with what are markers? What are the best markers? How do we determine a fibroblast? In our systems, right? And I'm glad I'm getting a response I expect from an expert. So good.
It's kind of like, how do you find an epithelial cell? It's, there's very many different markers for different ones, depending on where you're looking, I think. I'm wondering if we could talk a little bit about what kind of technologies do you think we need in this fibrosis field to move it forward in the next 10 years? What do the panelists think about what would be helpful for your work?
Well, I tried to touch on that a little bit in my talk. I think that these, I'm very interested in human translation, what's really going to work in people and not just in a mouse model. It's very hard to model something that really is a chronic disease, usually over many years before a patient presents at a clinic. How do we best model that? And the things that happen in our model systems, they haven't been that great at translating. I think Boris pointed that out in his talk. That's very true. So I think these model systems that are being developed with human cells is a big step forward. They need to get better and better and more reflective of the human situation. I love what's going on in omics. It's the great thing with sequencing the human genome is that we can now do this, and we can get, and single cell technologies keep improving and become cheaper and cheaper, hopefully. And putting all this information together. I mean, if I started my career over, I would definitely be paying attention to systems biology, because being able to interpret large data sets is going to be key to your success in the future, because all this information is going to keep flowing at you. And I really mean it. You could start a whole career based on the publicly available data at this point, and that's only going to get better and better. I'm not saying you don't need wet work. You certainly do, but that's why the model systems are needed. But I think that, taking the cell atlases, which is a huge effort by many groups, now taking them into disease atlases and then treatment atlases, I think that's going to be really helpful. It's all expensive, though. Ruslan, Boris?
A technology that wouldn't help me, but patients, kind of alluding to what Mike said, or the question is how early is fibrosis developing, is detection method, meaning medical detection methods, diagnostics to put a patient into some kind of a super MRI and be able to detect the fibrotic lesion in the lung at a time when it's still not repairable, but stoppable without too much loss of function. Because I think, Tom, you said it, when patients present in the clinics, it's already beyond the state of repair. So early detection would greatly help here, using possibly the matrix as an indicator.
Yeah, I would just add that related to Valerie's previous question, ECM seems like the hardest question currently, because none of these technologies really directly address this question. But what the kind of technology that might be helpful is to be able to visualize different ECM components in situ in order with enough resolution and granularity to try to discern the patterns of organization, how different components relate to each other. And I'm not sure what kind of technology that would be, but some kind of probes that would be ECM protein specific, where you can look at multiple at a time.
Yeah, people are using that for diagnostics now. There's MRI probes that are being used to stage fibrosis, and I think that'll be increasingly adopted in the clinical studies, at least.
And there are AI neural network analyses of ECM that profoundly predict outcome in things like pancreatic cancer. So it's shocking that the degree of fibrosis would be more important than anything else and how those patients do. So these things, I think, are going to be opened up, but it's a baby step.
Yeah. I do think that, one of the missions of this meeting, I think, was, where might Genelia invest? I would say, they have such amazing expertise in imaging that this, really bringing this field forward in spatial technologies would be super and a real place for them to invest. Because we're already seeing that in our own work that, transcriptomics is great, and applying it to single cell is great, but understanding where they are and how they interact is so much more revealing.
I want to ask one question, having Tom and Boris here, something I don't quite understand. So following injury or aging, there are two types of degeneration you can have. There's fibrotic degeneration, and then there's fatty degeneration. So, and I think Anastasia mentioned that in her talk about the difference in bone marrow with aging between different species. Question is, what dictates the choice between fibrotic versus fatty, whether you have expansion of fibroblasts in ECM or adipocytes?
Val, you should take
Valerie
that.
probably.
And Valerie, there's a two-way street there, isn't there?
Yeah, I think it's the signals in the tissue. So in the skin during age, it atrophies. The adipocytes kind of go away with age. But in other tissues like the bone marrow, it gets more fatty. In skeletal muscle, with age, there's more fat formed from the fibroblasts. And so I think it's with age, there's dysfunction in the signaling that pushes the fibroblast progenitors down the different developmental lineages, is my hypothesis.
But outside of aging, under normal conditions, these diseases presumably all reflection of some normal process that became dysregulated. Under normal conditions, what would it be? What information will dictate which path one would want to go, adipocyte versus fibroblast?
I think it's signaling from different cell types in the tissue that drive the differentiation process. But I also think the skin is a great model for fibrosis where adipocytes are lost. And that happens in a lot of fibrosis systems. And we have some data that's unpublished that suggests there are mechanisms that drive that process. So I think there's interaction in the tissue as adipocytes become dysfunctional during fibrosis. But we could talk about that over a beer sometime, Ruslan.
And Ruslan, Chuck Chan has a paper coming out in Nature that talks about the stromal changes with aging in bone marrow and how inflammation over time dictates why your marrow becomes fatty. But I do want to say, as a plastic surgeon, one of the benefits of aging is you lose your elasticity in the skin with sun damage and heal with 1/20th the amount of scarring. So should you live to be 95, you'll heal very well, Ruslan.
You don't get chronic wounds.
I think we're at time or over time. Thank you guys so much for this great discussion and session. It was great to be a part of it.
Thanks, everyone. That was great.
Thank you.
Thank you.
Thank you all. We will go ahead and take just a short 5-minute break right now and resume at around 1:06 p.m. Eastern. All right, everybody. I am very pleased to introduce Samir Bayaz from Cold Spring Harbor Lab and Ruth Franklin from Harvard Medical School, who will lead session 8. So over to you guys.
Great, thanks Janine, and thank you so much to the organizers for giving me this opportunity to participate in such an exciting meeting. It's been really great so far. So welcome everyone to this session on inflammation. Our first speaker will be Angelica Gonzalez from Yale University, and this is a really great segue from the previous discussion on fibrosis, because she'll be telling us a little bit more about the extracellular matrix and give us some insight on the important role of biomechanics.
Well, thank you for allowing me to present my work here today. As Ruth just mentioned, I'll be talking a bit about the extracellular matrix and really this idea of human biomimicry that we can use to investigate inflammation. And you'll see that there's some cross work that is relevant to the fibrosis discussion we just had. And so what I'm going to talk about is really this idea that our human systems are really complex, and complex in a way in which, you know, in the case of microvasculature, which is something I'll use as a model for our systems, and their interaction with the branching and peripheral vasculature means that they're also going to be embedded into an extracellular matrix that's usually not very well appreciated, as we heard from the discussion, or elucidated, as we heard from the discussion before. But even further, as we think about biomimicry and creating systems that look much more like humans, the complexity of even the basic structure. The microvasculature is composed of the endothelial cell, a mural pericyte that lines the outside of the vasculature, and then there's clear interactions with leukocytes here shown labeled with the L, that are migrating across both layers of the endothelial cell, a basement membrane, extracellular matrix sheath, the pericytes, in order to get into the interstitial space where they then enact their inflammatory behaviors. And what's really important here, thinking about human constructs or generating human-like constructs, is that we see clear differences in inflammatory diseases between mouse and man, and clearly the fibrotic models in which, as Professor Wynn described earlier, that inflammation is a precursor to fibrosis, maybe not the solution to reversal, but definitely contributory. And when we think about, as a biomedical engineer, when we think about biomimicry, we think about a number of things. The mechanical component means that we are really trying to get a system that is a substrate looking much closer to the human extracellular matrix or even the human basement membrane than we had previously developed. And so by different chemistries, different templating methods, different synthetic structures, we can get much closer to the human mechanics than we had before. We heard earlier from Laura Nicholson yesterday, our first speaker, who talked about what she's doing with her artificial lungs and regenerative tissues, but really she innovated the space of taking whole tissue, decellularizing that tissue, and what that has led to, as we think about biomimicry, is taking components of the whole tissue or cell-derived proteins specifically, and even peptide sequences within those exposed proteins to embed them into our synthetic structures in order to enable closer composition to the human matrix composition. And one thing that we've not explored as much as either mechanics or proteomics or the composition is really the architecture, which matters quite a bit. And when we talk about architecture, we're talking about the fibrillarity of the extracellular matrix, the pore diameter that is reflecting of the tightness of the fibrillar structure or the fibrillar diameter changes, and ultimately the effective pore area. Much of what we've investigated in vitro of cell-cell interactions has been done in transwell systems, and those, as you can see here as a comparator, do not really replicate the architecture of the human system. And so getting closer and closer to human biomimicry means that we need to take into account the mechanics, composition, and architecture all together to build something that's a bit more complex. And so what that means is that we can analyze the human basement membrane in this case, analyze the systems that we've been used to, either basic polymeric systems, polycarbonate transwells, and try to find some kind of synthetic way of developing matrix-like systems. The reason that we use synthetic chemistry is because that gives us the opportunity to include, to either be very reductionist in our approach of using peptide sequences, for example, or become much more complex with the whole tissue proteins. So it gives us a range of tools to play with. But really what becomes exciting about this is that we can take constructs and replace those transwells or replace the tissue culture plastic in a way that we have become very familiar with their uses, but use something that's much more mimetic of the human mechanics, composition, and architecture. So why do we do this, and why do we do this specifically with regard to inflammation? Well, inflammation in the animal looks very different from inflammation in the human. There's not the expression of, for example, in neutrophilic diseases, not the expression of IL-8 specifically by the endothelial cells. And one of the things that we don't see as often in the animal is this thickening of the basement membrane that is a remodeling event that could be seen as an injury or response to injury that is then repair. And in neutrophilic dermatosis specifically, we see that the microvasculature becomes really thickened in the basement membrane structure that facilitates myeloperoxidase stained neutrophils here in the remodeled space. When we evaluate the basement membrane specifically of a progressive Sweet syndrome or inflammatory microvessel, what we see is that that basement membrane becomes thickened primarily due to collagen 4 deposition. So a lot of collagen 4. Now this is different from what was described in fibrosis in that there's a large collagen 1 deposition. So what this suggests is that the pro-inflammatory remodeling event or reparative event of the microvessel is collagen 4 specific because that's a basement membrane protein as opposed to the collagen 1 enriched interstitium. When this remodeling occurs, it's not just the protein that is laid down, but the protein cross-links. You get other accessory proteins modifying the space so that you get a stiffened basement membrane that also changes its architecture in the formation of different pore sizes. Now, where engineering and in vitro analysis comes into play is that we can take human cells, in this case, the pericyte that's understudied in the role of inflammation, culture the human pericyte from the microvasculature and evaluate its response to the cytokines and chemokines that are being sent during this neutrophilic dermatoses. And what we see is that, in fact, in the presence of TNF-alpha and an IL-17A signal, that the pericyte is remodeling in a collagen 4 deposition manner. This collagen 4 then does, in this case of the human pericyte, cause an increase in the cross-linking density of the collagens, and thereby reducing the pore size of those that matrix. And when we then take the pericyte deposited extracellular matrix in the TNF-alpha IL-17A activated system, conjugated to matrices of different mechanics, we can see that the stiffening event actually does facilitate an enhancement in the neutrophil recruitment. So we see chemotaxis increase significantly when we have the stiffening event that was mediated again by the pericyte remodeling event. So what this tells us is that there is a true full and complex remodeling event that happens when the cells are activated by specific chemokines. Now, mimicking the role of the pericyte or the microvasculature on its own has been fun and very informative with regards to inflammation, but really we know that these microvascular structures exist within a tissue. And so because they exist, and we know they exist within a tissue, we are building extracellular matrices around these. And so with our collaborators, what we've been able to do is embed endothelial cells and pericytes into an extracellular matrix, and we can evaluate the role of these cells in propagating fibrotic remodeling after an injury. So here what I'm showing you is that we can take those models, trigger them with the TNF-alpha, and trigger them with a TGF-beta. And what we see is those pericytes will remodel the extracellular matrix to become enriched in collagen. And that subsequently activates presents a mechanically modified space. But what's really exciting for us is that not only can we induce in these artificial systems this disease state, but by treating it with nintedanib, which was mentioned in the earlier talks, we can actually get the cells to do a degenerative remodeling event that reduces the mechanical state of that extracellular matrix to something that's not significantly different from the healthy state. And further, we can get the cells to reduce their expression of myofibroblast-like markers, in this case, alpha SMA as one of those. So really what I'm trying to impart to you is this idea that mimicking tissues and mimicking the human microvasculature can really inform us about the role of inflammation in disease states. And as the last kind of example, what I'll show you here is that we can do this with brain tissue as well. If we're thinking about specific regions of a tissue, we know that the subventricular zone and the olfactory bulb, both in their architecture, their composition, and their mechanics look quite different. But by mimicking those structures and building out these models, activating them with pro-inflammatory cytokines that are being presented to the endothelial cell, we can then track and evaluate the chain-like migration of neural stem cells, which becomes really informative in their migratory behavior. And so as a conclusion, I'll tell you that we really believe that human biomimicry is key to understanding inflammation. It's very different in humans than it is in animals, so animals have been very informative, and that the environment matters. Composition, architecture, and mechanics of the extracellular matrix drive much of the cellular function, both in individual cells, but in multicellular components as well. And I'll thank you for your time and your attention.
Great, thank you so much, Angelica. While some questions come in, I'll go ahead and start. I just have to ask, in some of these models, if you've looked kind of in the opposite direction. So you looked, the effect of on neutrophil migration of the changes in stiffness, but have you looked in the opposite direction, the effect of adding neutrophils or adding monocyte-derived cells to see, what happens with that?
Oh, that's a beautiful question. And because we don't really think about the effector cells, the neutrophils really remodeling their space. And you're absolutely right. So in the skin model, what we found was there are a number of MMPs that are being generated by the neutrophil after their recruitment that is remodeling that space. And we can, slow down the recruitment by inhibiting, so through TIMPs or other factors by inhibiting that. But also, we think so much about the degenerative aspects of neutrophils during their degranulation process of migration. They're actually able to produce matrix remodeling programs, proteins that are regenerative. And so thinking a little bit forward, those might be the things we want to consider more, not just the enzymatic degradation of by these cells, but also their ability to remodel the space in a different way.
Yes, absolutely. I agree with that. I'm sure we'll learn a lot more about kind of these different functions. Maybe I'll go ahead and ask, we have Valerie has a question. So she says, is the ECM in this model produced by cells that you implant?
So we can do it both ways. So in some systems, we allow the cells to settle on a fairly naked environment, and their baseline then becomes the matrix that they themselves deposit. We can do it another way, which really is the benefit of engineering systems, is that you could give it what you want it to see right off the bat. So you could present a lot of collagen 4, for example, or laminins, fibronectins, or cryptic regions, as was mentioned in one of the previous discussions, cryptic regions of proteins that you want to use to send a signal to the cells, specific integrin binding motifs and such. And so in that way, you can really see, as I think Ruslan was hinting at earlier, what cells are presenting and depositing in response to their environment already. So are they dynamically remodeling in a collagen 4 specific way, in a collagen 1 specific manner, fibronectin, any of those proteins? We can start to track those things because we're signaling to the cells to see what their response to is going to be.
Great, and I'll ask another question. This might be better for the discussion at the end, but do you have any suggestions for researchers who really want to take ECM and biomechanics into consideration more in their work? Is it just simply when doing in vitro work, using substrates that are more similar to the tissue in the organism? Or I know this is kind of a broad question.
That's a broad question. So yes, I would say, well, be aware of what you're putting your cells on, right? So whether it's a matrigel, just a collagen 1, collagen 4, those are closer to mimetic, to a natural system. But also remember, when we are getting these cells out, we automatically throw them on tissue culture plastic, right? And so to some extent, there's going to be a memory. And what we're seeing is while we can get some reversal, are we really getting repair and regeneration? Like, is this true reversal in that the cells no longer have that sensitivity to TGF-beta, for example, or any other trigger? So as long as we're, slapping these things on some plastic plates, as soon as we get them out, we might not be able to completely mimic human systems.
Great, thanks so much. All right, in the interest of time, we'll move on to our next speaker, Paul Kubes from the University of Calgary.
Okay, thank you very much. Can you see everything? Is it all good?
Yeah, Paul, yeah, go ahead, put it in full screen mode, yep.
Yeah, okay,
Perfect.
Good. All right, so what I'll do today is I will share with you two issues. One is that inflammation is important for repair. And then my second part of the talk will touch on a new concept that really hasn't been addressed in the last 2 days, and that is something that I call border immunity. So we do imaging, and we just image tissues and try and understand what's happening. And so in the case of the liver, we will take a mouse, do a laparotomy, and that's it. And then we add just some antibodies via the tail vein, and then begin to visualize what's happening in the liver. So the blue are sinusoids, the purple are Kupffer cells at 480 positive. You can see this guy reaching out, this guy reaching out. They reach to their neighbors so that they know who they are. And during basement membrane deposition, these contacts are lost and these macrophages become quite different. Now, what I want to tell you about is just a really simple model of repair. And so, first of all, what I'll say is that this is a completely healed within 14 days. And anything that we've tried to improve on that has failed miserably. And I think that's because of millions of years of evolution pressure has just gotten it just right. So here's the model. We lower a thermal probe, touch the surface of the liver, and kill about 1,000 cells. Okay, so they take up propydium iodide, they're red, and then we asked, do neutrophils notice? And what you can see is over the first 2 hours, this spectacular neutrophilic recruitment. And people say, we don't see that. And I ask them, do you image? And they say no, and so I agree. Now, these neutrophils, they adhere in the sinusoids. They run towards these dead cells here in red. And we've characterized all of these events, and we've shown that the neutrophils run along the base of the endothelium. Chemokines are dispersed along in greater and greater amounts until they reach a point where the platelets actually surround this injury. These neutrophils run along the platelet carpet and reach this site. And formulated peptides from mitochondria function as important chemoattractants from these dead cells to get the neutrophils all the way to this site. The healthy tissue has beautiful sinusoids shown here. The dead tissue in green, you can see these vessels have completely collapsed. They're no longer patent. So these are nice vessels in the healthy area, some leakage of FITC albumin. These vessels are no longer patent. And the neutrophils come in and they surround these vessels. And then they begin to dismantle these dead vessels, this dead endothelium. And so you can see the blue neutrophils ripping apart the red endothelium. Here they've completely cleared it. So there's no more vessels. And now they start making these little tunnels for the new sleeves, for the new endothelium to grow in. These neutrophils, once they form these tunnels, they then notice these dead green cells and they begin to eat them all up. They never eat an entire cell. They eat these little particles, and I'll show you where those particles are coming from in the second part of the talk. You can use imaging to visualize basement membrane. So this is collagen. This is normal, healthy tissue. And you can see that during repair, there's nice collagen laid down. If you don't have neutrophils, even for the first and second day, you see absolutely no collagen deposition. That's because there's a bunch of stuff in the way and you can't lay down proper basement membrane. And if you can't lay that down, then you can't revascularize. This just shows the red sinusoids growing in. Without neutrophils, they will not grow properly. You can photoactivate neutrophils, shown here, and see them right at the site of the injury. Here's the healthy tissue, and then you can watch them. At about 16 hours, these neutrophils are done their job, and they start entering the vasculature and disappearing. So they all line up 1, 2, 3, 4, 5, and they're going home. They're punching their ticket. And this is also important. After 14 days, we see almost no injury. If you let the neutrophils or prevent them from leaving using various antiproteases, what you can see is that the injury does not heal. So it's important for neutrophils to come in. It's important for neutrophils to leave. And just as a quick note, other cell types obviously come in. Inflammatory monocytes, CCR2 high, lysic C high, monocytes will come in. They'll surround the injury. And then there's a second type of monocyte, a CX3CR1 high, lysic C low monocyte. Bottom line is these cells come in, but instead of going into the injury, these monocytes surround the injury. And you can see these nice rings by 48 hours, these beautiful rings of monocytes. And so that's just shown here. If you look at the less inflammatory monocyte, they form a ring as well, but it seems to be delayed by about 24 hours. And if you put reporters for both of these two cells on the same mouse, what you see is that these are the same cells. What happens is these inflammatory cells come in, they surround the injury, they get educated, they realize this is sterile injury, they slowly become less inflammatory, more anti-inflammatory, and then eventually enter the tissue. It takes 72 hours for us to start seeing markers of macrophage from these infiltrating monocytes. Here they are just starting to get F480 positive. 36 hours, they have no F480, no MERTK. That seemed odd to us. 72 hours for a macrophage to really start developing when you need repair right away. And that's why I want to introduce a second concept, border immunity. Here's Canada, here's the United States, and borders can be quite different. So you can see that some borders might be more difficult to infiltrate than others. Different characters will live at these borders. And sometimes you have a few border patrols, sometimes you got a whole bunch. And what I want to tell you is that the immune system actually lives around different organs at the borders. And so what we were puzzled by is when Jing Wang joined the group, she put a little bit of antibody on top of our injury, F480 antibody. And what she found was that within 1 hour, she saw lots of macrophage, okay? 12 hours, it was completely covered by macrophage. And these macrophage were not coming from the monocytes. They were actually these blue macrophage. They were not a monocyte-derived macrophage. They were coming from some other place. And if she did a 3D reconstruction, she found all these macrophages sitting on top of the injury. Now, Ruslan and two other groups actually showed that peritoneal macrophage have GATA6 as a transcription factor. And this allowed us now to track and see whether these new macrophages sitting on top of the liver injury were actually coming from the cavity, from the peritoneum. And indeed, they were all GATA6 positive. And I just want to show you an imaging of looking into the cavity at the liver after an injury. And so you can see the injury right here and watch these red macrophages within 13 minutes completely cover this injury, okay? And when we do a race between platelets, if we injure a vessel and the wall, the macrophage come in here just as quickly as the platelets. And these macrophages have been around for a very long time. Sea urchins, for example, they have a cavity. They have these platelet/macrophage-like cells that will plug up a hole the second the border is disrupted. And the phenotype of these peritoneal macrophages, one of the things these peritoneal macrophages in red do, and we're just looking on top of these macrophages now, here's a dead cell, and what you'll see is that they slowly rip apart this dead cell. All these little vesicles come off, and those neutrophils are eating up all of this stuff. So the macrophage from the border come in and help to degrade some of these cells. And you can see by 12 hours, this beautiful covering of the wound. And a lot of this is chromatin, histones. These are antimicrobial properties. And so what a wonderful way of covering that injury. If you don't have macrophage, you end up with this punctate, dead cells, no debris clearance, and you end up with poor revascularization. And we've now made a mouse where we can track these GATA6 macrophage. GATA6 disappears the second these macrophage enter the tissue. We've got a lineage reporter that tells us that at day 7, they are still in that injury doing something. And the peritoneal cavity is not the only cavity. We have the pericardial cavity. If you induce an injury, shown here, you get this nice scarring. We can't affect that. But if you don't have GATA6 macrophage, then the healthy tissue actually has a little bit more fibrosis. The heart's a little more stiff. And so these cells may be repair cells in a number of cavities. And in fact, the pleural cavity also has a very large population of these cells. Now, this isn't restricted to macrophage. There are many different cell types. I will tell you that the macrophage within the pleural cavity, pericardial cavity, peritoneal cavity are nearly identical. So these cells are in surrounding each and every organ. I just want to finish the talk by saying that a paper came out this month from Jonathan Kipnis in Science, and it beautifully shows this border immunity. So what he showed was that the skull bone marrow actually is a source, a very specific source for cells to come into the meninges, shown here, and then these cells may mature in this environment and then can infiltrate the parenchyma as needed, another border cell type. And so I think what we need in our lab to improve our work is to have more specific lineage reporters for all the different immune cells in a single mouse. I think that if we could image the way yesterday people were imaging fish embryos for a long time, it would be great. I'll stop there. Thank you very much.
Great, thanks so much, Paul. It was a really interesting talk. I'm just wondering what you think about, you use a lot of models of sterile damage. I'm wondering if you think that the repair that occurs in response to sterile damage, how it differs from, damage during infection, for example.
Yeah, so we have a paper under review on exactly that question. And there are some pretty profound differences. So for example, the monocytes seem to be very different as they differentiate in the context of an infection. The neutrophils come in, they actually gobble up all the bacteria, and I don't think that they actually leave the site. So that's another difference that we see. I think that the neutrophils in a sterile injury do leave. I think in an infection, they may just stick around and not spread. So I think that that's another difference. And then the other thing that we think happens, and this is also quite interesting, is that these border macrophage, these cavity macrophage, actually run away when they see an infection. So they're not as tough as we thought they were. They seem to disappear, and we're not sure exactly what they're doing. They may be clumping, they may be adhering to the vessel wall, but when we have an infection, it's hard to find them.
Okay. Great. And a question from the audience. Have you observed any differences between monocyte-derived cells versus tissue-resident cells in these different models? You touched on it a little bit just now, but do you see, clear differences between what kind of contributions they provide, in the different settings of injury?
Right. So this is a really good question. It's something that's occupied the field quite a bit. And I can tell you that people have looked at this in great detail. And so if you take a look at a macrophage and how it responds, for example, the Kupffer cell, okay, it has a certain inflammatory process with which it responds. If you deplete all the Kupffer cells and allow monocytes to come in and become Kupffer cell-like, in our hands, it takes about 6 weeks before that macrophage is pretty much identical to a Kupffer cell. And over that first 6 weeks, the monocyte-derived macrophage isn't quite as good at dealing with insults, and so it is more inflammatory. It seems to react in different ways to different insults. So it's not quite the same as the resident macrophage.
Great, thank you. And I guess we have time maybe for Ruslan. I see you have a question you want to ask.
Yes, thanks. Great talk, Paul. These parallels between platelets and GATA6 macrophages are really exciting. So in the case of platelets, we know that they sense the bridge by detecting the exposure of the basement membrane and through von Willebrand factor. In the case of GATA6 macrophages, is there some parallel mechanism that you know?
Yeah, so Ruslan, we've worked very hard to try and identify this. Hyaluronan, if it gets exposed, if the mesothelium comes off and you see hyaluronan, these cells have huge amounts of CD44, and we think that they will attach to the hyaluronan. But there's a lot more than that going on. They use scavenger receptors, and you probably know this sea urchin actually has thousands of scavenger receptors. We have only a few, luckily, and these seem to be also important for attaching to various molecules, negatively charged molecules, for example. So their attachment is, I would argue, a little primitive in the sense that they have they're not specific for a single molecule.
I see. Thanks.
Great, thanks. I'm now going to pass it over to my colleague Samir Bayez from Cold Spring Harbor Labs.
Great. Thank you, Ruth, and thank you, everyone, for joining us today. Our next speaker will be Mark Davis from Stanford University, and he will tell us a little bit about tonsil organoids that his lab pioneered recently, hopefully to model inflammation and regeneration one day. So, Mark, I think you're ready.
Sorry, I was muted. Okay, I'm good. Yeah, okay, right. I'm trying to relate this to inflammation, of course, and we have this is a slightly different take on it, but I think it'll be important in the not so distant future because there's an enormous number of chemokines and cytokines that are active in the immune system, and it's really one of the principal communication strategies the immune system has, that how the cells talk to each other and how they interact with particular tissues, and of course in times of infection or stress, injury and so forth. And we also know that a lot of these activities are important for a productive immune response, but there are any number of pathologies that involve too much inflammation. And I think also we have to move to a better way to describe these things, because just lumping 140 cytokines into something called inflammation is we can do better than that. And so I think we need to maybe develop a more specific nomenclature for the different inflammatory processes. I mean, there's already a well-developed literature on inflammasomes, but we could, I think, do more precise things. But also, I thought it would be useful to just relate this to aging, and we know that inflammation correlates with aging, and some people have more inflammation than their particular age group, and some people have less. And this is work David Furman in my group, and now on his own at the Buck Institute, had just came out with a really nice paper about this. And particularly, I don't know this is happening. Okay, particularly relating inflammation, a particular inflammatory score he developed, with all sorts of diseases and comorbidities. But the good news is that if you're a centenarian, we just talked about, getting to 95. Well, why not 100? Why not 105? And it turns out that centenarians have much less chronic inflammation. So I think that's the key, or maybe part of a key of what's happening. My favorite story about the older people, though, is the woman who was supposed to be 123 or 2, and they asked her what was the secret of her long life, and she said, "Well, I don't know, but when I was 110, I gave up smoking." So there, I think that's an important lesson for almost none of us. So what we've done recently, and this has been heroic work by Lisa Wagger, a postdoc in the lab and now on the faculty at UC Irvine, and just came out earlier this year, is to take advantage of an abundant immunological source. It's not easy to get lymph nodes or spleen from human beings, but it is easy to get tonsils. And tonsils are basically these large pair of lymph nodes in your throat that are sampling pathogens that are coming in that way. And hundreds of thousands of these are whacked out every year in the US alone, mostly from innocent little children, but also from adults up to, we recently got a tonsils from a 70-year-old. So it's possible to look across a very broad range. There are billions of lymphocytes in tonsils, so it's an enormous amount of tissue. The most lymphocytes you ever get by about 100-fold from a little kid, for example. But the important thing is that Lisa worked out a way of just a very simple dissociation and culturing system and transwells to reproduce basically all the major components of an adaptive immune response, particularly using vaccines, and our go-to vaccine has been the live attenuated flu vaccine, but we've also used other vaccines and generally get good results. These things mimic what we know happens normally in vivo, which is that you get germinal centers, especially with the antigen exposure and mobilization of both innate and adaptive immune cells. We get dark zones and light zones in these germinal centers, which is one of the hallmarks of those. And the germinal centers are important for both promoting somatic hypermutation in antibody genes and then selecting for the higher affinity antibody producing B cells that's still on the surface, and then ultimately they mature into plasmablasts and plasma cells and secrete those antibodies. So we get antibodies, we get specific antibodies in most cases with flu, but I said many others as well. This experiment is particularly cute, where we can remove all the high affinity flu specific B cells in the culture, stimulate with the flu vaccine, and then they come back, or they don't really come back, but lower affinity B cells that are still there mutate and are selected to be high affinity, and then you get back to where you want to be. And we can see that this on the sequence level as well, where if we've removed all the high affinity B cells, we find others that are 5 amino acids away from being high affinity. And then that happens, you get a lot of them. And you also have heavy chain switching. So you go from the IgM producing antibody to IgG or IgA pretty regularly the longer you go on. They're short-term cultures. These are not long-term, and basically they start pooping out at 2 weeks, but that's enough to go this far in terms of producing an antibody response. We think this has a tremendous potential for understanding the inflammatory circuitry in the immune system. And of course, the immune system is responsible for a lot of this. And you could also introduce other tissues as well, but mainly just picking apart who's doing what to who with what cytokine is important, just just to keep track of things, and so it's all there in the dish. And you know, Paul showed those gorgeous imaging pictures, and I hope we can see such things in this system in the not so distant future, because that's so powerful. But also that because it's in vitro, and because you're basically, you're mimicking pretty accurately a normal event, having it in vitro just gives you so many more degrees of freedom to understand what cells are producing what cytokine in what context. For instance, in the vaccine business, we know that various vaccines and adjuvants have been rejected because they trigger too much of a cytokine response. So it's not too distant from promoting a productive inflammatory response to a destructive one, and there's no reason why we couldn't do that here. And I would also say that since we know that inflammation of a sort, chronic inflammation, is a feature of aging, the fact that we could compare 2-year-old tonsils with 70-year-old tonsils could help to get at mechanisms in that particular circumstance, and learn something about it. Probably we won't get tonsils from centenarians, but I was thinking of hunting some down someday and seeing if they'd be willing to contribute. So that's it, really. I just want to introduce the system and have people think about possibilities and maybe using it. It's a very simple. Every hospital is doing a business in taking out tonsils, and I should also say that whereas in the past tonsillectomies were performed largely for tonsillitis, which is massive inflammation and infection of tonsils, that's very rare now with the advent of antibiotics and so forth. And so now mostly, the only tonsils we use are ones that are taken out for sleep apnea, where the little children have trouble breathing at night, as do adults, quite common. So even Stanford, it's not a big hospital. We typically get 1 or 2 or 3 tonsils a week. So it's by far the most easily accessible and abundant type of human lymphoid tissue. But we have also tested some lymph node and spleen tissues and done the same thing and gotten the same response. We haven't looked at them in depth, but it's something we will be doing in the future. But I'm pretty sure that they're going to be very, very similar, and so forth. So anyway, it's important to thank Lisa Wager, now on her own, and who did so much of this. Ben Wendell, Lauren Judd is now at the University of Washington as a medical fellow, and many other people in the group, and particularly David Furman has really educated me a lot about inflammation and aging. And so I'll stop there. Thanks.
Thank you, Mark. That was very good, I think, and for our session to show what we can do, hopefully, in the future to study inflammation and immune system in the context of tissue regeneration. So I'll start with one question as people type in their questions. How easy to propagate these organoids, or is it you take it from the patient, they self-organize on the dish, and then you do one experiment, or can you propagate those?
Well, even more importantly, you can freeze them. You get so much material. If you get 1 billion cells, you freeze them, and typically we freeze them into 50 million cell aliquots. And just in the normal way, you freeze down PBMCs, for example, and store them in liquid nitrogen, and so they're presumably will be good for years. So yeah, so it's basically basically you're slicing and dicing and pushing tonsils through a mesh. So it's a tedious procedure, but it's not difficult. It's a lot like other single cell dissociation procedures. And then you get a lot. And we're just started to try to get getting some spleens, which are much more limited supply, but an adult spleen is the equivalent to like 10 or 20 tonsils. So we have hopes to have a vast collection, and then you could do multiple experiments on the same donor.
Yeah.
Because there is tremendous human variation, and getting a grip on that variation is
great.
important.
I think Michael has a question. So Michael, do you want to ask it?
Yeah. Hi, Mark. It's Mike Longaker. That's really interesting. Children heal with a fairly robust fibrosis and scar formation. Is there any way to evaluate their relative immune system compared to that 70-year-old in terms of potentially igniting fibrosis in your system, or take cells out from your system and go in vivo?
I think with your help, we probably could figure out how to do that.
Well, we're colleagues. It's always good to see each other on Zoom, but we'll follow up offline on that. Thank you.
Yeah, we can even actually meet physically at some point.
Don't get carried away.
There's
an immune system going on here.
All right.
So I think we can go to the panel discussion session now. Thank you so much, Mark, for this great presentation. So I think you would appreciate that inflammation is a very complicated concept, and Mark alluded to this in his talk, that nomenclature probably should be revisited. But what I want to start the discussion with all of you and other panelists, I think, can contribute, is how inflammation can be a trigger for the regeneration and repair process. And in some contexts, I think we have seen this yesterday, especially. There were some remarks that were made about what are the upstream signals that start the regeneration process. So we have a diverse group of people here: immunologists, engineers, stem cell biologists. So what are your thoughts on how inflammation play a central role throughout this regeneration and repair process in a given tissue? And apologies, we have also Bobby, who's a young rising star who will be speaking on session 10, and he is working on how IL-22 signaling influences regeneration. Okay, so any thoughts?
I guess one thing that I've thought about quite a bit, was mentioned earlier as well, is that there's always remodeling occurring. So there's a homeostatic remodeling event that is going on, especially with regard to the extracellular matrix. And it's when that system gets out of whack or imbalanced that we start to see problems that are more fibrotic or scarring repair mechanisms. And with regards to inflammation, a lot of that is probably happening when we have unresolved or chronic inflammatory signals. For example, when we're looking at, we study a lot the vast microvascular cells, the pericytes. What we see is that if we have a prolonged TNF alpha activation, then we have a doubling of the TGF-beta receptor. So when that happens, or that chronic and persistent inflammatory response occurs, then that may be priming or sensitizing the cells to a reparative or fibrotic event. And so it may just be an imbalance at one point or another. And what that means for regeneration is that if we are not getting these appropriate signals to shut off and trigger appropriate matrix remodeling events, then you're going to have that aberrant scar as opposed to the healthy remodeling.
I think so. Like I study intestine, and in the intestine, you see a very dynamic plasticity of differentiated cells upon injury, assuming roles of tissue stem cells and have a unique cellular state that define those. And so this is observed in many injury models. So if you irradiate the mice, if you give an infection, if you kill the stem cells using genetic approaches, using diphtheria toxin, and you see this tissue remodeling that's happening. There were discussion about how apoptotic cells may be doing it, how other DAMPs or PAMPs are triggering it. I don't know if Tad is here. He has shown prostaglandins are doing it. There is a general theme that inflammation could be an essential trigger for the regeneration process. And in Paul's talk, I've noticed that he's also showing as early as it can get for someone to measure. He has seen these cells to come in to take a role. So maybe Paul, you can comment based on your model, what you think about those?
Well, I think that one of the things that we were wasting our time with was trying to do protein synthesis, because when you kill a cell, you get the release of ATP, you get the release of formulated peptides, you get the release of all kinds of different things. And then if you disrupt the basement membrane, you get different collagen fragments, hyaluronan fragments. All these things contribute and recruit immune cells. We know they activate immune cells. We know they recruit immune cells. And so you're absolutely right. I mean, the very first thing that happens is platelets surround this entire area. And that happens so quickly, we have real troubles putting the preparation up and imaging it. So it's an instantaneous response. You want to repair as quickly as you can. And so I think that's the first step. And to our surprise, the macrophage come in so quickly as well, and we didn't expect that. And they're probably releasing all kinds of growth hormones and things to start the repair process as well. So I think it's a very, very rapid event.
There aren't any other comments on that line. I'll just maybe switch gears a little bit. One of the kind of major questions in the field is how can we study inflammation at the whole tissue or organism level and how this changes over time. So, we've seen today the model that Mark showed with tonsils and using organoids in general, organoid systems can be really useful as well, of course, as in vivo studies. But can you all maybe suggest kind of ways that we can think about how to study these changes over time and in the actual organ in these different contexts?
I think, with regards to what Mark and Paul showed, the imaging is really where it's at, being able to see these cells. Leukocytes are so dynamic in their activity, and still we've not, I'm speaking for myself, I've not mastered the imaging abilities to be able to capture those rapid responses. And then, over time, thinking about this as a spatial temporal event, how the cells are migrating out of the space. We look a lot at those first events because they're very important and we've come to understand them. But these cells are experiencing a robust environment that is dynamic in its nature. And so how are they modifying their responses, whether it's retreat or remodeling, expression of new proteins. So all of that, I think imaging could be the foundation for how we start to investigate that.
Yeah, I agree. I mean, I'd love to have some of Paul's videos. Those striking. But there is there's something to be said for molecules and transcription and translation, and it definitely in the tonsil business, the fact that we can get this organ from very young children versus very much older people, adults. I really think we could see some influence. We know inflammation is just, there's a lot of chronic inflammation. Older people, it doesn't it doesn't wait till you're 80 or 100 years old. It's happening sort of continuously. So I bet we could see something. We are actually now looking across this whole huge age range with single cell transcriptional data and using flu vaccine. We also know that flu vaccines don't work as well in older people. So maybe that's because of inappropriate inflammatory cytokine expression. So I do think there's stuff to be gained here. Given anytime you have something you could do in vitro that so really quite well mimics what's happening in vivo in this context. And we know a lot of, I didn't mention, but in the paper we did remove dendritic cells, and then nothing worked when we removed dendritic cells, but then we could replace the dendritic cells with a cocktail of interferons. And so that kind of precision and mechanistic routine, that not only can you observe things, but you can intervene and test hypotheses. I mean, and also in terms of humans, people are always attacking me saying you can't do mechanistic experiments in humans. Yeah, neater. And which is bullshit, actually. But the organoids make it trivial. In fact, it's much better to have an in vitro system than a mouse. You just have that many more degrees of freedom. I'm preaching to the choir here, but
So we spoke about a little bit about the use of the in vitro systems, and I think, Mark, you are bringing again how it is important to develop those systems for mechanistic experiments. I want to ask maybe Angelica, you can comment on that. How much do we know about the human ECM, and can we generate an ECM atlas for these regenerative purposes? Again, speaking of the atlas efforts, for future of studying regeneration, how much do we know about ECM, and what else do we need?
Yeah, so I think that's absolutely right, that an atlas is needed, and the omics portion is what's going to come out first, right? The identification of proteins, as Paul elucidated, glycoproteins, all of the components that make up the matrix. What we really are, struggling with is identifying the variety of mechanics, for example. So when we've talked about extracellular matrix, we usually don't discriminate between basement membrane and interstitial tissue, not even in its mechanics, and they're significantly different. They're one's a very thin layer. We try to mimic the ultra thin structure, so you have direct cell-cell contact between the two cells that are decorating that basement membrane, as opposed to this thicker kind of three-dimensional house that cells live in, which is the interstitium, or in the interstitial space. And we haven't done very well. Just within one organ, so the lung, we could think about the basement membranes versus the interstitium. And then that's not even to mention the variety in the skin, in the brain. All these tissues are made of extracellular matrix that is, informed by the cell. And then the remodeling and regenerative and repair events. So we have a long way to go, not just in the omics, but then what that means for the mechanical characteristics of these tissues. And then once we get bulk mechanics, then we have to get to the microevent, because the microevent really is what dictates cell, in the case of neutrophils, for example. The neutrophils are crawling either along a fiber or through a pore. They're not crawling on a big, solid plane of matrix protein. So we've got some resolution issues and characteristic issues to get down to. But I think omics, the proteomics of it, will be the first step. I don't know, Paul, you know the space well too, so I'd love your...
I mean, I think I'll give an example based on what Ruslan presented today. A couple of people have taken that work that he published in Cell and have expanded or done specifics. And so I showed that Kupffer cell sitting in the vasculature. And what you can't see very well is that it's touching a stellate cell, and it's touching a hepatocyte, and it's talking to the endothelium. And you can recapitulate that in vitro in an organoid-like system. And you find that the second you take away a cell, that Kupffer cell stops being a Kupffer cell. And so the macrophage starts changing. And when you start laying down basement membrane around that Kupffer cell, it can't reach out anymore. So it retracts and becomes a de-differentiated macrophage and can't perform its functions. And so I think that, understanding and seeing molecules together with imaging, I think combining the two is going to be really, really critical.
Yeah, I think this gets to kind of one of the underlying themes that we've seen both in this discussion and earlier about how much the environment matters and how we really have to take, all of these cells and all of the molecules, metabolites and ECM and so on, how they all contribute to the phenotypes of cells. And again, with spatial transcriptomics and other technologies, I think having an idea of what all the things that are happening in a space and kind of how these cells interact is really important to kind of getting to the next step of understanding, how does inflammation instruct tissue repair and regeneration, which is the ultimate goal of, when it overshoots and goes too far and goes to fibrosis or other disease states, how can we fix that? And to kind of relate in something that Shruti just typed in the chat here, which is something that's come up several times, is this idea that cancer is often called a wound that doesn't heal, but many of these inflammatory diseases co-opt pathologies of repair. So in IBD, psoriasis, COPD. So how can we actually normalize tissues in these contexts? So I don't know if any of the panelists have some ideas about how we could learn from these disease models and try to normalize tissues that way.
Yeah, it's one idea we've had is could we transform some of these cells? I mean, they're common B-cell malignancies, for example. Can we mimic those and then put those into the tonsil organoids and more about how does the immune system deal with the incipient transformation event? But if you have enough mutations so that you're somehow avoiding the immune system and then growing, that would definitely be a great model to look at this question.
Yeah, I agree with Mark. I think if we can, have the opportunity to combine a tonsil organoid or a lymph node organoid with an epithelial organoid that harbors some immune cells and fibroblasts, I think that would allow us to monitor in culture, or if, what Paul suggested, if the imaging technologies advance to a great scale and for longer duration of time, I think that could help answering some of these questions.
We have actually, in a collaboration with Ash Elizadeh, Lisa did culture a lymphoma lymph node. And so that actually might even be the best type of system. We didn't look at it in this context, but that would be kind of ideal because you've got this lymphoid environment, and then you've got a real live set of cancer cells right in that environment, and doing whatever. You could just look and see.
Yeah. So because I know Bobby is working on the IL-22 axis, and I know also a little bit his past metabolism work, I want to stimulate the discussion on this interface of immune cells and stem cells in the context of homeostasis and tissue regeneration. So I think for the future, we clearly appreciating now that with the omics approaches, we will understand the tissue in better. And with the model systems that were presented, we will try to model these things. But what do you guys think about the challenges and potential open questions on stem cell, immune cell crosstalk studies in the context of regeneration? And Bobby, maybe you can highlight some of your opinions.
Yeah, definitely. I mean, I think as people have mentioned, the key thing are systems where you can have, human tissues, human epithelial tissues with all the different types of immune cells that are all interacting. So you can see that complexity. And yeah, one of the themes of the talk I'll give later is that a lot of the signaling molecules that drive the inflammatory processes are also acting, those same molecules are acting on the epithelial cells or even on the stem cells to promote repair. And so what I'm sort of interested in is using sort of protein engineering or synthetic biology to take these molecules and shift their properties. So in the case of diseases, you don't want the inflammatory effects anymore. You want to isolate the protective effects. And so how can we kind of engineer and shift the properties away from what they're naturally doing towards what might be beneficial therapeutically? But yeah, I think the organoid systems and also the imaging are exactly what I think are the kind of the key tools to test those things out and try to do that.
I think this, what Bobby just pointed out, this balance between dampening the pro-inflammatory damage signals as opposed to the promoting the regenerative signals is really important. I mentioned that we're doing neural stem cell vascular work and with inflammatory signals. And in one case, we encapsulate the neural stem cells into a neural extracellular matrix and deliver those into a stroke model. And the best case is when you don't completely deplete the inflammatory response, where you have some signals that are coming from microglia or other inflammatory cells in the damaged brain environment, so that you can actually get positive proliferative and differentiation signals to the stem cells. If you deplete the whole inflammatory response, then you don't have survival, you don't have the regenerative events that you're really looking for in repair, for repair after stroke. So there really is a balance. All inflammation is not bad, right? It's necessary for cleaning up the damaged space. It's necessary. Now we're understanding to driving cellular function. So I completely agree with Bobby that it's really about a balance of, for us, the environment as with regards to extracellular matrix and the cell's response.
All right, great. Thank you so much. I think in the interest of time, we will wrap up. This is a really great discussion, and thank you again to all of the speakers for a really interesting session, and looking forward to the next couple. Thanks a lot.
Great, thanks everyone. Let's take a quick 6-minute break and return at 2:20 p.m. Eastern for session 9. All right, everybody. Welcome back to session 9, which will be led by Dennis Disher from the University of Pennsylvania and Molly Stevens from Imperial College London.
Yeah, hi, you can hear me?
Yep.
Okay, very good. Yeah, so I'm Dennis, and let me just introduce the session, mechanics and engineering. We've heard a little bit about mechanics today, the most recent session, and a little bit yesterday as well. It's certainly relevant to fibrosis. The word stiffness came up a number of times in the earlier session today. With that, I'll have Molly introduce our first speaker.
Hi, everyone. Pleasure to be here online. I'm delighted to introduce our first speaker, who's a good friend of mine, Farsh Gulek, from Washington University, St. Louis. Go for it, Farsh.
Thank you, Molly. Thank you, Kara, Shruti, Mike, for the invitation. Is my slides okay? Everything look good?
Yeah, looks good.
Okay, great. Thank you. I couldn't have asked for a better conference and for a better introduction than the previous session. So what I'm going to talk about is something I'm calling metacells for regenerative medicine, basically modifying cells for synthetic biology to engineer artificial gene circuits. And so you were going to ask, what is a metacell? Well, I'm not sure. I just made up the word. But as an engineer, we study metamaterials a lot, and metamaterials are materials that have properties that aren't normally found in nature, such as this negative Poisson's ratio material. And with a couple of teenagers having passed through my house, we are into superheroes and metahumans. And so the definition seemed to fit that if we can create a cell and engineer it so it has properties that are not found in nature, but have improved properties for therapeutic purposes, we can really potentially use them in a number of applications. And to make these changes in the cells, of course, we're going to use some of the latest methods of genome engineering, like CRISPR methods, virus methods, to put synthetic receptors, gene circuits into cells. And we can do this with various types of stem cells or even primary cells. So none of us have really defined a stem cell, and particularly ones that are being used now in the clinic for regenerative therapies. And as you'll see from this advertisement, stem cells are specialized cells that know what you need, where you need it, and they go there and they become it. And that's, of course, absolutely untrue. And none of this stem cell therapies that are being used in clinics all over the world now are actually working, with the exception of hematopoietic stem cells, which we heard very elegantly about earlier today. The benefits of these types of cells is that they're readily accessible, they're multipotent in vitro, we can grow different tissues out of them, but they have terrible homing and retention capabilities. We've heard yesterday about, you inject these cells in the heart and they're all gone and they don't do anything. We found this, you can inject them anywhere in the body, and within 24 to 72 hours, they're mostly gone. They don't really have controlled therapeutic responses. They don't know how to differentiate in the absence of context or specific cues that we need to give them. There have been several reports of very serious adverse events like blindness, tumor formation, and so on. And there's a lot of variability in these cells. We don't even know what an MSC really is. Most of the data now suggests it's actually a pericyte. We've done single cell analysis of MSCs, and it's not one cell. It's anywhere from 3 to 10 different populations of cells. So one of our targets has been the topic that we've just been talking about of inflammation. And I do believe inflammation is good and it's necessary, but we need resolving inflammation, which is pro-regenerative. That you have early phases of inflammation where there's an early stage with inflammatory mediators, leukocytes come in, various immune cells come in. Then there's a resolution and a post-resolution phase where adaptive immune cells come in and do the cleanup. The problem oftentimes is chronic inflammation. And this can happen due to a number of reasons, but it's non-resolving and it's associated with degeneration, fibrosis, cancer, as we've heard. And the current therapy is continuous protein drug. And this is particularly common for autoimmune diseases such as rheumatoid arthritis or Crohn's, things like that, where you will just completely shut down the immune system with inhibitors of, for example, TNF-alpha and other inflammatory molecules, which are necessary for fighting inflammation. So these types of drug therapies, protein delivery systems, can work. And in certain cases, they are actually very useful, particularly in acute systems. But for chronic diseases, it's hard to get that level of drug being delivered over a long period of time in a controlled manner. And some materials now are being developed that are stimulus responsive. But in general, protein drug delivery is like a leaky bucket, and the drug leaks out over time, and you either have to refill the bucket or put in a new bucket of drug. Gene therapy has been useful in several cases, but oftentimes it's gushing out drug in an uncontrolled manner. We and others have developed inducible promoter systems for gene therapy, and there you can tune and induce drug delivery, but you still have to know when and where to do it. So our thought was, can we create smart cells using this concept of genome editing and manipulation of receptors and gene circuits so that we have an automatic drug delivery system that can sense a stimulus, whatever it is, in this case, inflammation, and deliver drug. And so when we look at the cell, we have a lot of parts to work with. We have receptors, adhesion molecules, ion channels, we have the internal gene circuits to play with, and then we have the outputs. And those outputs can be any range of cytokines, pain pathways, pro-regenerative factors, and so on. So what we've wanted to do is basically deconstruct these pathologic responses that certain cells have, then going in with CRISPR or viral methods, cut out the pathologic response or at least override it, and in its place, insert a biologic drug. So we have a feedback controlled system. And I make this analogous to a very simple computer program like you put in your iPhone, short snippets of code that can do these simple feedback loop type commands. The first system like this that we made, our grad student, Jonathan Brunger, tried to create anti-inflammatory cells targeting TNF-alpha and IL-1. And what he did was deconstruct these pathways and basically create an artificial gene circuit around the TNF or IL-1 receptors, where he basically isolated CCL2 as a great surrogate gene marker for TNF levels outside the body. And using CRISPR, what he did was go in and cut open the CCL2 promoter, and in its place put the drug inhibitor, either soluble TNF receptor or IL-1 receptor antagonist. Now, when this cell sees TNF, instead of expressing CCL2, it creates TNF inhibitor, which goes in a feedback system and blocks the drug. And this is a tunable and inducible system. So we learned again, that you can't inject these cells and have them engraft. So in its place, we developed artificial implants of basically cartilage. These are in iPSCs. And by differentiating them into cartilage, we could plant them subcutaneously, and they would last for months because cartilage doesn't need a blood supply and it gets all of its nutrients from diffusion. So we put these in mice with rheumatoid arthritis, and work by Yoonrak Choi and Kelsey Collins in our lab showed that we could actually treat rheumatoid arthritis using this self-regulating system that only delivers IL-1 inhibitor or TNF inhibitor when there is inflammation present. And so in this model, we saw a complete remission of pain sensitivity, 40% decrease in the arthritis score, and a complete elimination of bone erosions, which no drug therapy clinically can even do right now. And that's by modulating the drug response. So with this concept that we can make little programs to put into cells, we've created a whole bunch of other programs so we can see where the cells are. These are obvious ones because we can label the cells with luciferase or GFP, but if you tie the fluorescent or luciferase signal to the drug output, you can get a signal intensity. We have systems being developed to shut off temporarily these drug delivery systems. Let's say you have an infection and you need to shut it off. We can do it with a TET off system. These are iPSCs, so you need a self-destruct system. And our lab and others have started developing much more complex system. Ron Weiss at MIT has developed some very cool systems using microRNAs as switches in these types of controllable systems. So to bring it to mechanics for our session, one of the most recent developments that we've had is to create a force-sensitive touch sensor. And basically, we know that cells in cartilage are highly sensitive to mechanical load, and depending on the type of load, they respond very differently. We've identified that low-level loading is transduced through the ion channel TRPV4, which is a mechanical and osmotically sensitive calcium channel, and it regulates the response of chondrocytes, anabolic response of chondrocytes to loading, whereas high-level strains are transduced by the piezo ion channels, synergy between piezo 1 and piezo 2. So this would take about an hour to explain in detail, but the simple steps are that we identified those channels as transducers of mechanical signals, spent years deconstructing the transduction pathways using gene array and now RNA sequencing, use a bioinformatic analysis to identify mechanosensitive driver genes and transcription factors as the hub genes where these occur, then built synthetic gene circuits with either CRISPR or lentivirus using those mechanosensitive motifs to create tunable and inducible responses, basically a field we're calling mechanogenetics, sort of like optogenetics, that a mechanical load will now induce a gene of interest response, in this case, a therapeutic drug. And so Bob Nims and Lara Ferdehert in our lab developed several of these motifs. This is just one of them based on 5 NF-kappa B responsive motifs, which are mechanosensitive, and engineered that into a piece of cartilage. Once you mechanically load it, these cells, which would never make anywhere near this amount of IL-1 receptor antagonists on their own, now create drug in response to dynamic mechanical loading. So this isn't the only type of mechanogenetic circuit. Other groups have developed ones that are based on ultrasound, for example, to open piezo 1, piezo 2, to activate CAR.
Just one more minute, Fosh.
Okay, yep. Wrapping up, activate CAR or YAP-TAZ to respond to stiffness in the tumor system, or in our case, exogenous mechanical loading. So future opportunities, creating self-regulating cells for pain, inflammation. We've created a anti-fibrotic cell that's TGF-beta sensitive, and we'll release decorin or TGF-beta receptor 2 as an inhibitor. We can target growth differentiation. Other groups have looked at multicellular systems spatiotemporal control, and even complex Boolean gene circuits. Kevin is going to talk about the role of ECM and biomaterials. Obviously, this is going to control mechanical responses. And these are iPSCs, so we can differentiate them into all different types of cells. So with that, I will conclude and thank my lab who did all of this exciting work, a number of collaborators, and funding for this work. Thanks.
Thanks so much, Fosh. That was so exciting. As you know, I'm a bioengineer. Can you tell us how conserved these pathways are from rodent models to human? Are there any differences we should be aware of?
Yes, that's a great question. So we have done most of this in rodent models and mouse models, but we've done a lot of it in larger animals. So the mechanogenetic circuit is actually done in pig cells, and we've now transferred it over to human adipose-derived stem cells that have been immortalized and working on it. So there's a lot of, it's a great question because there's a lot of specificity and detail of having all of that machinery in place. So for example, those ion channels need to be expressed. They're very well conserved, but as a cell goes through differentiation process, it's not expressing mechanosensitive channels as a stem cell, but it develops them as it differentiates. So those subtle questions are really important in the operation of it. As I mentioned, very dependent on the context of the material that they're in, whether it's ECM or an artificial material like a hydrogel.
And I'm just wondering, because as you'll know, rodent models are not quite as representative for cartilage as some of the other goat and models like that. So does that matter in terms of looking at these mechanosensitive pathways, or is it just going for proving the principle and then moving it up to different preclinical systems?
Yeah, no, that's a great question. In that regard, they've been very, very similar in the rodent and higher mammal models, because basically we're just taking cells and growing a large piece of cartilage, but it's being done out of mouse cells. So it's about 1 millimeter thick, 4 millimeter diameter piece of cartilage like you would get out of a larger animal. But what really controls it is diffusion and mechanical loading through the cartilage, which is very similar among different cartilages.
And then there's a question from the audience, but I was actually thinking the same thing. Once this sort of thing finally does go towards human, for example, do you need to be concerned about differences in weight bearing, essentially, amongst different people in the population?
Yes, so for a mechanogenetic system, that is a great point. So basically, if that's implanted in the joint and it's mechanically loaded, then different amounts of loading are going to activate different systems. So what we want to do is really investigate it and understand that, that tunability of it, because we can change all of the sensitivity, like how much drug is produced for how much loading, and we still need to determine how sensitive the overall response is to that type of stimulus.
Perfect. Thank you. That was extremely exciting. Thank you very much. I'll pass over to Dennis now.
Yeah, thanks a lot. So our next speaker is Samira Musa from Duke University. I'm not seeing her quite here. Samira, are you?
Yes. Hello, everybody.
There you are. Great.
Thank you. Yes. Thank you so much, Dennis, for the introduction. Thank you also to the organizers for the invitation to speak. I'm going to try to share my slide here. Just a moment. So my lab is ultimately a stem cell engineering lab, but our approach is really to derive organ-specific cell types and then use biomaterial systems to develop microphysiological systems in vitro that would allow us to more closely model human physiological responses. The goal is to be able to use some of these engineered in vitro platforms to better understand human disease mechanisms. And I think a lot of the conversations we've had in this program so far has highlighted some of the tissue-specific differences and how mechanisms don't always translate. So our hope is to be able to use these platforms also as potential alternatives to animal models for understanding human-specific responses, especially to drugs and also as a platform for therapeutic development. So much of our strategies really, we draw inspiration from nature. And so we think about how a cell decides to transition to different fate or stages of development, and what signals are required to drive that transition. So many of these cues have also been, I think, are now increasingly being appreciated, that there are multiple factors within the cell's environment that work in concert to drive cell fate decisions, which include cells' interaction with soluble factors, as well as cell interaction with their neighbors, the cell-cell interaction, and the mechanics, as well as the biochemistry of the extracellular matrix. Some other factors that probably are yet to be fully explored also include fluid dynamics and how it influences tissue development and function, and even disease progression. More increasingly, we're learning about how stretch and relaxation motions in different tissues like the lung, and some of our work in the kidneys also show how some of these biophysical forces or cues can drive tissue development and function. And in the kidneys, which I'll talk about a little bit, we also think about how osmotic pressure drives function of the organ, especially in the nephron, which is one of the most easily degenerated tissue in the body, and also molecular gradient. So we think about how we can use chemistry and biochemistry to design microenvironments that would allow us to present cells with many of these signals that we know would be required to drive their fate transition. So by considering a lot of these factors, we've developed methodologies that consider biomechanics and other biophysical forces, and also biomolecular signals to drive pluripotency or promote pluripotency in human iPS cells. And by also tuning some of these signals, we develop methodologies to drive the specific differentiation of organ-specific cell types, including neuronal cells and specialized endothelium, but particularly a fenestrated endothelium, which have remained impossible to drive in vitro. And we're just beginning to do some of that and discovering how some of these tunable signals can be really modulated in vitro, including mechanical strain and interfacing cells with appropriate cell types to drive their cell fate decisions. And in some of our more recent work, we also develop methodologies that really takes advantage of activating specific signaling pathways and mechanotransduction pathways to drive the derivation of kidney podocytes, a specialized epithelial cell type in the kidneys that when these cells are damaged in tissue in the kidneys, a patient is more likely to progress to end-stage kidney failure. So these together really provides us with platforms that allow us to hopefully more closely mimic human tissue function in vitro. And I should highlight that I think YAP came up a few times also, but one of the things we know is that sometimes the function of these pathways, whether it's induced mechanically or biochemically, have organ-specific context or lead to different phenotypes. And we show that activation of YAP is important for stem cell pluripotency, or specifically in human-induced pluripotency. But inhibition of YAP is actually a very potent mechanism for driving neurogenesis. And we took this from, although we discovered the phenomenon mechanically, we showed that you can actually target YAP biochemically or even via genetic means to still drive neurogenesis, really showing that some of these mechanisms that we derive or understand or discover from mechanical manipulations are translatable or at least transferable to other platforms or biomaterial systems. So our hope, what we do when we have these organ-specific cell types is then to use them as building blocks to build or engineer functional models of human tissues and organs. And some of the organs that others also working in this area have developed included lung, worked on kidneys, and also are working on the brain and other the cardiovascular system as well. But you can imagine if you have pluripotent stem cells that can differentiate into potentially any of the cell types that would be required for building these models, we could someday truly have engineered organ-specific models that are truly derived from individual patients, and be able to someday fluidically link these and study human physiological or pharmacokinetics and pharmacodynamics in vitro. So this, of course, could be a platform that you can imagine could be used to study just about any organ's development, function, and disease progressions. One direction we've used this is to help us understand how the kidneys work. And I should highlight that we know that one of the key functions of the kidney is to remove toxins from the body by filtering specific molecules from the blood. And here you're looking at the functional unit of the kidney, where the proximal tubules and also the glomerulus here. But the primary site for blood filtration is the glomerulus. So it turns out that the glomerulus is also the key target of many forms of kidney diseases. And so we tried to engineer this model using several core research areas to help us understand both the development mechanisms of disease and how it targets this particular functional unit, and why this particular region in the kidney is so not able to repair injury, or it does not, there's no evidence that it can regenerate at all. So by understanding how these different cell types are derived in the kidneys, we're now beginning to understand some of the mechanisms involved in both the development and disease progression, and using how extended how specialized these cells are that we derive in vitro to build microfluidic devices that more closely recapitulate the function of the kidney. So we think about how compartmentalized the kidneys are, and it's basically impossible for us to model human kidney function using traditional tissue culture plates. So we needed devices that would allow us to have those dedicated fluidic compartments, and the microfluidic system allow us to do that and create these molecular and cellular barriers that would together function to allow for that selective filtration of molecule. More importantly, we found that using these systems, we can engineer a platform that mimics the selective filtration of molecules in the kidney, much as an in vivo system would. And we also discovered that mechanics play a key role in being able to derive in vitro models that recapitulate different developmental stages. So by applying mechanical strain, for example, we can engineer a system that actually mimics more mature form of a kidney. And by removing that signal, we get an immature type of kidney. And we actually understand a lot of the molecular mechanisms underlying how the kidneys develop. And also interfacing these cells with the epithelial cells with endothelium in the same system, we're able to tune how these different cell types mature and contribute to the overall tissue development and function in vitro. By essentially modulating, in this case, just the mechanics alone, we develop models that recapitulate different stages of development that has proven powerful for us in terms of truly understanding what makes a tissue able to respond to injury or exhibit really high toxicity to a drug, for example. And so we hope that these platforms will be useful for understanding injury response mechanisms and why some of these tissues don't regenerate, and how we might be able to target some of the pathways involved in that process. Some technologies we hope would help us advance this field include really understanding, first of all, why the kidneys don't even repair themselves in the first place, especially the glomeruli. And how different diseases, because we know that there are so many ways to damage your kidneys. It could be drug-induced, even over-the-counter drugs could damage kidneys quite easily. diabetes is a common cause or at least association factor for kidney disease. There's genetics involved. And we know this viral mediated, this is very common in patients with COVID-19, for example, as well as HIV. So we try to engineer these different types of disease models and ask some of the common and distinct pathways in these human disease models and how these can be genetically tuned using some of the technologies Farsh actually highlighted beautifully in his talk to potentially reprogram these cells respond or how they choose to respond to injury. So then instead of maybe dying and degenerating completely, we might get them to start to acquire some functional phenotype. Or perhaps, since we know now a lot of the mechanisms involved in the self-pay decision, we might be able to drive these cells to specialize or recruit endogenous cell types and allow them to localize and potentially switch lineage in order to repair a damaged tissue, for example. So these are all technologies that currently do not exist for the kidneys, and I'm sure a lot of other organs as well, but would be super cool to have in advancing some of these technologies. And my hope, and I think that's true for a lot of others, is to be able to use these platforms to both to really understand specifically human physiological responses as potential alternatives to human or to animal models, but perhaps also useful for understanding some of the species-specific differences that have been kind of like a roadblock to advancing the field in the future. Okay. So with that, I would thank my group, funding sources, and everyone for your attention. Thank you.
Okay, thanks, Sonara. Yeah, just a little bit long, but talk is open for questions. There's not any posted quite yet. Can I ask, this came up earlier when we were chatting about the kidney being exposed to high osmotic pressure, which you listed. My limited study of this in the literature was that osmotic pressure differences of 100, 200, 300 milligaussmolar, I think, are achieved in the concentrating in the kidney through concentrating solutes. It's an extremely high pressure, and it's enough to extract solvent out of the cell, water out of the cells, and even lead to DNA damage and things like this. Do you have data along those lines? Is that this equates to a hydrostatic pressure? But what are your thoughts on that sort of on the osmotic effects and/or hydrostatic pressure effects in this sorts of system, particularly the kidney? Yeah.
Yeah, so we do know that at least when you look at the nephron, so as I highlighted, you have the glomerulus, which is the entry point for our blood. So, and really, like we know that about 20, when blood is filtered in the kidney with every cardiac cycle, and about 25% of your cardiac output goes straight to the kidney. So like you pointed out, that alone creates a lot of pressure in the glomerulus as well. But as fluid is being filtered, you get variation as it goes through the nephron and the tubules and things like that. So that's where you really do get osmotic pressure also influencing molecular reabsorption in the tubules. But in the kidney is so, the pressure is so high that you do get flow in one direction. So I do think that when we have models that can be fluidically linked, like I said, with a different organ on chip systems, that could be very useful for understanding how some of these forces or cues modulate the function of these different parts of the kidneys. Yeah.
Perfect. Yeah. All right. Yeah. So there's a couple of questions along exactly where you were finishing. You were hooking up different organ systems. And so please take a look at those. I think we're running out of time, but terrific talk. Lots of compliments there. And some
Thank you.
Thanks a lot. Molly, back to you, Molly.
Hey, thanks very much. So we've now got our talk from Kevin Healy, who's a professor at UC Berkeley. Go for it, Kevin. Are you muted, Kevin?
How about now? Thank you, Molly.
Yeah, that's good. That's
great.
good.
Okay, yeah. I have some disclosures. There they are. Okay, so we heard a lot of great talks on the cell therapy aspect of what we're considering regenerative medicine. There's also gene editing, which Farsh discussed a little bit. I think what I'll discuss more is what we call biomaterials therapy. And if we look at the challenges we've faced for over 30 years in basically cell-based regenerative medicine, I mean, there's a lot of advances, you know, sort of depicted on the slide. But we still face, and it was mentioned a number of times throughout the conference, things like cell engraftment, certainly vascularization. If you don't get vascularization, whatever you transplant is going to die short term. And then the consequences of that can be positive or negative. And then ultimately for function, the system has to be innervated. So when we decided to divvy up the talks, I said I would take up the aspect of the matrix. It was brought up a number of times this morning already, this afternoon, maybe for you folks. And so, you know, all cells, except for maybe exclusion of hematopoietic cells, sit in a matrix, especially stem cells have been already articulated well throughout the conference that you need a matrix to survive, right? So, you know, you're all familiar with this concept. So if you decellularize this matrix, if you basically take the cells out, what do you have left? Well, you have, you know, proteins that are expressing cell binding domains. You have what is supposed to be the theme of this session. You have mechanical properties such as the elastic or shear modulus of the material. I'll get into that in a little bit more detail. You have growth factor presentation. You know, in the native matrix, growth factors are not soluble floating around. They're presented off a matrix itself, usually proteoglycan or heparin-based systems. And then degradation kinetics. And so, you know, the central tenets in biomaterials therapy now has to incorporate all of these systems if one is going to make a system that is going to be amenable to handling things like cell transplantation and improved function. So if we look at, you know, what we heard earlier in the conference about just taking that decellularized matrix, you can take that and use that as a therapeutic, and it has all those components in it. But from the standpoint of and maybe this has been going on for about 20 years, but from the standpoint of the synthetic aspect, about 30 years of effort have been pushed in the areas of hydrogels. And the two areas are actually coming to the consensus based on those tenets I just defined, that we're going to have material or matrix assisted transplantation, and if we're going to improve on our regenerative capacity. So, you know, we and others in the community have centered on this material, hyaluronic acid. And the rationale behind that is it's easily functionalized chemically. So we can introduce these we can alter the mechanical properties, and we often say these are tunable or modular. You'll see what I mean by that in a moment. We can introduce biological ligands, proteases for degradation, and control their degradation rate by altering the Michaelis-Menten kinetics of the enzymatic degradation, usually by MMPs, and they can capture and present growth factors either exogenously added or endogenously synthesized by the cells. So the cartoon for this looks something like this, where the gray is the hyaluronic acid. You introduce these peptide-based cross-linkers, so they are just mimics of what you would find in collagen, laminin, protein of your choice. And you choose them nominally to be responsive to MMPs that we know are in the wound healing environment. So for example, MMP 2, 9, and 13 are the ones that we and others have looked at. The mechanical properties are clearly important. And as Farsh pointed out in his presentation and yesterday in the closed session, that it's not just mechanical properties, but it's under shear, compression, tension, viscoelasticity, so time-dependent behavior, and was mentioned earlier this morning, localization. You know, bulk, what we do is make bulk measurements. Many people make bulk measurements, but microreology is really important in terms of what's the localized stiffness of a material in the presence of the cell. There's peptides of cell adhesion, and you can just dial up, you know, a number of different variants in this space, as well as growth factors for basically differentiation. And they're sequestered in the matrix by adding this yellow component is just heparin. So we and others have exploited materials like this, and to bring the mechanics into play. Here's a CD31 stain of cardioprogenitor cells. These are cardio-derived cells, the cardioderived cells that can have tri-potential, and we're pushing them into the endothelial phenotype by introducing a large RDD sequence, which integrates with alpha V beta 3, and then some TGF beta in the system. And what you can do with these modular systems is create these response surface maps so that you can modulate essentially in a de-optimal design experimental strategy. You can create a modulus axis, a peptide concentration axis. You can see the results pretty poor, right? So if this is what you chose to start your experimental work, you might deselect this kind of cell if you were testing it in this basis to promote, in this is similar to a UVEC network formation, in this case, an assay for network, in this case, tube length and then tube thickness based on the system. So clearly you can run experiments that demonstrate this activity. The nice part about the hyaluronic acid model is that we can use enzymes that are unique to hyaluronic acid to degrade the system and then collect the proteins and put those on essentially a focused array. So in this case, we're looking at a focused array. Just pay attention to the gray bar, which is the full hydrogel that I just described. You can see expression of molecules like VEGF, MMP3, HGF, all supportive of basically EC proliferation, migration, and then also vascular stabilization. And this is also can be organized in standard cluster format, just to show the array of androgenic activity. In this particular case, if we block with an antibody to CD105, this basically stops this interaction and totally alters the response. And then hyaluronic acid by itself doesn't show the same response, right? So it's this complete system that's important. So I'm not going to go into examples in detail, but I'll just give you sort of the high water mark. So what we know is that systems like this, when we transplant cells, we can have those cells survive. Not only do they survive, they actually promote angiogenesis and create these beautiful blood vessels that basically anastomose with the host. And I'll show you an example of that in a moment. MMP degradation actually controls this rate of, we would say, or a potential for engraftment. And then I won't say anything about this, but we can also control metabolic function with transplanting totally different cell types. So the system is robust and universal in our hands with respect to its modularity. In this experiment, we just transplanted hydrogel just sub-Q above the tibialis anterior muscle, and then injected a perfusate with isolectin to demonstrate that the blood vessels that were formed in the transplanted cells anastomosed to the host tissue. Again, this is just building on the in vitro studies that were largely recapitulated in vivo. If we look at those protein expression arrays, again, now in the context of different cross-linkers that have different mechanism and kinetics, I'll just point out that they're just significant. And you can just look at this, sort of just grasp the difference of how robust just changing that cross-linker is. And the results bore out again in terms of the androgenic potential of transplanted cells with the correct cross-linker in the system. I have one or two slides to finish up and just to say that you don't always need cells for repair. In the context of this, maybe it's better in the Q&A, but if you create an injury model, in this case, this collaboration with George Chris for volumetric muscle loss, again, in the TA muscle, you basically get really nice, robust regeneration when you just use the hydrogel I described, in this case without any TGF beta, where we could discuss this in the Q&A, but what we believe what's happening is we're just preventing the fibrosis from happening and soft tissue prolapse by just having material present and then letting the cells invade into the matrix. And the cells that invade, that's the big question. But early wound healing cells are clearly neutrophils, macrophages are the ones that are pioneering into the matrix. And then in that case, it seems to be enough to prevent the fibrosis. We know we have innervation in this system because when the physical
just
muscle,
one more minute.
Yep, thank you. The physical muscle is tested, the peritoneal nerve is stimulated. And so I think the main thing to point out is this is the maximum force that we can recover because it not only is a TA muscle cut, but other associate tendons are cut as well. And so you can see with essentially the hydrogel treatment, you almost get that 80% recovery. And again, pointing out that clearly this is based on a model where we get innervation. I'm not showing the data. We also get the angiogenesis just with the hydrogel alone. So, the challenges are still there, cell engraftment, vascularization, innervation. The details of what I talked about are really important. Time and scale dependent mechanical properties, ligands of interest, protease degradation, growth factor, presentation, how it's presented. And then, all of this is context dependent. Which tissue, what cells, that all of you have been working on are placed in the environment and what the surrounding environment is challenging those cells with. And as was just, elegantly presented just before me, we've spent a lot of time thinking about faster cycle time and have a lot of, matter of fact, the major part of my lab is in these microphysiological systems. So you can use these systems to actually improve the cycle time and understand what's going on. I'll stop here and just thank all the folks that did this work, mainly Mitja and Kevin Tharp as postdocs in the lab, and Shane Brown and our collaborator.
Thanks so much, Kevin. Can you tell us a little bit more about the choice of hyaluronic acid versus other hydrogel systems like PEG or alginates or other systems that people also use as blank canvases?
Yeah, about 25 years ago, our first NIH grant in this space was with a thermoreversible polymer called NIPAM, and it's a propylacrylamide, and we did a lot of work in that space. And we slowly migrated to hyaluronic acid because we just felt that its results in vivo were just better. That's the only way I could describe it. Even though they're blank slates, I could tell you when we use PEG-based systems with our equivalent types of peptides for adhesion and cross-linkers, we don't see the same robust response. I can't comment on alginate. I would just put that in the same category. Alginate doesn't have a natural degradation mechanism where hyaluronic acid does. And so we tend to refer to these materials as semi-synthetic because we're building off a natural biopolymer.
Yeah, I was wondering actually whether it was the kind of hyaluronidases that change the degradation in a way that you don't with those other systems, and whether that gets you some benefits.
Yeah, it could be, or it could be the breakdown products are just natural, right? They're fragments of hyaluronic acid, which depending on how you read the literature, can at some molecular weight promote angiogenesis itself.
And then you showed us this really nice figure with looking at how some of the different parameters can be modified to get different responses. There's potentially so many different things you can modify. Do you guys ever employ sort of design of experiment type approaches to kind of optimize the data analytics on those for prediction of where to do your experiments?
Yeah, all the time. I think we might have been one of the first in the biomaterials community to bring the surface response methods into looking at a three-dimensional space, right? And so we use de-optimization designs, Taguchi methods. We've done that also for biointerface design. We're doing it now. We're also using a differential evolution strategy for looking at optimized media conditions. So we're really, I would say, a strong proponent of trying to reduce cycle time, in our experiments.
Yeah, I totally agree.
Plus, these grad students love it. They're like, I have to do 9 experiments instead of 81. Thank you very much.
Yeah, it makes a lot of sense. Perfect. So I think in the interest of time, we're going to move to the panel discussion section of this session. Thank you very much, Kevin.
Thank you.
Right. Okay. So I should first introduce Yuan, who spoke yesterday. Correct? I forget the session, but he's done fascinating work and hopefully contribute to the panel discussion here, which is supposed to be forward thinking, high level forward looking. I should say forward thinking as well. And so of course. We finished off with biomaterials, including attention to the origins, which is the biochemistry, the mechanics, part of the session here, and we started the session. I just should remind with getting inside the cell through Farsh's metacell. Is this what you call it? Farsh,
it's the current word.
Yeah.
Designer cell kind of reminds me of Gucci bags, and
right.
Smart cell, which we've called it, is insulting to people's other cells. So,
but so from the outside, or it started inside, went outside, and in both cases, we were hearing about synthetic, yes, but also really a cyborg kind of matrix, right? Hyaluronic acid, naturally derived, modified, optimized, right? And so there's a lot of engineering with attention to mechanics, whether it's the pathway or the properties, as I say, the viscoelasticity that Kevin told us about. So there's a lot of opportunity and optimization, certainly, and new directions to take this. But what do you after hearing the talks and now at the end of 2 days of workshops here, just throw it out there as our task. Where are we going to be in this field 10, 15, 20 years from now? Maybe that's some place to just get if we can, a brief comment on. Maybe start with Farsh and move on back through. So Farsh, you've now heard these talks. I know listened in. So what are your thoughts?
Yeah, let me just say I love the other 2 talks, and I thought everything brought forth these recurring themes of the whole meeting of fibrosis, inflammation, how it affects regeneration, and stem cell fate. And I think we have a lot of tools now to start to translate things and develop methods like these biomaterial methods that Kevin showed, and microphysiologic systems like Samira's, and synthetic gene circuits. We still need more basic science, in my opinion, because we need the targets. Like, we need to know how does what mode of inflammation do we want to maintain? We don't want to get rid of it completely. We don't want to wipe it out. We want to give it in a highly controlled temporal system. And this is in these very complex contexts, like some of the questions that came up is, okay, in one cell, we may want a different pattern than another cell. At the same time that you're having mechanical loading, you also have inflammation, you also have aging, and so on. So we really need to understand those contexts. And then I think we can improve the way that we apply these tools that you saw in this session. Yeah. Not that we need to refine the tools. I mean, that's certainly left to do, but it's going back and forth, that we can't do one without the other. So I just want to put a plug in for the basic science as well as this type of applied translational and engineering work. That
if you don't mind, before we transition to the other, to ask for an opinion on these, what's next. But so how much, at least in high technology engineering, when something fails, you analyze it very, very deeply analyze your failures, right? So when you engineer your systems and go in vivo, and whether it's a failure or success, whatever, how deeply do you understand? You brought up the inflammatory response to say your metacell, right? How deeply do you make all the measurements you possibly can on that system versus a control versus other perturbations, and with all the tools, as you say, available, constantly hear about, from single cell RNA-seq, et cetera, proteomics. generate huge data sets to understand deeply what your perturbation does to a particular system, and then whether it's AI or something, progress from there. Deeply understand your system, right?
I know that's a great question, and as you know, it's a balance between celebrating our failures and actually getting the project done and the paper written.
No, right.
So,
no, you're right.
In some cases, I think when we have really unexpected failures, then we have to go back and figure out what really went wrong. And the tools that we have now, like single cell sequencing and massive informatics systems, really helps do that. In other cases, things fail because there's a dosing problem, for example, and we can go back and very simply do trial and error, which would be a lot faster than some multi-year machine learning algorithm to figure out what went wrong. So I think in all of these systems, we tend to apply engineering design principles, which is a balance of predictive modeling, predictive methods, combinatorial systems with good old trial and error, because we think this is going to work and we can tweak it. In a few months, we can get a whole bunch of answers that we wouldn't get in the most exact method. And in the ideal world, I would love to do what you're saying, but it's just a balance.
Okay. All right, Samira.
Yeah, sure. I think, at least from my perspective, is that sometimes I think we might get carried away by how many types of signals are in an organ, right? And when we design some of these microphysiological systems, we think about how many of those signals do we want to recapitulate in these engineered systems. But maybe a developing kidney probably doesn't really care about the flow rate, right? But we have to actually find out, or when we reach a certain maturation stage, we need to actually apply a certain biophysical cue. And so one of the things that I think would be really useful is kind of, as we develop some of these engineered models, really understanding that during development, we have a lot of changes and differences in how tissues pattern themselves and then organize into functional units. And so application of some of these signals might not have to be there from the very beginning, say from a stem cell stage or a pluripotent cell stage, but rather might just have to be temporally applied or applied in a very specific manner. Other things that I think, other considerations that I mentioned is also recognizing that these organs aren't isolated, right? Naturally, we don't really have our organs sitting around by themselves. So thinking about real inter-organ communication and how to bring some of these engineered systems together to better understand how maybe a drug is processed by the liver and how other organs receive the metabolites and other derivatives of that is very important. So for me, that's one of the reasons why I'm excited about these interconnected systems and the potential of being able to engineer these organ models from specific patients so that I would know that maybe Dennis's liver reacts this way to this drug versus Farsh's liver. And so I think those are powerful technologies, but we do definitely need the ability to derive all of these organ-specific cell types to be able to repopulate those. So I totally agree that we still need that fundamental basic biology advances to continue so that we can build some of these more advanced models.
Okay. I just want to note that my liver, you need to understand alcohol, how it processes. But you
we'll have you on a chip someday.
Exactly. Yifan, you please share your thoughts.
Sure. I'm thinking like well, from my perspective, I'm a tissue engineer, and like in about 10 or 20 years, for me, like I would like to basically build a really real human model that can really recapitulate what happened in human, like in vitro. Because currently I'm thinking the animal model really cannot recapitulate what happened in the human physiology. So, and also like by doing that, like currently, like what we are doing is using the whole lung tissue engineering. And so like specific to for my project, basically I want to recapitulate what happened in the pulmonary vasculature, and to really mimic the pulmonary vasculature to simulate what happened in the human tissue. I want to more like understand what's happening, like using the single cell or other multi-omics analysis to first understand the biology, like as previous speaker said, and so like understand what's the niche cues over there and regulate the, for example, vasculature, what's the endothelial cell, the parasite and other cells are crosstalk to each other, and then whether or not we can rationally design or culture conditions to really pushing the in vitro modeling system to more simulate the human microenvironment. And also, yeah.
Yeah, can I ask you, Yifan? So that's you know, the forward-looking view. Do you contemplate much more interactions with cell biologists in doing that? That would be, you know, not just using drugs to inhibit or, you know, agonize pathways, but knocking down, overexpressing, and getting at detailed molecular mechanism, that level of understanding, or more traditional tissue engineering understanding of how cells are interacting with each other without looking so deeply under the hood.
I think definitely going to be like at both levels. And first, like I'm thinking to understand the overview of the cell crosstalk to each other, and then build the whole tissue. And like by doing that, also like I understand the basic biology on that. For example, using drug inhibitors and everything, understand the basic biology. And also by doing that, not only we can understand the biology of that, we can also understand some of the pathological and pathology of that. For example, currently we don't really have a modeling system, in vitro modeling system, study the COVID-19, and we don't really have the alveolar model. And because really like the animal model cannot recapture what happened in the human. So like what Samira said, like what she wants to do is building a tissue chip that can recapture what happened in the human tissue, which is very useful, I think.
Okay, Kevin?
Yeah, thanks. Well, I would see a large effort that would focus on human-based models, not zebrafish, but examples where we know regeneration occurs. The mechanisms for doing that could be these complex in vitro models, which includes microphysiological systems. But I think what's more important is building large teams, right? All of the types of folks that are on this call and were on the call yesterday need to come together in some, you know, convergent science focused way in which we have, you know, cell biologists, we have, you know, matrix designers, we have imaging experts, we have, you know, synthetic biology, data science experts to really push the field. And we have to reduce the cycle time. We will reduce the cycle time, I guess, if it's visionary, right? Because the cycle time's too long. You know, being from New York and getting older, I'm really impatient now. And so, you know, to wait 8 to 6 months for response back on histological sections and staining, this is, I think this is one of the problems, right? Why we don't advance so fast.
If I can interrupt, I mean, this is an aspect of your talk, and Farsh certainly resonated with it, which perhaps I missed in previous sessions of the workshop, was this idea of designing, you know, these huge, you know, many variables in these experiments, right? And at least in terms of in vitro systems, trying to optimize experimental design to cover a very large space. You know, you illustrated the peptide and stiffness and, you know, some output, and it's bigger than that, right? And so that's an additional aspect of doing experimentation, right? Which, you know, Genentech Farms does, right? So as you say, you know, it addresses the impatience of everyone, right? You know,
right, because we couldn't have arrived at the hydrogel, you know, our flavor of hydrogel, right? We couldn't have arrived at that without those experimental strategies.
Yeah,
right?
yeah.
And that goes back, Dennis, you know, when you were doing the early modulus work. We were doing, in vivo with this case NIPAM. It turned out not to be the best material, but the conceptual framework was there. I think there's going to have to be advances in technology, right? So I think there was a question that came up to us by panelists, so I'll answer it, which is, how are we going to get to the microreology, right? So the bulk measurements are easy. We can use rheology to get the mechanical properties, and those are physically measured. But the mechano aspects, the mechanobiology that might be happening on a specific cell, and then the viscoelasticity. This is questions I used to get early on, which is like, Kevin, what's you keep mentioning viscoelasticity. So what's the cycle time like that a cell pulls on something and has to have a viscoelastic response, right? I think we arrived at 1 second, after Hertz, which is after years of discussion with colleagues, but there's no real answer to that. We don't really truly know, and it's probably cell type and context dependent again. So advanced tools, and that could come from both the combination of engineers and imaging strategists. So certainly we take advantage a lot of really the best imaging facilities we have here in Berkeley, including, some of the folks we stole from Genelia to work here and really look at, subcellular structures. And I think this all has to come together. I think there are scales in which we can move forward as individuals, like, transplantation of cells, maybe with or without matrix, and looking at the biology and the result. But the visionary strike has to be a much larger team effort, right? I see this more like large program project type initiatives. If Genelia was to give money in such a way, that's how I would look at it, not as like classic R1s.
Yeah. We're running out of time. Molly, I please, back to you.
Yeah, I'm really interested. Our lab does a lot of fundamental science. Also, have developed new imaging techniques, but equally, we've translated some materials into clinical approval across Europe. And I'm curious, probably particularly Farsh and Kevin, how you keep that balance in your head. And maybe starting with Farsh, because you stress the importance of fundamental science, which I completely agree with. But you also have some interests and links to industry. So how do you see these kind of mechanoresponsive gene approaches translating, given that they're a bit more complicated than some of the other things that have previously translated?
Yeah, that's a great question, Molly. And of course, for every different approach, there's going to be a different time scale, different applications, and so on. Some will go much faster, and certainly, things that are acellular, forget, iPSCs that are CRISPR edited inside a material is going to be a lot harder to get through regulatory versus something that's a straight material or a, autologous cell that's minimally manipulated. So, everybody has a different approach. Ours has been that when we start a project, we try to think ahead of, can it translate? And that may be, a few years, it may be 20 years, but are there total hindrances from it ever translating? Then that's probably not for our lab. Along the way, oftentimes we have to answer some of the basic science questions ourselves, but it's still very applied basic science compared to a true, what you would call a true basic science laboratory that's doing more discovery research. Sometimes we just go back and forth and, answer those interesting curiosity questions along the way, but always trying to keep it in mind that we want to do this for the field of regenerative medicine, that we actually want to get it to medicine at some point. So I don't know, that was a wishy-washy answer, but basically I don't have a true roadmap to it. It's more that, okay, this is exciting. Could this possibly translate? Will it solve a problem? Let's try it out.
And Kevin, are you looking at sort of acellular biomaterials more in this context, or combination products?
Thanks, Mike. We're funded by CDoctor, which Mike Longaker is a big part of as well. And that's a regenerative, craniofacial regenerative medicine program. And unlike most NIH programs, they do allow you to have a commercial, it's sort of like an academic SBIR, if you will. So we are pushing this, and what we've learned a lot. So taking the debate, sort of the material science, outfit off for a moment, since how are we going to translate this, commercially? So the chemistry, manufacture, and controls, the so-called CMCs. There's advice like, how do you make this thing simpler, right? How do you make it so, I didn't talk about how this gel is in situ, and there's a lot of things that are cool about a gel, but the idea is like, oh, you need a form factor that might be like a collagen sponge, like preformed. So regulatory says, okay, I'm just approving this, right? And our regulatory path is one where we'll pursue similar to decellularized matrices or other synthetic matrices that are regulated as a device. So device first, because that's the way to get this in the clinic. And then if you bring in cells, then you have now a predicate device. You have the device, as you know, Molly, with your own work, you have the device approved, and now you can add in other things, other components. Again, you could add in, if you knew what molecule to add in, you could add an exogenous growth factor, for example.
Thank you. Dennis, I think we might be ready to wrap up on time.
Oh yeah, I think that'll do. Thanks to all, and I think we are the final session, and after that, look forward to following up with some or all of you. So thanks again.
Yeah, thank you so much, everyone.
Thank you.
Great session. Let's take about a 7-minute break and return at 3:35 p.m. Eastern. All right, everyone, let's get started with the last session of the workshop, which will be chaired by Kara McKinley.
Thanks, Janine, and welcome back, everyone. It is my absolute greatest pleasure to be able to chair this session where we get to highlight some of the emerging leaders of regeneration. And we have really a fantastic grand finale on tap for you. So let's just go ahead and get started with Ahmad Nabhan from Genentech.
All right, let me just try to present my slides. If you could just confirm that you can see them.
Looks great.
All right, so thank you everyone for sticking around towards the end. My name is Ahmad Nabhan. I'm a postdoc in the labs of Vishva Dixit and Joe Aaron at Genentech. And I'm excited to tell you about our ongoing efforts to understand and sculpt tissue regeneration. So I'm going to be talking about this in the context of the alveolar epithelium, which is a distalmost compartment of the lung where gas exchange takes place. And this compartment is made up of 2 cells. So we have alveolar type 1 cells, which are these beautiful, incredibly thin cells, and it's their unique morphology that enables the exchange of gases across their cytoplasm. And then in stark contrast, we have the alveolar type 2 cells, which are relatively less good looking secretory cells that turn out to have some stem cell tricks up their sleeves. And so this tissue, in the absence of injury, is remarkably quiescent. And so why are we interested in its stem cell biology, you might wonder. And that's because the dysfunction of this compartment stem cell is a major source of disease. And for that reason, those cells are, at least I believe, are amongst the most dangerous in your body. And so, for example, the failure of alveolar stem cells due to genetic or environmental causes leaves behind these gaps in the epithelium that are filled in with scar tissue by fibroblasts. And ultimately, the scar tissue is unable to gas exchange, leading to the failure of the lung as an organ and death. On the flip side, in cancer, we've learned that these alveolar epithelial stem cells are the cells of origin for non-small cell lung adenocarcinoma, which is the #1 cancer killer. And so based on this, I became very interested in the identity of these cells. So what's the molecular identity of alveolar stem cells? What pathways regulate their identity and activity? And how can we manipulate them when they go awry? And so we've learned an enormous amount about this over the past 5 to 10 years. So I'm gonna summarize this schematically and tell you that it turns out that fully differentiated physiologically functional alveolar type 2 cells, so the boring secretory cells, are the stem cells. And so they're able to self-renew and give rise to the very different looking alveolar type 1 cells. In order to function as stem cells, these AT2 cells need a Wnt signal, which during homeostasis or aging is provided by as a paracrine signal from a fibroblast. And some AT2 cells have access to the signal and are able to self-renew and give rise to AT1 cells again and again, while their neighbors remain quiescent. During injury, the expanded need for stem cells leads to the expansion of Wnt signaling via a distinct transient autocrine pulsive Wnt, which increases or expands the pool of AT2 cells acting as stem cells transiently until the sort of regenerative process resolves. And so we had a naive thought about this because our understanding of fibrosis nowadays is that it begins with a lack of alveolar epithelial activity, which is then filled in by fibroblasts. So we thought perhaps we could push regeneration into overdrive and fill in those gaps, thus preempting fibrosis. But this idea turns out not to be so simple because epithelial progenitors are not the only ones dependent on Wnt signaling. And so in fibrotic foci, it's long been known that Wnt signaling is very active, as indicated by this staining of a foci for nuclear beta-catenin. And inhibition of Wnt in a model of lung injury seemed to reduce the amount of fibrosis observed. And so if you inhibit it, there was less hydroxyproline. And so thus we're left with this strange situation where to treat this disease, ideally we would like to activate and inhibit the same signaling pathway, Wnt, in two cells that are literally right on top of each other. And so after the completion of this first paper, I was kind of asking myself, how is Wnt selectively activated in AT2 cells during regeneration? And how can we mimic these mechanisms to achieve cell-specific Wnt signaling to treat diseases in patients? And I would say this scenario I've described to you in the alveolus is actually observed in many tissues where Wnt-responsive epithelial stem cells fail and are sort of filled in by these Wnt-dependent fibroblasts. So I think this is a general phenomena that is relevant to many diseases. So I think this is a good time to introduce Wnt signaling. And so the signaling pathway is named after the ligand, Wnt. So there's not one, but 19 Wnt ligands. And Wnt works by oligomerizing the frizzled receptor and the LERP co-receptor. And so there's 10 frizzles and 2 LERPs. So this is mind-bogglingly complex. But all of this complexity sort of centers down on this transducer beta-catenin. So the binding of Wnt inhibits the degradation of beta-catenin, and then it can translocate into the nucleus and drive expression of its target genes. And so this pathway is important in many settings across the animal kingdom. So whether you're talking about flatworms or adult mammalian organs like the gut, and of course it's hijacked in cancer. So we get back to this question, how is differential Wnt signaling achieved in the alveolus? And so during the second half of my PhD, I worked on the generation of these single cell atlases, and I specifically worked on generating the single cell atlas of the human lung. Classically, because there are so many Wnt genes and because they're lowly expressed, mapping their expression in a comprehensive manner has been a major challenge. And so when I got this data back and I got the full transcriptome for all 60 lung cell types, I went back this question. What I noticed is that even though this Wnt signaling pathway is active in many places, the receptors, frizzles, are expressed in a distinct and differential manner. So epithelial cells, for example, in the stisne, express frizzled 5, but not other frizzles. And later I confirmed this with single molecule in situ hybridization. And so this is not just true for the epithelial cells, but fibroblasts specifically express frizzled 1, and endothelial cells specifically expressed frizzled 4. And so this is interesting because frizzles and Wnt's have distinct affinities for one another. So in this classical experiment from the Nathan's lab, they transfected 293 T cells with each Wnt, each ligand, and a reporter plasmid. And what they observed is that depending on the combination of receptor and ligand, they got varying amounts of Wnt signaling activity. And so this suggested that these different the different receptors and ligands have distinct biochemical affinities for one another. And combined with the differential expression that I just told you about, one can imagine a Wnt code where specific cells can talk to each other in separate Wnt channels using these distinct receptors. Furthermore, I think this is a sort of foothold for us to overcome Wnt off-target effects by selectively hitting one cell type but not another. So for example, in fibrosis, we would activate frizzled 5 and we would inhibit frizzled 1. And so we wanted to sort of begin on this question and ask, is cell-specific Wnt signaling achieved through the differential expression of frizzles? And the first question we wanted to ask is the signal being transduced by distinct receptors? And so it's difficult to ask this question because Wnt ligands are notoriously difficult to purify and their specificity is not completely understood. But we can circumvent this problem using Wnt mimetics, which are antibody-based molecules that oligomerize frizzled and LERP through a distinct synthetic mechanism. And we were particularly attracted to the work of Def Sidhu, who had created reagents that can bind and activate any of the 10 frizzles with high selectivity. And so this is what we did. We decided to test this using organoids, which offer a simple and physiologically relevant model. And so we purified our alveolar stem cells, put them in a culture condition for organoid growth, and then inhibited endogenous Wnt secretion using the small molecule, and then attempted to rescue the loss of Wnt using agonists for the specific frizzled. And indeed, we observed that if you stimulate with frizzled 1 or 2, you don't get much growth. But if you stimulate with frizzled 5, you do see the organoids grow out, indicating that indeed this is the relevant receptor. We then did the necessary loss of function experiments where we repeated our experiment, but this time, instead of inhibiting Wnt, we specifically deleted frizzled 5 or other frizzleds in these alveolar cells. And we observed that only deletion of frizzled 5 caused a failure of organoid growth, while deleting other frizzleds did not. And so now we're attempting to see if we can decouple the effects of Wnt in vivo using these frizzle-specific agonists. And this work is it's ongoing, it's not complete, so I don't have the answer for you, but I'll tell you about another result that I think is related but distinct. And so the question is, I think this has came up a couple of times, which is, can we stimulate cell growth pathways in the context of injury to sort of greater levels of injury to drive a supraphysiological regenerative outcome? And so I did a simple experiment where I modeled lung injury using bleomycin, which is instilled in the trachea of mice. And then I measured proliferation integratively by putting the cells on, or putting the mice on EDU drinking water. And then we either injected with a control or with our frizzled 5 agonist. And what we observed is that the sort of in this, imperfect model of lung injury and fibrosis, stimulating frizzled 5 specifically increase the output of the alveolar stem cells by about 6-fold. And so I think I'm almost out of time, so I'll just end here and give you sort of my model, which is classically, if you read most reviews, you'll sort of, Wnt is presented as a hopeless cacophony of, a bunch of receptors, a bunch of ligands that all are talking to each other and then converging on a single transducer. But what I think is going on is that there's actually a Wnt code that is where each cell listens to a distinct Wnt signal, and then you sort of get the same transcriptional output once it goes through. So with that, I'll just go on to, I'll skip this and just thank the people, Vishva and Joe for their support and confidence, our collaborators in Toronto and others at Genentech who've helped me do these experiments. So thank you.
Fantastic. Thank you so much. That is a really exciting body of work. It's an incredible amount of advance that you've been able to have in this field. Everyone, please go ahead and type your questions into the Q&A, but I will get started. I have a lot of questions, but I'll try to share the floor. Okay, so I think my first question is that what is the physiological point of having these Wnt responses in the fibroblasts? Is there any reason under which, kind of coming back to this question of, is there any point at which we might consider a fibrotic response of value? Is there something you might want to toggle there?
Yeah, certainly the fibroblast response, at least in the context of, I'll just talk about lung injury because that's what I know the best. People have tried to inhibit it. They're sort of saying, okay, well, let's just stop the fibroblasts from growing in a acute lung injury model. And what you end up with is massive edema, and the mice will very quickly drop dead. And so you definitely want the fibroblasts there, and you want them to close the gaps initially until you restore the epithelium. Another thing that's really sort of has not been studied is the turnover of fibroblasts. Generally, nobody studies fibroblasts or their turnover, but we don't know if, there probably have to be fibroblast progenitors that then maintain the population, and that is also dependent on Wnt signaling. And so you certainly need them, and you need the sort of Wnt response to maintain the structure of the tissue. I think it's just is understudied.
Got it. And sort of further going down this path of like two sides of a coin problem, obviously, as you mentioned, the downside of Wnt is its cancer promoting activities. When you think about applying this in a physiological setting, do you envision combining it with maybe something like Farsh talked about, where you would have an off switch for this? And have you seen what the set point for that off switch should be?
Yeah, so I have a sort of, I think maybe a controversial take on this, which is that I don't think giving Wnt or any growth factor to an organ is going to be in and of itself oncogenic, because usually cancer arises from mutations that turn these pathways on, constantly. And that's dependent on a pressure, on like a selection pressure because of the lack of these. So giving them extra Wynn will take away that pressure. I don't know, maybe reduce cancer. Obviously, if you put people on them indefinitely, you're going to have uncontrolled growth. But the idea would be that you would put people on until you rebuild the tissue, and then you would take away the Wynn or whatever activating molecules. So I personally don't think it's a concern, but obviously I'm biased, and I get asked this quite a lot at Genentech, as you can imagine, because people are really worried about off-target toxicity.
I think my last question just is maybe I missed something, but the epithelial cells express Frizzled 5 and 6, but the deletion of 5 seems to matter, but 6 not so much.
Yeah, so Frizzled 6 is a non-canonical Wnt receptor, so I didn't get into this sort of aspect of Wnt. So there are some receptors, a canonical Wnt pathway, which I described, but there's also a non-canonical Wnt pathway. And so Frizzled 6 essentially, it binds a specific Wnt 5A, which then brings it together with a completely different set of signaling machinery and signals something else. And so it's not involved in canonical Wnt signaling at all. But you can knock out any of the other ones. But I knocked out Frizzled 6 because at least I knew it was expressed to some degree, but it's not supposed to signal.
Got it. Perfect. Okay. Well, I think then we are actually right on time, and we can thank you again. That was a really beautiful talk, and I do think the translational implications are very exciting. And we'll go over now to Allison Goldstein from the NIH.
Can you see my slides okay?
Looks great.
Perfect. All right, so hello, my name is Allison Goldstein. I'm a previous undergraduate or a recent undergraduate, and I'm currently working at the NIH in Dr. Brant Weinstein's lab. I'd like to thank the organizers for allowing me to present my data today and tell you a little bit about our novel epigenetic reporter line and how I've been using it to study fin regeneration. Oops. And so I want to start out by just reiterating that regenerative ability differs greatly amongst organisms. We know that injury to cardiac muscle or spinal tissue or the loss of an appendage in a human or other mammals will result in long-term fibrosis. Whereas the same injuries in a zebrafish model will be able to fully regenerate. And so models such as the zebrafish act as an attractive model to be able to study these mechanisms underlying regenerative capacity. I am specifically interested in studying regeneration of the zebrafish caudal fin, which is this back tail fin here, which is an excellent model because it's easily accessible and visible. It also regenerates within 2 weeks. You can see the amount of progress that is able to be made here over the first 9 days alone. And this is not a new model system to be able to study regeneration. Multiple teams have already spent numerous years focusing on this and characterizing some of the key events and players such as transcription factors and morphogens that take a significant role in here. What these earlier studies have also emphasized is that there's a significant reprogramming event that has to happen after injury in order to have successful regeneration. And so this reprogramming event is what I'm particularly interested in, and specifically looking at it through an epigenetic lens. We know that other reprogramming events and other events of significant development, such as gamete development and the maternal to zygotic transition, as well as lineage specification, relies heavily on epigenetic modifications, such as DNA modifications, chromatin remodeling, histone modifications, and small RNAs. And so given this, it's not unlikely that the large reprogramming process required for this regenerative ability might also depend on epigenetic modifications, which lends us to ask this question: are there a group of cells undergoing epigenetic reprogramming in this regeneration? In order to do this, we first wanted to develop a tool in order to be able to help us visualize the tissue-specific epigenetic silencing and activating events in a living animal. And so my mentor, Aniket Gore, decided to create an epigenetic transgenic line, or an epitag line. In order to do this, we've placed a CPG island from the DAZZLE gene upstream of an EF1 alpha promoter, a ubiquitous promoter, driving destabilized GFP expression. And we chose specifically to use this destabilized GFP, which has a half-life of 2 hours, because we thought it would be more accurate to reflect the dynamic nature of activating and silencing events. The DAZZLE CPG island is specifically known to be unmethylated and active in adult gametogenesis, however, is targeted for methylation and silencing in adult somatic tissue. And so the idea behind this construct is that In an event when there is no methylation or significant demethylation, we would have expression of GFP. However, when there's methylation to the CpG island, and based on other cell culture experiments, we know that this methylation can spread, we would have spreading of methylation to the nearby promoter region and silencing of our destabilized GFP. We can see this in an early time course series where we have GFP expression at 6 hours post-fertilization, shortly after the maternal to zygotic transition. Then a peak in our expression between 24 and 48 hours, and silencing beginning around 4 to 5 days post-fertilization, when we know lineage specification is occurring and DNA methylation marks are taking place. As a control construct, we've inserted a vector sequence rather than our CpG island. And in a similar time course series, you can see that there is ubiquitous and constant GFP expression, which persists into the adult fish, whereas in our epitag line, there's silencing in the adult somatic tissue, similar to that in a wild type fish. And so before we wanted to try to utilize this in a regenerative model, we first wanted to validate this line further. The first thing we wanted to do was confirm that methylation events to our CpG island in fact influence the neighbor promoter region. To do this, we carried out targeted bisulfite sequencing of the EF1 alpha promoter region, of which I've highlighted in a red box here. And below are the results, where circles indicate cytosine residues and filled circles indicate methylation of those cytosine residues. As you can see, in contrast to our control construct in the top two panels, in our epitag line, there's significant increase in methylation at this EF1 alpha promoter site from 24 hours post-fertilization to 5 days post-fertilization. And so given this, we next want to see if this was associated with any other changes in epigenetic marks. Using ChIP-seq analysis, we saw that there was a significant increase in known silencing histone marks, such as histone 3 lysine 27 charm methylation, and a significant decrease in known histone activating transcription marks, such as histone 3 lysine 9 acetylation. However, there was no change in our control construct. Taken together, this and our DNA methylation data suggests that our CpG island can promote methylation and induce epigenetic silencing of our nearby EF1 alpha promoter region. And so given this, we then wanted to verify that our line is a reporter responding to epigenetic changes. In order to do so, we disrupted epigenetic silencing via chemical treatment, inhibition of enzymes known to be important for epigenetic remodeling, and carcinogenic treatment, all of which resulted in an upregulation of GFP. Thus, we concluded that our line serves as an effective reporter of epigenetic changes. And so given this validation, we then wanted to turn and see how we could utilize this in a regeneration model. At 1.5 hours after an amputation to the caudal fin, there's no expression of our line, as you would expect in adult somatic tissue. However, strikingly, at 1 day post-injury, there is robust GFP expression by the wound site. And this persists multiple days later and begins to become silenced around 3 and 4 days post-injury, and is essentially silenced by 9 days post-injury. If we take a closer look at 1 day post-injury, we see that these GFP positive cells localize around the exoskeletal elements directly below the injury site. Yet at 2 days post-injury, these localize to the tips of the exoskeletal elements. And taken together, we can conclude that this suggests a large-scale epigenetic reprogramming. And so for the last few minutes of my talk, I want to take a turn and tell you a little bit about my ongoing process and current events that I'm doing in lab to try to understand some of these questions that have been raised from this initial analysis. First, what are these GFP positive cells? Specifically, where do they come from in the uninjured fin? And ultimately, where do they contribute to in the fully regenerated fin? To do so, I have taken a single cell RNA-seq approach, and I've collected fin tissue at different time points along the injury and regenerative process. What I plan to do is analyze this single cell sequencing data and try to ask which of my lineages also express my GFP positive transcripts. This would allow me to understand which cells are becoming reprogrammed in this process. If I then utilize a pseudotime analysis at specific time points, but also comparison between different time points along this process, I can get a better understanding of where my GFP cells are coming from and where they might ultimately contribute to in the regenerated fin. However, what this does not tell me is the specific epigenetic changes that are taking place in these regenerating cells. In order to do this, I would like to have a large analysis of a large group of cells. However, I would need to be able to separate my GFP positive cells from my GFP negative cells. those that supposedly are undergoing reprogramming, and those that are supposedly supporting the reprogramming cells. In order to do this, I can separate them via a FACS sorting analysis and have two distinct populations. I can then use bulk RNA sequencing to try to understand the differential gene expression between these two subgroups, and via epigenetic marking experiments such as bisulfite sequencing and chip sequencing analysis, I can get a better idea of the epigenetic marks that are in reprogramming cells versus those that are supporting the reprogramming cells, again, at different time points along the reprogramming regeneration event.
That's good.
And so I just want to finish with a summary of what I've shown you today. First, our reliable epigenetic reporter line that I presented, and how I've utilized this in the regeneration of a caudal fin model. So we have also used this in regeneration processes of fin regeneration, the heart regeneration in the zebrafish in response to injury, as well as injury to the skeletal muscle. And furthermore, I've shown you a little bit about my current ongoing process to identify and track the dynamics of these reprogramming cells, as well as my processes to determine the epigenetic marks precisely in cells undergoing regeneration. Finally, I just want to thank my PI, Brant Weinstein, my direct manager, Annikit Gore, who's spearheaded this project, and my photography advisor, Daniel Castronova. Thank you.
Thank you, Allison. That really is a very exciting tool. And I'm excited to see where you take it too. I was curious, maybe my preface question is basically whether you've done or plan to do any live imaging that's telling you a little bit about the behavior of these cells.
Yeah, that's a great question. So I feel like one of the things that's inhibiting us right now is we're currently looking at the adult fish. So being able to stabilize the adult fish over a long period of time is quite difficult. There are ways to intubate the fish, the alive adult fish, for long periods of time. But what's difficult, because I'm specifically working with this caudal fin, is being able to have the fin stabilized in such a way that it is unable to move, while also allowing for any cell processes that are occurring. We know that in this early regenerative processes, there are plenty of cell migration events that are taking place. So you wouldn't want to glue the fin down per se, but trying to figure out how to keep it stabilized. So we're currently working on that, but it would be very interesting to see.
Awesome. And I guess that sort of brings me up. Oh, wait, let me stop asking my own questions. Monty has a question in the chat. She says, thanks for an excellent talk. Are you planning on applying single cell ATAC-seq or other single cell resolution approaches to study this?
Yes, we are debating whether we want to take an ATAC-seq single cell approach. I know currently there are ways that are offered to have a combination of single cell sequencing and ATAC sequencing at once. We've already carried out our single cell sequencing approach, so we need to go back and see if we want to also do an ATAC-seq approach as well.
And I was also sort of wondering, as pertains to the lifetime over which this is happening and the migration you were mentioning in the imaging side of things, I guess my first question is, how actively are these cells proliferating, and to what extent are you tracking descendants in that initial, partly related to the change in position that you see in those two time points? Are you looking at descendants? Are you looking at cells that have migrated, and to what extent do you think that this reprogramming is happening through courses of cell division through which this mark is inherited, or that it's just kind of happening in one cell that's changing fate without dividing?
I think that's a really good question. I think that we know that though this reporter is able to be used quite early in this reprogramming event, we also know that the significant cell migration and proliferation takes place very early, even 1 hour after injury. And so being able to utilize this reporter at that early of a time stage might be difficult.
Got it.
But in the later time points, you could use.
And maybe that actually makes me think of another question, which is, I love your destabilized GFP to have a strong off rate. What is your on rate? How soon, I guess what do you conclude or guess is the lag between when these marks are actually coming on and when you can detect the GFP?
So we've seen increase in our mRNA folds prior to our protein expression about 4 to 6 hours earlier. Then we see robust GFP expression, but also this is more for purposes of visualization with the eyesight.
Got it. Perfect. Okay. Well, I think that we can close it out there. And thank you so much again, really an exquisite tool and beautiful talk. Thank you.
Thank you.
Okay, and now I'm delighted to introduce Isbran Mesa, who is a postdoc at the University of Calgary.
All right. See if I can fix this here. Sorry, 1 second. All right, whoops, don't delete. And all right, how's that? Does it look good?
Looks good, yeah.
Great, thanks. Thanks, Kara. Thanks to the organizers for inviting me to talk today. And I'm happy to share my project today, looking at sterile injury, inflammation, and repair of the liver. So injuries to tissues that are actually capable of regenerating are going to be repaired from a switch that changes from a homeostatic program to a regenerative program. So as we've heard a lot over the last few days, this regenerative program could possibly be due to the activity of stem cells, or maybe the reactivation of a developmental program, or perhaps something else entirely. But one thing that's very clear is that we don't really know how this program is initiated. And fundamentally, we want to know how tissues actually sense that an injury has occurred and change from this homeostatic program to this reparative program. So one potential regulator of this is actually the immune system, because upon injuries, there is inflammation. And since the immune system plays dynamic roles in sensing the environment and also sensing perturbations, it's really poised to act as a kind of dynamic niche, which could help dictate the behavior of tissues during repair. So I'm mostly trained as a stem cell biologist, but I've joined Paul Kub's lab, who actually talked earlier today, who's an immunologist who developed this really interesting model of liver injury. And luckily, Paul talked earlier today about this model, so maybe I don't have to go into too much detail about it. But what I would say is that we have an interesting model of liver injury in which we use a thermal probe to ablate a small section of tissue on the surface of the liver, which kills the hepatocytes, the endotheliums, and all the other kind of liver resident cells. And these injuries heal over the course of about 7 to 14 days. As part of this injury, there is an accompanying inflammatory response, which Paul talked about earlier. In this example here, we can see that macrophages are highly and specifically recruited to the site of injury. And what was interesting for me is that if you do interfere with this immune response, it's often deleterious for healing. So in this example here, in mice that have macrophages depleted, the injury size is increased in these macrophage depleted cells over time relative to controls. So this suggests that the immune system might be helpful in repairing these injuries, but we don't really know the specific way in which the immune system actually acts on the tissue. One additional advantage of this model that Paul mentioned is that we can actually do intravital imaging on these livers and watch actually how these immune cells behave. So we can visualize different types of immune cells as they enter the damaged region, but also how they interact with these kind of cells on the margin. And these are the cells that are going to ultimately probably regenerate the damaged tissue. So all in all, this is a really great system for studying how inflammation might assist tissue regrowth after injury. Next slide. Okay. So the first thing that I've been doing is to try to kind of characterize the regenerative response from the tissue side, not focusing so much on the immunology side. So here you can see a cross-section of the liver in which one day after injury, we can see this necrotic area, which has been ablated by the burn. And we can start to see CD45 positive immune cells that have begun to invade into this zone. However, there isn't really very much regrowth happening at this time point. So if we look at EDU incorporation, which marks cells going into S phase or proliferating cells, we don't really see any EDU positive cells one day after injury. It's really at the second day where we start to see EDU positive cells, both within the injury, but more importantly, we see these EDU positive hepatocytes. I've marked the hepatocytes using albumin Cree across to a TD tomato reporter. And we can see these marginal hepatocytes right next to the burn that have undergone proliferation. So over the course of the regenerative process, we see if I give repeated doses of EDU every other day to try to label every single cell that ever has proliferated over this time course, we see that many hepatocytes in this regenerative zone immediately adjacent to the burn have undergone proliferation. Now, what's interesting is that these hepatocytes are preferentially located next to the burn and not in this kind of more distal region. So that to me suggests that there's some sort of local factor, which might be emanating out of the burn itself, which triggers proliferation of the parenchymal cells. Obviously, the next question is to ask what pathways might be activated within this regenerative region. So unfortunately, answering this question is not exactly trivial due to some technical factors with working with the liver and specifically with these cells. But mainly because since this is highly localized to a very small area of the liver, I couldn't really do a traditional sequencing experiment using sorting or anything like that. To answer this question, we're planning to do a somewhat new technique for our lab, which is to be conducting a spatial transcriptomic experiment, which has been mentioned a few times during this conference. So we're planning to use the Visium 10x spatial sequencing technology to try to map the transcriptional response to injury. So this is a technology that allows you to mount tissue sections on a special slide, which has these bar-coded spots, which allows you to capture RNA and then using the barcodes map the transcript back to the precise region where they came from. So we're hoping to use this approach to map the regenerative process to these burns. And in theory, we'd like to focus on genes that are elicited within this regenerative region right next to the burn. And hopefully using this data set, we'll be able to identify gene sets and pathways that are going to be activated in these regenerative regions. And then hopefully identify upstream regulators and then try to link that to the inflammatory process. Now, if I can go back to my, trying to understand how the immune system plays a role in this process, as part of my characterization studies, I did quantify the representation of various populations of immune cells within the burn at various time points. And this was motivated to try to identify which cell populations present during very critical periods of regeneration were there and might be candidate sources of cytokines or other growth factors. So I found that neutrophils were highly abundant, as the lab had previously shown many times, as well as various macrophage populations. But one novel cell population were eosinophils, which were highly and abundantly recruited to the site of these injuries. These are intriguing cells as they're potent secretors of various growth factors that could be important in repair, but so far they haven't really been described to be have an important role in injury repair. So I am, of course, being in Paul's lab doing a lot of imaging, and we've set up an imaging capability to image these eosinophils by crossing eotaxin Cree reporter mice to AR6 reporters. And what we can see is that these green GFP positive eosinophils are migrating into the site of these burns. They appear very rapidly after injury. Within 3 hours was the earliest I've looked at. We don't have many of these mice, but in future experiments, we're going to try to look at precisely when they show up. And in other ongoing experiments, we're going to be trying to test what the long-term behavior of these cells are, whether they interact with other immune cells, the parenchymal cells that are adjacent to the burn, and of course do functional testing to see if eosinophil deficient mice have any phenotypes in regrowth. So one thing that I guess that we were asked to talk about what kind of tools we needed to pursue our studies more. Certainly, I think that these transcriptome or spatial transcriptomic experiments will be very important. One thing I didn't really mention is that the resolution of the technology at the moment is not that great. We don't really have single cell resolution. We can kind of see what happens in broader areas of about 50 microns. So as that technology improves, I think that's going to be really, really valuable to try to really understand what's happening in injury repair. But one thing I also wanted to suggest is that we really need better genetic tools to specifically delete, label, or modify cells of interest. I know it's a big point of emphasis for Genelia to really understand how cells are working in tissues. And in order to really do that, we can't really label cells of interest depending on the expression of one key gene. One thing that can be done is by using intersectional genetic tools. So depending on not one, but two genes to drive the expression of maybe a Cre and a flipase, or actually, as mentioned by an earlier talk today, a DRE recombinase. By using these intersectional reporters, we can really hone down on specific cells within certain organs. And I think that will be a really great tool for understanding what these cells are doing in these organs, and also to be able to do functional studies without causing off-target effects. So with that, I'd like to thank Paul and the other members of my lab, also Elizabeth Jacobson for the mice, and also funding, especially from the Damon Runyon. Thanks very much.
Fantastic. Thank you so much, Eisbrand. I am super excited to check back in with you soon and hear the outcome of a bunch of those experiments. It sounds really like it's going in very fun directions.
Great. Sorry, I didn't have much hard data for you, but these are the plans.
It sounds amazing. So we have first a question from Jason Mills. I'm sure that there are signals that come from the burned region and stimulate regeneration, but how can you rule out that the cells in the zone may also be injured from the burn, but just not enough to die, and that your response is you're seeing part of their response to their own injury?
Yeah, I think that's entirely possible. Yeah, perhaps I set it up as being pretty biased, is in favor of that the immune system is what it's doing. And I actually really liked Jason's comment earlier about the fact that maybe dead or dying cells could be secreting factors that are important. One thing I will say, if I look at my early time point here, we do see a lot of necrotic and dead cells at day one, but we don't see proliferation. Perhaps we just haven't had enough time yet, but it's the dynamics of this don't really suggest that if there are just dead cells around, that the cells instantly start proliferating. So maybe there's multiple signals that are needed, but I certainly haven't ruled that out. It will be really interesting to see in our sequencing data set if we do see evidence of cytokines being of acting on these cells, but certainly it's totally possible that the changes in the matrix or secreted factors from dead or dying cells could play a role as well, and that would be really interesting as well.
Great. Jason just had a quick follow-up to say that it takes a while for the cells to retool and enter the cell cycle, and that could be, I think, the source of the lag. But maybe a question I can ask to follow up on that is, have you seen anything that modulates the size of the repair zone? Is it modulated by the size of the injury? Is it different in different regions of the liver, or is it relatively constant?
I've been consistently doing our burns in one zone or one lobe of the liver and in the same zone. I haven't looked in different places. And I've also tried to keep the burn sizes consistent for sure. So I don't really know a specific, I can't really answer that, I guess, at this particular time. Certainly the lab has shown that if you modify different aspects of the immune response, you do modify how quickly the burns do heal.
Okay, and we have another question, which is how specific do you think the eosinophil response is to the burn? Would you think you'd see this in other injury types, like say a punch biopsy or something else that's relevant in this context?
Yeah, that's an interesting question. I would suspect that they probably are recruited to many injuries. Canonically, they're described to be involved in anti-parasitic immunity. Certainly, that's not the case here. I haven't personally looked at other injury models within the liver, such as some of the chemical models or any of those, but that would certainly be of interest as well.
Great. And sort of a related question to this, do you think that you would see, or do you have data to say that you would see the same infiltration of eosinophils without the necrotic area? Is there a way to kind of cut that out or something?
So what would that experiment be? So I would somehow have an injury without
or somehow clear the debris. I don't know.
That's a good question. Yeah, I don't know. I don't know. I think yeah, certainly maybe we could. You know, we maybe it would be interesting to look at like if we could inducibly kill small numbers of cells, like by crossing to maybe a DTR construct and just kill a few cells here or there, so that there isn't this massive necrotic region, and see what happens. That would certainly be of interest.
Great. And Tom just has a comment that there's extensive literature on eosinophils and fibrosis, so it'll be interesting to look at repair and fibrosis in your model.
Great.
Perfect. Okay, so thank you so much. That was terrific. Love live imaging always, so always fun to see. And now we can move over to Bobby Saxton, who is a postdoc at Stanford.
Okay, thanks, Kara. Can you see my screen?
Yes, looks great.
Okay, so thanks, and thanks everyone for hanging around for the last talk of the day. So as Kara mentioned, I'm a postdoc at Stanford in Chris Garcia's lab, studying the role of cytokines in controlling tissue adaptation to inflammation. And I really come at this topic of tissue repair and regeneration sort of indirectly from the observation that's been alluded to a lot today, that many of the molecules that naturally promote repair and regeneration are also very pro-inflammatory, as in the case of many cytokines, for example. And this link between inflammatory signaling and tissue regeneration, of course, makes a lot of sense physiologically. But in terms of developing new therapeutics, it actually presents a pretty significant barrier in that many of the molecules we might want to use to push regeneration also come with a lot of pro-inflammatory side effects that might actually contribute to or worsen disease pathology in the context of things like autoimmunity or chronic inflammation. And a really nice example of this phenomenon, I think, is with the cytokine interleukin 22 or IL-22. And so IL-22 is produced during inflammation by a variety of different lymphocyte populations. And what's really unique about IL-22 is that it acts exclusively on epithelial cells from a variety of different tissues, including the GI tract, as well as the liver and the skin. And on the one hand, IL-22 is very tissue protective. So it promotes things like epithelial barrier integrity, as well as tissue regeneration by acting directly on the stem cells in some of these tissues. But on the flip side, IL-22 can also be very pro-inflammatory as well, in part through the induction of chemokines that can recruit new immune cells into these tissues, as well as the induction of hepatic acute phase response proteins, which are both markers of systemic inflammation, but also can drive inflammatory processes as well. And this sort of dichotomy has also been seen for IL-22 in the context of human clinical trials, where there's been a lot of interest in using IL-22 in the context of things like graft versus host disease or inflammatory bowel disease. And although there is a lot of indication that IL-22 is effective in these contexts, there's also been seen a number of very pro-inflammatory side effects that might be quite limiting here. And so really my question with this has been, can we decouple the tissue protective functions of IL-22 from the pro-inflammatory, potentially pathogenic functions? And so mechanistically, the way that IL-22 works is by engaging a heterodimeric receptor complex on the surface of epithelial cells. And what's very interesting about this receptor is that it simultaneously activates multiple different signaling pathways. So on the one hand, it activates STAT3, which is sort of a homeostatic transcription factor that promotes tissue protection, repair, and regeneration downstream of IL-22. But on the other hand, it also activates STAT1, which is sort of canonically an interferon-induced transcription factor that really drives inflammation. And so what I wanted to ask was whether we could design or develop a version of IL-22 that selectively signals through STAT3, but not STAT1. So in other words, a STAT3 biased agonist of this IL-22 receptor. And so in order to do this, we first wanted to solve the structure of the IL-22 receptor complex, which was not known. And we were ultimately able to do that to 2.6 angstrom resolution. And you can see that ternary complex here with IL-22 in yellow, IL-22R alpha in blue, and IL-10R beta in pink, kind of as it would look sitting on the plasma membrane. And what this structure allowed us to do is to see in molecular detail how IL-22 engages its receptor, so that we could then go in and make really targeted, precise mutations to sort of systematically destabilize the interaction between IL-22 and its receptor subunits in various ways. And what we found that was particularly exciting was that specifically when we made mutations that slightly weakened the interaction with IL-10R beta, we were able to generate molecules that at high concentration still really strongly activated STAT3, but showed very weak activation of STAT1. And you can see that a bit better here, where these engineered variants B1, B2, and B3 all really nicely activate phospho-STAT3, but show little, if any, activation of STAT1. And we also see this STAT3 biased effect in vivo. So we could treat mice with either wild type IL-22 or one of these engineered variants, 22B3 here, and then look at the different tissues, in this case, looking at the colon. And we see that whereas wild type IL-22 nicely activates both STAT3 and STAT1, 22B3 really selectively activates STAT3. And we also see this in the transcriptomics data. So we then looked at these tissues by RNA-seq, again, looking at the colon here. And when we look first at the tissue protective genes that are upregulated by IL-22, we see that 22B3 shows pretty much an identical pattern. So it still strongly upregulates all of these key tissue protection and repair genes. But when we look at the pro-inflammatory genes that are upregulated by IL-22, such as these interferon response genes or TNF pathway components, we see this really complete, almost complete decoupling, where this 22B3 no longer upregulates these pro-inflammatory factors. And to look at this sort of more functionally, in a functional model, we collaborated with Alan Hanash at Sloan Kettering, who sort of discovered the fact that IL-22 can promote intestinal stem cell function and promote regeneration. And so we used some of his organoid models here, and we found quite nicely that this 22B3 is still able to promote the viability of these intestinal organoids following radiation damage. It still upregulates the intestinal stem cell markers like LGR5 and OLFM4. And then looking at an in vivo model of intestinal radiation damage, we see that 22B3 is still just as effective, if not a bit better, at promoting the sort of repair and regrowth of the intestine following this damage. And we also see this in some other tissues. So here looking at the pancreas, we again see this nice decoupling where 22B3 still upregulates these key protective genes, these regenerative islet genes, Reg3 beta, Reg3 gamma, but it shows much weaker induction of this pro-inflammatory chemokine, CXCL1, which is a key neutrophil recruiting chemokine. And then looking at a model of acute pancreatitis here, where we induce pancreatic damage and then look at levels of pancreatic enzymes that are released into the blood, we see that, again, 22B3 is very effective at protecting the pancreas. So from all this, it seems like 22B3 really retains these key tissue protective features of IL-22. But then what was really key is when we looked at the skin and the liver, which are kind of the key sites where the pro-inflammatory side effects of IL-22 are seen in humans, we again see very nicely that 22B3 shows really no induction of these pro-inflammatory factors that are normally strongly upregulated by wild-type IL-22, such as, again, the chemokine CXCL1 and also these pro-inflammatory cytokines like IL-6 and IL-1 beta. And then really critically, when we looked at the serum levels of the acute phase response proteins like serum amyloid A and haptoglobin, which again are kind of key markers of systemic inflammation, we found that just a single dose of wild-type IL-22 really shows a huge increase in these acute phase proteins, whereas 22B3, even at really, really high concentrations, we don't see any increase in these proteins, suggesting that it really loses its kind of systemic inflammatory properties. And so together, we think that using this structure-based protein engineering approach to develop this biased IL-22 receptor agonist, we've really been able to separate the tissue protective functions of IL-22 from the pro-inflammatory functions. And so kind of briefly, just kind of looking in the future directions, we're of course very excited to try to develop 22B3 and use some of these other molecules for clinical use in humans, and also of course see how generalizable these kind of the protein engineering tools are to other molecules that are kind of both pro-inflammatory, but also promote important tissue protective functions. And then we're also very interested in diving a bit more into the mechanism of how these molecules like 22B3 work, and also how they might synergize with other pro-regenerative molecules, like Ahmad was mentioning something like Wynn, which promotes regeneration through an entirely different signaling pathway. And then sort of in terms of tools and the approaches that would be really enabling here, this has again been brought up many times already, but sort of what I would really love to have access to would be sort of models that we can test these types of engineered ligands in human tissues in the context of their full immune microenvironment, where we can see not just how do these tissues affect the epithelial cells or the stem cells, but also how do they change the interplay and the interactions between the epithelial cells and the stem cells with the different types of immune cells. So that's something I would be very excited about. And so with that, I'd just like to thank my mentor, Chris Garcia, as well as everyone in the Garcia lab, also our collaborator, Alan Hanash, and my funding sources, and thanks to everyone for listening.
Awesome. Thank you so much, Bobby. We have questions coming in. One from Tom Wynn. Have you attempted chronic dosing with wild type N22B3? Does the activation of STAT1, STAT3 by wild type 22 or 22B3 change over time?
So yeah, that's a very good question. We're sort of currently working on that in some chronic IBD models. Everything we've done so far has been acute tissue damage. So yes, we are excited to see how it looks in long-term dosing.
Okay, well, just to validate that plan, one of the questions was, what is the outcome of 22B3 treatment in the presence of ongoing injury and inflammation? So people are interested. I also had a question, which was, I guess, partly related to the predictability of what you did. This is sort of wildly exposing my ignorance here, but the mutations that you made, how much of this was, you expected the outcome that you got, or did you make a lot of mutations at these interfaces and see some effect on STAT1, some effect on STAT3, and went with the one that worked? Or essentially how sure were you that this was going to be the outcome when you made these mutations?
Yeah, I mean, not very sure. I mean, essentially the plan was make, partial agonists, and what we've seen with some other receptors, like with IL-10 that I've worked on as well, is when you make partial agonists that are a bit weaker, you see some of the signaling outputs drop off more easily than others. And so we got a bit lucky, I guess, that the one we wanted to keep is the one we kept, right? That STAT3 was sort of more robust than STAT1. And then actually through that finding in our paper on this, we kind of showed the mechanism for why that is, why the STAT3 signal is more robust to lower affinity ligands than STAT1. But yeah, we were pretty confident we could manipulate the signaling in some way, but I guess we got a bit lucky that we could keep STAT3 and lose STAT1. So we weren't confident that would happen.
Perfect. I'm going to super quickly ask this question, even though I'm looking at Janine's face to see how displeased she is. But do you know if there is any variant that is similar to 22B3? Has nature in some way sampled this space already?
Yeah, that's a great question. In the case of IL-22, there's not. There's only one molecule. Interestingly, in the case of some other cytokines, like most notably type 1 interferons, there are many different types of ligands that all naturally act on the same receptors and have slightly different affinities. So we kind of think we are hijacking a mechanism that nature has used, although I'm not sure that it's been used for IL-22 specifically.
Perfect. Okay. Thank you so much, Bobby. And thank you to all of our speakers in this session. I think it really has underlined that, the future is extremely bright, and we're excited to see where your trajectories take you. So now is our opportunity just to close out the whole session. And I want to start by broadening out that thanks to all of our speakers and discussion leaders in this meeting. I think it's, certainly required a lot of input and investment from the panelists, and I think the yield has been very high, but we also certainly appreciate the effort and input that took. I definitely want to thank Janine and her team. Again, I know Mike did it at the beginning, but there is a ridiculous amount of coordination involved in pulling this off, and she does it so seamlessly while also being a delight. So thank you, Janine. And yes, we just sort of, in closing this out, I think want to mention that, I think the intention behind this that we, the organizers, had was to really try and make connections between people and systems and disciplines that might otherwise not be happening, and have really appreciated the way that you've all really leaned into that. I think, I can at least speak for myself to say that I've certainly learned a lot from being put in a different virtual microenvironment than I normally am. And I think we've seen a lot of really cool themes come out through this, relating to sort of the opportunities and limitations of animal models, the need for new tools that let us study dynamics in an in vivo context, the need for specific atlasing and studies of the damage regeneration state specifically to be able to actually identify the therapeutically valuable targets we want. And also the importance of sort of complementing a cell-centric view, like the one I take, with a matrix-centric view and things like that. So I think with that, I will pass it over to Shruti for any last comments.
I think you've covered it all. The only thing I want to say is the one thing that struck me is, despite the fact that folks came from so many different backgrounds, they seem to converge on very similar themes. And I think that I just want to urge everyone to use this opportunity to connect across disciplines. And the other hope that we sort of have is we can bring everyone back together in a few years and see how much of what we've discussed has been implemented, and the progress we've made as a field towards really the question that Mike posed in the beginning, which is how can we have regenerative outcomes in humans? So thank you all so much for your time, for your input. I've learned so much, and it's been wonderful. And I'm going to pass it on to Mike if he's around. Mike?
Yes, I would simply echo the thank you to the Janelia folks, especially Janine and her team. I hope that they appreciate the amount of excitement and demand that people want for new technologies to move the ball forward, because at the end of the day, a child or adult has an injury, whether it's a chemical or physical, and unfortunately, in almost all situations, they don't regenerate. So they need our help, and we hope that these connections persist, as my two colleagues have just said, and we'd like to come back in a couple of years and see where we're at with progress or lack thereof. So I thank everyone, as my co-chairs have, and turn it over to Janine.
Perfect. Thank you, guys. I appreciate it. I agree. It was a great meeting, and I thank you, the organizers, and also all of the speakers and discussion leaders. It was fantastic. So before you run too far away, please join us for the last, the closed panel discussion. Panelists, I'm going to send an email right now with the link for that. And I was thinking, since we're ending so much on time, that we would start that at 4:45 instead of 4:50. So maybe just take 10 minutes for a quick break, and then we will get started with the panel discussion. And to the audience, thank you so much for joining us. You can look at 4dcpjanelia.org if you want to see the future workshops that we have on tap. Thanks, everyone.
Thanks, Janine.
Thanks, everyone. Thanks, Janine.